Hello? Hello?
Patrick, can you hear us? I can hear you.
Can you hear me?
Yes, yes. Can you hear us?
Yes. And what about the co-hosting the questionnaire raising hands? Did you guys permit me to ask that?
You can ask the questions, Patrick. You can let people ask whoever raises the hand.
No, you have to permit me to take that question and I can allow them to talk.
Yeah, you can make him host.
Can you pull it? Yeah, I made it. Patrick, now you are the co-host.
Yeah, let me see. Somebody can raise the hand, please.
People will speak.
No, one else is here.
No, one is there.
There are only 3 people in the Zoom now.
Can you raise somebody hands? Okay.
Not like that. No, not like that.
I know. In the screen for the question, can you please, now let me, okay. Ask, now I should say ask to unmute, right?
Yes. Yeah.
So now you can ask question?
Yeah, we can.
Okay, fine. Now then I can ask. So whoever raised the hand, I will unmute them to ask the question.
Correct.
And what about the Q&A session? Should they going to put the messages? Should I read it or somebody else going to do it?
If they are putting on the Zoom, go ahead and read, Patrick.
Yeah, so okay, I can also read that one. Let me see where is that session? I cannot access the Q&A icon.
It's the chat box.
No, chat is different than a Q&A. Actually, have you guys made Q&A separately?
No, Patrick, this is not webinar. Q&A comes only in webinar because they can't speak there.
Okay, because I know that's fine. I just came from.
People will put it in chat or they can.
Okay, fine. No, Sandeep. Okay, because I just came from Vidyoshi webinar. They had a separate icon. I could read the question because today's speaker was, our divisional chair.
Okay. Yeah, because that is webinar. In webinar, that option is there. This is meeting.
Okay.
And Patrick, can we check your camera? If you turn it on. Oh, great.
You can see, right?
Yes.
Okay, so things are set. Participate. Yes. Okay.
Yeah.
So how many question maximum for 5 or 25 plus 5 minutes?
No, Patrick, 1 or 2. Look at the time, huh? Look at the time.
Maximum
keep
2.
it short.
They feel bad. 2 questions.
No, Patrick. Look,
Next speakers will be.
Everyone asks, we are off on time, we get into trouble. The plenary talks and all.
Okay, so what is the time limit? 30 plus 5 or 25 plus?
25 plus 5.
Okay, fine.
Camera, shall I turn a little bit towards you?
No, because everyone extends, then it accumulates. Huh?
See, again, looking up.
Yeah, because the height.
Okay. Can you close this thing?
You can close.
Until you finish it.
Yeah, now better. Cut also, yeah. Yes, perfect.
So you don't get to Ragarajan, you can't see properly, right?
Is the screen should have a background of 100 years, that screen? No, it's not available, is it?
No, I'll share now. I unshare because we are speaking to you.
Okay.
We'll mute now and stop the video and then share the screen.
Sure. That is fine, right?
Hello, are you able to hear?
Yes.
Sir, we are able to hear.
Can you hear me now?
Yeah, we can hear you.
I can hear you. Should we try sharing my thing just for a trial and then I'll unshare?
Yeah, try.
Yeah, go ahead.
Okay, 1 minute. Hello, are you able to hear?
It's very weak though.
This is YouTube.
Oh.
Okay,
Okay, fine. Yeah, we can see your screen.
Okay, good. Good. So then I think it will be fine. I'll stop sharing.
Okay, fine.
Great, thanks.
Hello, Dr. Mishra.
Good morning.
Hello, sir.
Hello.
Good morning.
Sandeep, is Professor John Bowman already there?
Yeah. I made him co-host here. JP.
The director is also joining.
Yeah.
Hello, sir.
Hi, Nishant.
Hi, sir.
We have 2 more minutes.
Hello, sir.
Hello,
Hello, sir.
Rangarajan.
Hello, sir. Professor Amarasan. Hello, sir.
Yeah.
It's still not sharp image.
Hello, Professor Bhamji. Rao.
Professor Bomji, you can log in. No, could log in, no problem. Okay. Who else is there? Can I just scroll down? More someone else it is there. Professor Usha, you're there?
Hello, yes, I'm there.
Okay, great.
Why?
Just asked, I just want to make sure it's you.
Oh, you want to see me? Is it? Okay,
No,
few more minutes.
no, I just
I'll
want
be
to
in
make
my
sure.
office. No,
Okay.
I'll be in my office in few minutes. But I am online.
Okay, yeah. Should I check whether it is logged? Professor Rangarajan, have you logged in? Govindan Rangarajan, Professor Rangarajan?
Yeah, I am logged in. Yes.
Oh, great. Thank you. Thanks. Okay, we can start. Tell her. Debanjali?
Yes, sir.
We want to start.
Okay, am I audible?
Yes.
Yes, go ahead.
Good afternoon, ladies and gentlemen. I am Debanjali, and I welcome you all to the concluding chapter of the centenary celebrations of the Department of Biochemistry of the Indian Institute of Science. Before we begin, I request you all to kindly put your electronic devices on mute. The department was founded a century ago in the year of 1921 and has remained a forefront for biochemical research in the country since then. The centenary celebration was kick-started on 21st February 2020 with a 3-day conference series graced by many eminent scientists and enlightening talks. Since then, we have hosted a series of conferences to keep the centenary celebrations going. It has been almost a 2-year journey, and now we have come to the final chapter of it. On this momentous occasion, which marks the closure of the centenary celebrations, we are graced with a constellation of luminary scientists who have kindly agreed to give us a glimpse into their wonderful research during the 3-day international conference series from 10th to 12th December 2021. We have a rich biodiversity of lectures ranging from alumni talks to student talks to invited speakers and other plenary talks covering different aspects of the biological world. With that, I would now like to introduce the first chairperson, Professor P.N. Rangarajan, to begin the centenary celebration. Professor P.N. Rangarajan is the present chair of the Department of Biochemistry. He joined the department as a faculty in 1993 after having finished his PhD from IISc and postdoc from Salk Institute of Biological Sciences, USA. He has made some remarkable research contributions which led to the development of an indigenous recombinant hepatitis B vaccine and a candidate rabies DNA vaccine. His current research is focused on the study of regulation of carbon metabolism in the methylotropic yeast, Pichia pastoris. I call upon Professor Rangarajan to give the welcome speech and begin the proceedings of the centenary celebration.
Thank you. Thank you very much, Debanjali. Professor Govindan Rangarajan, Director, Institute of Science. Professor Usha Vijayaraghavan, Dean, Division of Biological Sciences, Indian Institute of Science. Professor M.R.S. Rao, former President, JNCMS ASR. Faculty, colleagues in Indian Institute of Science, students, staff, distinguished speakers who very kindly agreed to our request to speak on this occasion, all the alumni of our department, and all of you who have logged in via Zoom and YouTube. Good afternoon to all of you. It's my pleasure to welcome you all for this 3-day international conference, which is the final event of the year-long celebrations towards the centenary of this department, which was established in Indian Institute of Science in the year 1921. I consider it a privilege to be the chair of this department in this centenary year. During the last 1 year, thanks to the unstating support we received from the alumni, faculty, staff, and students of the department, we have been able to carry out several events. We have organized scientific conferences. We have organized several lectures by distinguished scientists. And except for the inaugural event, which was held in the last February 2020, all other events have to be done virtually because of the COVID pandemic. And I hope many of you have enjoyed all these events. In case some of you have missed out these events, please let us know. All these events are available as YouTube links, and we'll be happy to share it with you all. Now, this conference being held today, tomorrow, and day after was originally planned to be held last year, December, along with an alumni reunion. Since when we started contacting many of the alumni, many of them are very, very enthusiastic to visit the campus and share their nostalgic moments as students in this campus. Unfortunately, this alumni reunion has not materialized so far. Although the COVID guidelines have been relaxed so far, people are still hesitant to join large gatherings, and therefore it will not be fruitful to have the alumni reunion this year. We hope that it will materialize at least next year. Now, several of the alumni have come forward and has participated in the events in the last 1 year. They have also made several generous donations for the centenary celebration events. I thank all of them for their contributions. I wish to inform all those who have contributed to the centenary celebrations of the department that we still have a large sum of money which has not been spent. After this centenary conference, we wish to now start a centenary corpus fund. So all the unspent money will be now transferred to the centenary corpus fund. Although the fund that had in my mind is something around 10 crores, which I feel the interest accruing from this 10 crore corpus fund will be used for maintaining the central facilities of the department, which has become very, very crucial for research activities. But I wish to inform you that we are nowhere near realizing this ambition. And I request the alumni, philanthropists, as well as any of you and well-wishers of the department to help me out in this effort. And before I step down as chairman next year, if I can really establish a centenary corpus fund for the benefit of the department, it will give a tremendous satisfaction as my stint as chairman during these centenary celebrations. Now, during the last 1 year as chair of the department, I have been interacting with several alumni, well-wishers, companies who have interacted with the department. The warmth, love, and affection that they have shared with me and many of our faculty colleagues are amazing. We therefore requested many of our alumni to share their nostalgic moments, experiences when they were students here and in writing, and many of them readily came forward and shared with us in writing their nostalgic moment they spent in the campus. And this is the basis for the reminiscences book that materialized and that we released today by our director. Now, before I request the director to release the book, I call upon Professor Deepankar Nandi, who has spent enormous time and effort in contacting alumni and requesting for these write-ups and putting them together. For almost 1 year, he has worked tirelessly along with Professor D.N. Rao. And this project is the sincere effort of both of them. And I therefore request to give a very brief overview of this reminiscences book. Deepankar, please.
Yeah, thank you, Professor Rangarajan. Is my screen visible?
No.
No. Yes.
No.
Is it
Yes.
visible?
Yes, yes.
Okay, fine. You're able to hear me, I suppose, right?
Oh, you're not sharing the screen.
Okay. All right. Thank you, Professor Rangarajan. Honored guests, faculty, students, and all attendees. Thank you so much for this opportunity for me to discuss a little bit about the book. So let me see. All right, here we go. So the book is entitled Reminiscences, and it consists of several anecdotes or personal anecdotes or stories that different alumni have experienced while they have been over here. So there are several stories, and so what I will be telling is the story behind the stories. So why reminiscences? PhD time is an important phase in any student's life. It's a phase of enormous growth. And therefore, reminiscing about these events can be informative as well as enriching. And multiple such records, not just single ones, but multiple such records encompassing the highs and lows of research and life is an important part of written history. And it was important that we thought that we bring forth, different people into light that brought the department and gave it shape and substance. So in this context, what the book documents are individual recollections of a vast multitude of students, staff, faculty, and well-wishers. So what's important that we did not restrict ourselves to alumni, but we have broadened it to staff as well as well-wishers. And I think that's an important aspect of it. Let me just give you a brief overview of the book. So the oldest record is that of a student who joined the department in 1951. And thankfully, the student has an excellent memory and, crystal clear memory as it was yesterday. So it gives us a glimpse into life in the early 50s. The latest records are by students, and there are some students who have written anecdotes in 2021. So in essence, what the book covers is about 70 years. So out of 100 years, about 70 years of written history is covered in some aspects or the other. So there are a total of 105 contributions, and the book is fairly large. It consists of about 248 pages. So what I'll do is give you just a brief glimpse of these aspects. So, and if you read,
we can't see your screen. Sorry to interrupt.
Yeah. Oh, sorry. I thought it was being shared. Is it?
Is it visible? Visible it is.
It's visible, right?
Yeah.
No, we can't see it.
I can see.
I can see too, but not the screen.
I can see from my office.
Oh.
How about people outside the institute? If someone, Mitali?
No, we can't see it.
Yeah, we are from outside institute. We can't see.
Yeah.
I can see from Maryland.
If you could unshare and share again, it might help.
Okay, sure. I'll do that. It's okay, fine. Sure.
Yeah, now it's showing up.
Yes, it's
now it's showing up.
Yes,
okay.
Thank you.
Okay, great, thank you. Sorry about that. Okay, so let me begin. Let me give you a brief overview. So the earliest record that we have is of a student who joined in 1951, and thankfully, this person has an excellent memory as if it was just yesterday. So it's great because it tells us about life in the early 1950s. We also have students who have written in 2021. So in essence, what the book covers is about 70 years. So out of 100 years, about 70 years of aspects of written history are covered. So we have a total of about 105 contributions, and the book contains about 248 pages. So it's a fairly big book. And if you read the book, you will see, alumni have freely expressed themselves. And what's important is they've discussed their experiences in the departments, which have impacted their lives subsequently. And, they've gone ahead and done many wonderful things, but, they discuss about their life in the department. So what we have had to do is reach out to alumni and in some cases their families. And we are very happy that they have shared these memories with us. And I guess the unique aspect is the large collection of personal anecdotes covering, such a long period in an academic department. I guess that's what gives this exercise a unique touch. Let me briefly discuss some highlights of the book. In fact, I'll cover just 3 to 4. So as one alumnus puts it, that in the early 50s, annual working costs were low, instruments were limited, and so things had to be shared. But as this alumnus points out, that there was at that time lots of water and electricity those days, and today it's the opposite. So again, in those days, refrigerated centrifuges were not available. So what you had to do is take the centrifuge and cool it. So you had to get ice from Yeshwanthpur, and Yeshwanthpur was still available at that time. So you just put ice around it and then cool it to isolate your enzymes and do your assays. So it's something that I thought was useful. In the older days also, there were no computers, so you had to get theses typed. And it got to a point where the person, and usually there was one person who was very good, so all the students would go to this person. And this person started, even though he was not a scientist, he got very good at looking at data. And sometimes would point out, there's a, lack of controls in this experiment, right? So, I thought that was, that was interesting. Another alumnus points out that, after, while giving his colloquium, there was an earthquake. So everybody rushed out of the lecture hall. And then when they assembled back, and then he finished his colloquium, the chair was, perceptive enough to point out and thank the student for delivering an earth-shaking colloquium. So I think, again, so most of the memories that people have shared are happy ones. So talks of famous scientists, of gatherings of the Biochemical Society, picnics, and so on, right? So this is essentially the book. This is the front cover of the book. And there are 3 photographs, and you will see the transition from black and white to color. So this is in 1957. This is the oldest photograph that we got. And we can see Professor Giri here, who is the chairperson of the department. And this is in the main building of IISc. This one is 1976, and the background is the Department of Biochemistry, which is the old Department of Biochemistry. And finally, we've transited on to 2020, and this is the Biological Sciences building. And this was in February 2020 last year. The back cover shows IISc and, of course, our transit to the new building. But the highlight, of course, is the old biochemistry department. Lots of alumni have, fond feelings about the old department, and especially the lecture hall. These are some snapshots from here. This is a conference on carotenoids in 1975. These are some wonderful pictures that we were able to secure. This is the faculty of the Department of Biochemistry in 1989. You could see there were a large number of faculty over here, and some of them were newly joined. This is more recent. This is 2011. And if you see carefully, you can see the previous chair, Professor Jayabhaskaran over here, the current chair, a younger version of the current chair, Professor Rangarajan. You also can spot Professor D.N. Rao, who is the BC Centenary chair. All right. There are lots of photographs, but the one that takes the cake is this one, where a newly joined assistant professor, that's Professor Mahadevan, giving a haircut to a graduate student, and this is Dr. Satyanarayana. So this is a striking one, and our editor, thought this was, the most striking photograph among the ones that we had.
Yeah, they should log in half an hour early, no?
I was thinking that, something like this would be sort of unthinkable in today's world. And also, I wonder what the Office of Lab Safety would think about this. They would certainly frown at this sort, but this is a wonderful photograph from those days. So as I mentioned, usually the photographs are happy memories, and this is from a journal club, and you can see younger versions of Professor Ajit Kumar and Professor Vijaya over here. And again, these are from some functions the Biochemical Society. As far as the younger ones are concerned, the recent write-ups, the energy levels are just striking. And this is from a trip to Kudremukh. And there is another one of students in the Gymkhana. And it's, again, as I said, the energy over there that comes out of the photographs is very striking. And I think the Gymkhana has a special place, especially if you read the younger anecdotes. Okay, apart from the write-ups, we had other ones. So this is a poem, as a photograph, this is a poem, and the person has also taken the photograph. These are silver linings. We have, some very nice poetry. In addition, there are paintings, and this is again by a current student entitled Emancipation. These are rangolis that were taken just a few months back in Diwali 2021. There are lots more of such nuggets, but you will need to read the book in order to both read and then see the images. Okay, it's my pleasant job now to acknowledge a lot of people who were involved in this. Certainly, we thank the contributing authors and their families for sharing their precious memories and talents. The faculty, staff, and students of the Department of Biochemistry for their encouragements. Professor Kaushal Verma for his active interest in this project. Nitya Shukla was our editor, and this was in fact her first job in IISc, and she's done a wonderful job. Shakir Hamid is our experienced designer. He does most of the work for the Office of Communications and was very useful in designing the book. My personal thanks to Raj, Sandeep, and Mahipal who helped. We would meet every week with Nitya to discuss the progress, and the student volunteers for going over the drafts. Thank you all for attending. We hope you will enjoy the book as much as we have enjoyed communicating with all the contributors and putting it together. Thank you so much. Professor Rangarajan, back to you.
Thank you very much, Professor Dipankar, for a very nice overview of the Reminiscences book and also for the efforts you have put in. I now request Professor Govindan Rangarajan, Director, Indian Institute of Science, to formally release the book. Professor Govindan Rangarajan, please.
Thank you, Rangarajan. So let me formally, release the book now. So since it's online, there are no packages to be opened. So let me therefore release it and request all of you to go through it. I went through some parts of it. I found the reminiscences by Dr. P.R. Krishnaswamy very interesting, describing himself as a black sheep, and also Professor Padmanabhan giving the reason why he grew a beard in the first place, because he was mistaken for a student by an assistant registrar. So it's a lot of interesting and humorous anecdotes. I think all of you will greatly enjoy, going through this book. So let me congratulate Dipankar Nandi and D.N. Rao for putting this together. I think they have done a tremendous job, and it has come out very nicely. Okay, thank you all for participating. Thank you.
Thank you very much, Professor Govindan Rangarajan. Thank you. So with us, we come to the end of the inaugural session. I now hand over it to Ms. Sananda to start the scientific session.
Thank you, sir. I would like to take this opportunity to welcome everybody to the first session of the Biochemistry Centenary celebrations. Session 1 would be based on synthetic biology and consists of talks by Professor John Bowman, Professor Irene Coyne, and Professor Sarah Teichmann. This would shortly be followed by the biochemistry alumni and students talk. The upcoming session would be chaired by Professor Patrick De Silva. Before I begin the session, may I please request the audience to mute their electronic devices? I would also request the audience to kindly raise their hand after the end of each talk only, and the respective session chairperson would allow the audience to speak in the permitted time limit. Also, audience can type the question on Zoom and those would be addressed by the chair. For participants in YouTube, please post your questions in the chat box. Professor Patrick De Silva joined the Department of Biochemistry in the year 2007 and is now a professor in the department. His research primarily revolves around investigation of protein folding and understanding mitochondrial biology. I would now like to welcome Professor Patrick De Silva and kindly introduce the speakers of this session.
Okay, thank you, Sananda, for the brief introduction. It's my pleasure to chair the today's synthetic biology session. In this session, we have three speakers, as Sananda said, Professor John from Australia, Professor Irene from Germany, and Professor Sarah from UK. So let me introduce today's first speaker, Professor John Bowman. He is a professor at Monash University, Australia. Professor John did his BS in biochemistry from the University of Illinois, PhD at Caltech with Elliot Meyerowitz, working with Arabidopsis flower development. This is the time, I guess, Professor John overlapped with Professor Usha Ujaya-Raghavan at Caltech. I think he also visited our campus a long time back. He did his postdoc with the David Smythe Monash University 1991-1995, working on flower development. He served as then back in as a faculty University of California, Davis, 1995 to 2006, working on the leaf development. He then moved back to Australia. From 2006 onwards, he is serving as a professor in Monash University, Australia. In 2014, he is selected as a fellow of Australian Academy of Sciences. His present work focuses on investigating fundamental question in land plant development with an evolutionary perspective. So today's talk, he will be talking on why all eukaryotes have homoidomine genes. So Professor John, would you mind sharing your talk with us today?
Hey, good day. I thought you might show it from there. I was going to have a pre-recorded talk.
Yeah, please go ahead.
Today, it's a great honor and a pleasure to be invited to give a talk at the BC Centenary of the Indian Institute of Science. It'd be nicer to be there in person, but I have some history being at Indian Institute of Science. I was there almost 3 decades ago now, working with the lab of Usha Ujaya-Raghavan for a few months, and I have many fond memories from the experience.
Hi, John.
Today I'm going to tell you a short story.
Yeah, Usha is around.
What a rare photograph.
Factors. They're most famous for being involved in the patterning of both animals and plants. In animals, a class of homeobox genes called the Hox class or Antennapedia class specify the identities of segments of the anterior-posterior axis in most metazoans. And in plants, another class of homeobox
We don't hear anything.
Yeah, we can't hear anything, John. Kindly unmute. Kindly unmute.
No, he has recorded it. He needs to increase the volume.
Yeah, probably, yes.
Hi, John. Can you please answer? We'll present from here.
This is inferred because
Can you hear it now?
Yeah, you can increase still volume.
Plants have both Antennapedia class and tail class homeodomain genes. Interestingly, neither bacteria nor archaea have homeodomain genes, suggesting that homeodomains evolved in the ancestral eukaryote. And as such, they must have had some function in the ancestral eukaryote. You know, sometimes predicting gene functions back in time is simpler if the genes encode enzymes that are known to act in very conserved biochemical pathways. But with transcription factors, it would suggest that the transcription factor is regulating some process that was conserved across eukaryotes and was present in the ancestral eukaryote. The story I'll tell you today is primarily concentrating on the tail class box genes, and in plants, they come in two flavors, Knox and Bell. And Knox and Bell act as an obligate heterodimer in most cases. In other words, if you have a Knox by itself or a Bell by itself, they're not active. However, if you have Knox and Bell together, they can heterodimerize, move into the nucleus, and activate gene expression. Some work from Ursula Goodenough's lab in Chlamydomonas some years ago showed that the Knox and Bell genes were involved in the life cycle of Chlamydomonas. And Chlamydomonas probably has a life cycle similar to that could be envisioned from the ancestral eukaryote, in that it has vegetative haploid cells, and then in response to some environmental condition, those haploid cells will be induced to differentiate into gametes. And the gametes of Chlamydomonas are isomorphic. You can't tell them apart. Biochemically, they're different. Minus gametes have a Knox protein expressed, and plus gametes have a Bell protein expressed. Upon gamete fusion, of course, the Knox and the Bell proteins can get together, heterodimerize, and then can immediately activate diploid gene expression. So you can think about this as a way to preload your gametes such that as soon as they fuse, in the diploid, you can initiate completely different genetic programs than were present in the haploid. And in the case of Chlamydomonas, this happens within about 10 minutes of gamete fusion. And some of the genes they activate are genes involved in producing enzymes that remodel the cell wall of the zygote. And in this case, the Chlamydomonas acts as, or the zygote acts as the dispersal agent and the resting stage of the life cycle of Chlamydomonas. Multicellularity, though, has evolved multiple times independently in different lineages of eukaryotes. And we're most familiar, of course, with what happens in animals. And in animals, what happens is that following fusion of the gametes and before meiosis, you have mitoses interpolated there. And then only a subset of the cells in each of the diploid bodies will undergo meiosis to produce the haploid gametes again. However, that's not the only way multicellularity has evolved in eukaryotes. For example, in the algae that are most closely related to land plants, they're called the Periphycean algae. In the haploid phase, you have mitoses interpolated following meiosis and before fertilization or fusion of the gametes. And so this leads to a complex multicellular haploid body in which some cells will differentiate as eggs and others as sperm to complete the life cycle. In the diploid phase of these algae, the zygote does not undergo mitoses, but immediately undergoes meiosis. So it's a single cell diploid phase and a complex multicellular haploid phase. Land plants have probably retained the complex multicellular haploid phase from their algal ancestor, but then they've also evolved a complex multicellular diploid phase by the interpolation of mitoses between fertilization and meiosis. So it has a complex multicellular body in both the haploid and the diploid phases of the life cycle. This is our favorite model organism, Arcantia, where it's dioecious, meaning it has male and female plants. And the female plants produces these sexual structures called archegonia, in which there's an egg cell inside. And the male plants produce motile sperm, which swims over to fertilize the egg cell, give rise to the diploid generation. So this seems like a complex life cycle. Why might it have evolved? One idea is that if we think about the algal ancestor, each fertilization event only produces 4 haploid progeny. And this is not a big problem if aquatic fertilization can occur at most times during the year. However, in land plants, they've retained this aquatic fertilization with motile sperm needing some aqueous medium to swim through to get to the egg cell. However, if they're growing on land, the opportunity for fertilization may be ephemeral, limited to times of flooding or dew. And obviously, if each fertilization event could have given rise to more than just 4 haploid progeny, that would be selected for. And so having a multicellular sporophyte or diploid body here would allow hundreds or thousands of cells to undergo meiosis. So each fertilization event will produce many, many more progeny than if it was just a single zygote. So that was probably the major adaptive reason why why land plants evolved a multicellular diploid generation. The other thing that happened is that the ancestor, just as Chlamydomonas, the dispersal agent is the zygote, whereas in land plants, dispersal agent is now the haploid spore, which allows actually much broader dissemination and many more genetic combinations to likely be available to interbreed. So those are 2 adaptations to land plants, of land plants that probably allowed them to dominate the terrestrial ecosystem starting about 450 million years ago. So we became interested in looking at the Knox-Bell system in our favorite organism, Marchantia, and a lot of this work was done by Tom Dierschke, who's a postdoc in the lab. We first sort of sorted the bells, or the genes by their expression pattern, and we find that some bell genes are expressed in the male, and this Knox gene is expressed in the female. And some of these same bell genes that are expressed in the male turned out to be expressed in the female as well. And 2 other genes are expressed only later during diploid and the sporophyte development. So we'll go through each of these in some moderate detail. So the first question we asked was, is NOX1 provided by the female? If we make an analogy between Marchantia life cycle and Chlamydomonas life cycle, if the egg cell is equivalent to the minus gamete, you'd expect the maternal NOX1 expression here to be required to go through the life cycle. And that is indeed the case. We first looked at expression of NOX1, and it starts in the egg cell. It's also intriguingly expressed throughout the sporophyte, the diploid body, later in development, suggesting that it's involved in the proliferation of those cells. And when Tom deleted the NOX1 locus by genome editing, he saw a phenotype in which the plants never progressed beyond forming a zygote. So this is wild type male sperm and a mutant female, and the cell we know that it's fertilized by changes in the gametophytic tissues that surround the zygote, but it never undergoes any cell division after zygote formation. So NOX1 is absolutely essential to start the diploid generation. We next asked, what about Bell? In analogy with Chlamydomonas, we expected the Bell to be paternally expressed and provide the partner for the NOX1. However, we noted by transcriptome analysis that Bell 3 and 4 are expressed not only in the male, but also in the female. So we asked we also asked what the role of Bell in the female was. And so again, we addressed this question by the same approach, first by looking at expression, and sure enough, Bell is expressed by in situ hybridization. Bell is expressed in the antheridia, and each one of these little cuboidal cells will divide diagonally to produce 2 sperm. And it's also expressed in the egg cell within the archegonia, as well as some tissue surrounding it, and later during sporophyte development. But when we constructed reporter genes, regulatory sequences driving the full-length Bell loci are restricted to the male and are not expressed in the female. However, female expression is driven by an internal promoter here, producing a smaller, shorter protein product, but that can still encode a homeodomain that's expressed in the female there. So we have male and female specific proteins, but both of them have the potential to encode homeodomains. So next, Tom eliminated the activity of all three genes by genome editing. And when he looked at male mutants, so using a wild type female crossed to a mutant male, he found that a large percentage of the embryos were aborted either at the zygote stage or a bit later. However, he still saw some embryos that made it to maturity, suggesting that it's not absolutely required in the male, but it definitely helps. So then he did the reciprocal cross with a mutant female and a wild type male, and basically he got the same result. So this bell in the female is playing a role similar in some ways, at least phenotypically, to the bell in the male. Only when he made a cross between a bell mutant female and a bell mutant male, then you basically don't get any sporophyte development like you see in the NOX1 mutant. So the system seems to be working very similar to what we observe in clammy, although there's a complication with bell being in both, and we don't actually know the precise molecular mechanism by which all these proteins must be interacting post-fertilization to initiate the diploid gene expression. So that just summarizes it there. We next asked what the function of these other two proteins were, and these proteins are only expressed later during sporophyte development, and they're not involved in the transition from the haploid phase to the diploid phase. When Tom knocked out either of these genes by genome editing, he sees the same phenotype, and that is primarily the spores tend to die, and we think that they're not being coated by sporopollenin correctly. And sporopollenin is this incredibly tough biopolymer that's known from fossils 450 million years ago. It's like the most durable biological polymer known to man. And it coats the spores and pollen grains of land plants and protects them from desiccation. So we next did an experiment where we took these two genes that were normally only active in sporophyte development, and we co-expressed them in the vegetative gametophyte and looked for target genes. That is normally only on in the sporophyte, but when you co-express these two genes here, it comes on ectopically in the gametophyte. And we were interested in this particular transcription factor because the orthologs of this transcription factor in flowering plants, such as Arabidopsis, are known to be expressed in the tapetum and activate sporopollenin biosynthesis on the pollen grains themselves. So we looked in our ectopic expression of NOX2 and BEL1 in the vegetative gametophyte, and we could see that genes encoding putative sporopollenin biosynthesis pathway genes were ectopically expressed in this gain of function in the vegetative gametophyte as well, giving us further evidence that NOX2 is somehow regulating sporopollenin biosynthesis. So Jonathan Levins, a honor student in the lab, targeted BHLH by genome editing, and he noted that if you eliminate BHLH37, you basically fail to form sporopollenin, and there's a bunch of other cell wall defects as well. So in wild type, this is a spore where you have layers of sporopollenin here on the outside that is sculpted into two so-called exine layers. It gives you these patterns on the spores. And in the mutant, basically, it looks like the precursors, an electron-dense lipid precursor, accumulates in the spores themselves, but it isn't exported appropriately. And the spores remain in tetrads and fail to coat themselves with sporopollenin, and they're desiccation intolerant. However, if you scoop them out of the sporophyte and provide them a moist environment to germinate, they're still viable. So there's nothing wrong with the spores per se, but rather the coating of them. And this takes us back to what I mentioned earlier about this transition from dispersal agent as a zygote to dispersal agent as the meiotic products, the spores themselves. And a long time ago, the people thought that there was a sporopollenin or a heterochronic shift in the developmental deposition of sporopollenin called the sporopollenin transfer hypothesis. And we think that perhaps NOX2 ancestrally may have been regulating sporopollenin biosynthesis, just as NOX regulates cell wall deposition in Chlamydomonas, and that this interpolation, BHLH37, provided a more complex regulatory network that may have been involved in this heterochronic shift in timing of deposition. So from an earlier slide, I mentioned two major land plant innovations. One was the formation of the sporophyte, so you get many more progeny per fertilization event. And we think that the NOX1 gene with the cognate Bell partners may be involved in initiating sporophyte development and early proliferation of the sporophyte. So that's one of the major innovations of land plants. And we think that the NOX2 and Bell may have retained an ancestral function in regulating cell wall remodeling, but that this has now been shifted to later in development so that sporopollenin biosynthesis occurs around the pollen as well. So it looks like both of these genes are involved in major key innovations in land plant evolution. It also has very, at least the NOX1 system is reminiscent of what happens in Chlamydomonas. And so we think that this is probably the ancestral system in green organisms where you have NOX in one gamete and Bell in the other gamete. And this NOX or this Bell expression in the female may be some sort of innovation of land plants that we're still trying to explain the function of. If we look more broadly, if you remember back to your undergraduate descriptions of yeast, you might recall that their life cycle is very similar to that of the Chlamydomonas, and that A mating type yeast express an A protein, which turns out to be an antennapedia or non-tail homeodomain protein, and the alpha mating type expresses the alpha protein, which is a tail homeodomain, and that these heterodimerize in the diploid to activate diploid expression in yeast. A very similar system occurs in Basidiomycetes, except for this heterodimer actually starts to pattern the initial multicellular development of the mushroom body here. So there's a common theme here where two paralogous homeodomain proteins are expressed in the different gametes to activate diploid gene expression. And more recently, it's been shown that this system occurs also in brown algae. And Mark Koch's group has shown this with two tail homeodomain proteins. And Dictyostelium also uses two homeodomain proteins to activate diploid gene expression. So this is, thinking back to the ancestral eukaryote, this is probably the ancestral system for the transition from haploid to diploid gene expression in eukaryotes. And it provides a rationale why all eukaryotes have homeodomain genes. Perhaps ironically, this system appears to be lost in metazoans. And animals don't really activate gene expression in zygotes, but they do it quite a bit later during mid-blastular transition, or at least at a multicellular stage. However, they have retained homeodomain proteins to pattern their multicellular diploid body. You can think about it, if these genes were ancestrally activating diploid gene expression, they're already expressed in the right time and place for patterning diploid bodies if they evolve to become multicellular. Angiosperms, one of the most studied plant system, also seems to have lost this. And so they've evolved another mechanism to activate diploid gene expression. However, again, they've retained the homeodomain genes to pattern the diploid body because they were co-opted probably quite early in land plant development for that. So I'll end there, and I'll thank Tom Gierschke and Jonathan Levins, who did all the work that I presented, and all our funding comes from the Australian government, the Australian Research Council. And I'll take any questions if you have them.
Okay, thank you, John. Professor John's talk is open for the discussion now. Please raise your hands, so as well as you can drop your question in the chat box.
Can you just turn on the video?
Yes. Put your hand up. Put hand up.
Hi, John. This is Devabruta.
Hey,
Hi,
hey,
it's a nice talk. Maybe I start with the question actually. So you said that this bail. Can you hear me, John?
Yep.
Yeah. So you mentioned that the female can also provide the bail, no? The bail gene.
The female, yeah.
Yeah. So how did you identify that? Actually, I missed that experiment part.
Well, we identified it by transcriptome analysis originally, where you could see a short transcript in the female. And only later, when we mutate it, and you can, because Marcantia is dioecious, meaning it has male and female individuals, you can mutate the genes specifically in the male or in the female. And so we mutated in the female, and you see a phenotype basically equivalent to what we see in the male. So we don't actually know why. I mean, if the Bell gene is already, or the Bell protein, as we know, is already in the female, and the NOX1 protein is in the female, why doesn't it initiate diploid gene expression already? And that we do not know. We suspect that it has something to do with the fertilization event itself. In other words, some sort of biochemical change, whether it be a redox or a calcium change due to fertilization, that you actually get functional complexes formed between these proteins.
Okay, great. Maybe I follow up one quick technical question actually. So normally, when you try to generate the complementary line, so how much promoter fragment normally you're considering? And this I'm always puzzled. Is it like Arabidopsis, we often consider 2 kV promoter fragment. For here, how do you define that, the promoter fragment element in Marcantia?
In Marcantia's genome looks a lot like Arabidopsis, except for all of the genes are just stretched out. So rather than like an Arabidopsis, they cover maybe 5 kb on average. In Marcantia, it's more like 12 kb. And so we cannot just take X number of kb upstream, but rather we have to actually take, we try to have some idea by comparing related species such that you can identify the conserved non-coding sequences, which would then give you a clue to what really might be the regulatory sequences.
How much you should go for it? Yeah. Okay. Absolutely.
Yeah. It could be 1 kb, but it could be 15 kb. You know, it's a problem.
Okay. Great. Thank you so
much. It's not as big a problem as it is in animals, where you might have hundreds of kb. But,
So we have another question from Usha Ujaragan. Please go ahead, Usha.
Hi, John. Fantastic talk, and great to hear you championing Marcantia and its coincidentally, this morning I was speaking to a online, lecture for high school students, and I showed them the picture of the agamus mutant, of the agamus mutant. Anyway, so coming to the question, I was curious to know whether there was any signal dependent dimerization, which you just now speculated could be calcium or something like that, which is a signal dependent dimerization between the tail and the non-tail factor, NOx and Bell, which then triggers downstream gene expression. So that part got answered. So the second part of my question was that, do you think that the female expressed Bell, which is a partial or a truncated protein, has any function? Does it have, because the homeodomains are often in the C-terminal end of the proteins. So does it have a DNA binding domain, and does it actually do something in the female gametophytic tissue?
We think it does. We think it has a positive role. It has a homeodomain. One thing I didn't mention in the talk because of time is that both BEL3 and BEL4 actually have 3 homeodomains, which is basically unknown in other systems.
Yeah, very unusual. Yeah.
However, the short female expressed version would have 1 intact homeodomain. And we think it doesn't act as a poison product. I mean, that's the obvious thing, is that maybe it interacts and it negates the function of the NOx in the egg cell, but we think it acts as a positive way, because when you mutate it, you lose the diploid, the formation of the diploid generation. And so, as you sort of alluded to before, we think that there must be some sort of biochemical change that's initiated by fertilization that enables the interaction. Because if we just take those proteins and put them together, they can interact in a 2 hybrid system or in a BiFC system. One might think about this from another perspective, is we think it might be some sort of, the lack of interaction might be some sort of fail-safe mechanism, such that you need fertilization in order for things to get rolling. Because as a land plant, you only have that 1 egg, and it's a rare fertilization event. And so maybe that there are some safety mechanisms built in to make sure that you really want to go ahead. Whereas in algae, it doesn't matter so much, because you can in theory have fertilizations at any time.
Okay, so thank you. So in the absence of more question, let's thank you, John, for giving an insightful talk on our centenary occasion.
Question.
Okay, who is that? Okay.
This is Utpal.
I couldn't see in your chat raising your hand.
I raised my hand and I started painting, so I put it
Yeah,
up.
please go ahead. Make it quick. Make it small question.
Very quick question. It must be
yeah.
late in the night for you, John.
Yep.
Yeah. So anyway, we learned NOx gene from you. Thank you very much. So is there any organism where there is a single copy of NOx genes?
Only when you go back to algae.
Brown algae, don't brown algae have a single one? This lady
or even green algae, like
even green algae. Okay.
Yeah. So did they diversify in the haploid generation?
So NOx diversified in the Caryophycean algae before land plants.
Okay.
But it's always, as far as we know, it's always been a diploid specific gene. In other words, it's only expressed in the haploid phase, it's only expressed in the gametes. And then it's expressed in the diploid phase.
Okay. Okay, thank you, John, for your insightful talk on our centenary occasion. Much appreciated. We'll move on to the next. Yeah, thank you.
Bye, John.
Cheers.
Bye. Yeah.
Bye.
We'll move on to the second speaker today, Dr. Irene Coyne. She's there? Yeah.
Yes,
Irene is
here.
joined. Yeah, thank you, John, Irene. She is a professor at the Institute of Biochemistry, University of Leipzig, Germany. Professor Irene did her bachelor degree in chemistry at the University of Padua and completed her diploma working with the group of Claudio Toniolo. She did her PhD work at the Leibniz Institute of Molecular Pharmacology, Berlin, Germany. She completed her postdoc work with the Li Wang at the Salk Institute of Biological Studies as a DFG and Marie Curie Fellow. She returned back to Germany 2013 and was awarded a Hemi-Neuther grant by the DFG to set up her independent research group. She won many awards, naming few of them: Hemi-Neuther grant from DFG 2013, German Life Science Award by Gesellschaft for Biochemie Molecular Biology, sponsored by Roche Biodiagnostic, Research Fellowship from Max Buchner Forschung Stiftung 2015. Her research work focuses on mapping protein-protein interaction surface in a live cell by combining chemical biology tools and modern molecular biology techniques. Today, she will be talking on genetically encoded chemical tools for protein studies in the live cell. So, Professor Irene, would you mind sharing your talk with us today?
I think I did.
Okay, go ahead, please.
Okay, so I hope you can hear me and everything is fine. So in case just interrupt me. Thank you so much for this very kind introduction, and thank you for inviting me to tell you about our research here. This is a real year shift compared to the previous talk. So we deal with the much smaller systems, and in a certain sense, I could say very smaller problems that we try to solve also with very small tools. So we are our specialty, our special technique that we have in our lab is the incorporation of artificial amino acids into the proteins using the natural ribosomal machinery. And so just for one of you who are not familiar with this technique, this is the concept is pretty simple. So what you do is that you import in the cell you want to make your experiment in a pair of an orthogonal of amino acyl tRNA synthetase. So the synthetase that recognizes the artificial amino acid you want to incorporate. Sorry. This synthetase charges the amino acid on a tRNA, and this tRNA then incorporates the unnatural amino acids at the ribosome, takes it to the ribosome, which connects us to the peptide chains that's growing. So to do this, we have to recode a codon, because we know that all codons are busy, or 64 codons are occupied. So what we do is that we take the less frequent stop codon, which is the amber UAG stop, and we recode it for incorporating amino acids that don't belong to regular protein synthesis. So which kind of amino acids do we work with? So there are hundreds of amino acids. The technique was developed in last centuries, in the late years. So there were a lot of experiments. And finally, so the first papers where the technique was applied in live cells were at the beginning of the 2000s, and now it's a really mature technique. We have hundreds of unnatural amino acids that can be incorporated in live cells in this way, and we are talking about E. coli, but also mammalian cells, of course. And we can I can show just a couple of selected amino acids. So one important group are photo and chemical cross-linkers. I will tell you right away about those. Then we have amino acids that are dedicated to special chemists that you can do in live cells without interfering with other chemical groups in the cells. Then we have light-sensitive amino acids that can switch conformation with light, or photocaged amino acid that you have a protected group like caging serine, for instance, then you irradiate with UV light and you free this serine. This is important to study signaling, for instance, or something like this. And we have also biophysical probes. So we have nitroxide probes for EPR. We have small fluorophores, IR sensors, and NMR sensors. So this is a bit what's on the market. And as said, there are many more than those. And I will show you what we do with photon chemical crosslinker first, and then depending on time, I will show you about some labeling chemistry in the live cells. So the biological objects we work with in the lab are G protein coupled receptors. So everybody knows. So these are 7 transmembrane receptors in the membrane of the eukaryotic cells that are activated by binding of a signal molecule, so-called agonist. Agonist activates that receptor in that it promotes a conformational change. Conformational change comes, G protein is activated, G proteins leaves the receptors get phosphorylated by G protein coupled receptors kinase, GRK. The phosphorylated receptor is done bound by regulatory molecule, regulatory protein, arrestin, and arrestin stops the G protein signal and starts, in some cases, second signal wave, and importantly, it leads the receptor in the internalization. So this is the paradigm of GPCR activation and desensitization via arrestin. What we did in the first years, we have investigated binding of ligands to GPCRs using photocross-linking amino acids. But this is the old story that you can see in the literature. What I would like to tell you today is a new story about the investigation of interaction of GPCR with arrestin that we are about to publish right now. So the questions is, so why you want to investigate this? So we have about 800 GPCRs that are encoded in our genome, but only 4 arrestins. And these 4 arrestins are not even ubiquitous. So we have 2 arrestins, arrestin 1 and arrestin 4, that are exclusively present in our eyes and regulates rhodopsin and visual opsins. So, and then you have arrestin 2 and arrestin 3 that take care of all other GPCRs. So the question is how? So there is no recognition motif that is conserved in the GPCRs where you would expect arrestin bind there. So there is very, very big variability about GPCRs in the intracellular domain, also in the distribution of the phosphorylation sites. So the question is, how can these 2 arrestin 2 and arrestin 3 recognize so many GPCRs? So when we started this project, there were no structures available of GPCR arrestin complexes. Now we have 3-4 structures. But still, this structure leave many questions open. And the one thing that we thought is that we can explore this interaction using photocross-linking amino acids. So we started with this is a structure of arrestin. We started with substituting a region of arrestin that's called as finger loop, and this is a region that was known from biochemical studies to be involved in the interaction with the receptors with the photocross linker. So we took this BPA, benzoyl phenylalanine, this classical cross-link amino acid. We incorporated 1 by 1 in 24 positions of arrestin, and we checked if we could see cross-linking with the receptor. So are we able to see this complex of arrestin with the receptor? What you do, the experiment is in fact really simple. You express in the same cells. This is all in live cells. All what I'm showing you is all in live cells. You express your receptor, you express the arrestin with the photochemically active amino acids. So with the cross-linker, you activate the receptor, arrestin binds to the receptor, you irradiate with UV light, and then you get the covalent complex. So we tested cross-linking of these photoactive arrestins with 3 receptors: the vasopressin receptor, V2, the parathyroid hormone receptor, PTH, and the corticotropin releasing factor receptor, CRF1. These receptors have this different behavior compared with respect to arrestin binding. So V2 and PTH build stable complexes with arrestin. CRF builds only transient complexes with the arrestin. So that's why we picked a little bit different receptors to see if they are conserved motifs or what happens if we see all these complex. So this was a really explorative experiment. The results are here. So you see, this is the Western blot of the cell lysate at the end of the cross-linking experiment. You see down here, you see the band of arrestin. So this is an antibody that detects arrestin. So you see here the band of arrestin, and you see at high molecular weight the cross-linking bands. And what was immediately clear to us is that every receptor has a different pattern of cross-linking hits. So vasopressin has a very diffuse cross-linking in the finger loop, whereas PTH has only very intense bands in a precise region of the finger loop, while CRF has very weak cross-linking in the finger loop and one strong band at the C-terminus of the finger loop. And also a different behavior is in the segment following the finger loop, where V2 and PTH gave strong cross-linking and CF gives basically no cross-linking. So what we immediately see here is that every receptor seems to give a own pattern respect to arrestin interaction. So there is no conserved pattern of cross-linking between the receptor, and it doesn't matter if stable or transient complexes, and it also doesn't matter the phylogenetic class of the receptor, because PTH and CRF receptors are very phylogenetically very close, but still you see the interaction is very different. And you will be probably irritated by these bands that are out of frame, so they are a different wrong, let's say, molecular weight. We have identified these bands as arrestin dimers, so we have characterized those. These are arrestin dimers. If you're interested in details, you can see our publication. And there are also other bands that disturbed us at the beginning, and so these are, for instance, these bands that you see underneath the molecular, underneath the arrestin. So how is it possible you have a cross-linking product that is lighter than the starting protein? And this is possible because we show that these are intramolecular cross-linking. So it means sometimes arrestin captures itself, and this is why you see also these bands. So once we have established this system, then we explored the interaction of arrestin with the receptor completely. So we took we started from the N-terminus of arrestin and incorporated the PTH, the BPA at all positions of arrestin. So it's about 400 amino acids, and checked cross-linking with all these positions. So this is, you see here in the N-terminus, we have very strong cross-linking position. Then we have a region with no cross-linking. Then we get cross-linking again, and then you go on like this up to the end of the protein. So Yasmin and Tore are PhD students who did this huge work, and what you collect at the end is a map. So it's a footprint of the PTH receptor on the arrestin, you see there are in red, we have highlighted the cross-linking hits, so we know where the PTH receptor touches arrestin. So this is the first information about the interaction. So you see you have here very large interaction domains. This is the finger loop. This is the other side. So this is the same protein. We just rotated it 180 degrees, so we can see it from both sides. So once we have this footprint, of course, it's a pity that we only know the results from the side. We know the hits on the side of the arrestin, but we don't know which residue of the PTH receptor was captured by the cross-linking. So to do so, we apply so-called pairwise cross-linking. So this is different kind of cross-linking. This is not dependent on light. This is really a chemical cross-linking. So you have we incorporate an electrophile in the arrestin. So this is brom alkyl group. So this is electrophilic carbon here, and this is electrophilic enough that a thiol of cysteine can attack it, but it happens only if the two guys are close in space, because otherwise the reaction is not favorable. So this amino acid is stable for pretty a long time in physiological condition, and it reacts only if, because of structural reasons, a cysteine in its proximity, it's fixed in its proximity, and so then you have reaction. So in this case, it's like playing battleship, right? You make a matrix, you substitute electrophile in arrestin and cysteine in the receptor. The reason that this is feasible is that we have the footprint, so we don't need to investigate all positions in arrestin. We need to investigate only positions that gave photocross linking, so the matrix is simplified. Otherwise, it would be unfeasible, would be too many possibilities to check. And in this way, we checked out all possible cross-linking hits, and we got a matrix of the interaction between arrestin and the PTH receptor. In red are a strong cross-linking, in orange are weaker cross-linking, and in yellow very weak cross-linking. Based on these results, so we have tested more than 1,000 combinations, and we have a pool of cross-links that involved 37 position of arrestin, 88 position in PTH. So what do we do? What do we do with this? Sorry, my computer is suffering. I just wanted to show you that we take all these cross-linking hits and we apply those to computational experiments, and we build with this a computational model of the arrestin PTH receptor interaction. This model, which is important, this model is based on the cross-linking constraints. So it's not just a simple docking that you can do with every homology model or so. This is a model that's really guided by the cross-linking constraints. So I see that the PowerPoint is really crashed, unfortunately. So I will restart. I think it's the fastest thing I can do right now. Sorry about that. Oh,
yeah,
no.
It's coming.
Yeah, okay, now, yeah, sorry about this. Sometimes it happens. So I think I will take away the pointer, because this is what makes problems. Okay, so let's so this is the model that we have generated, and you see in green the receptor, in gray is the arrestin, and you see that we can. This is an important aspect. So the blue regions and the yellow regions on the receptor are the regions where we have cross-linking constraints. So we show here how good the model is guided by our cross-linking constraints. So for all colored regions, so we have cross-linking constraint that guide this model. And this is, for instance, very important for this region here. This is the intracellular loop 3, and this is a blind spot in almost all structures of GPCRs. Because it's a flexible region, so you usually don't see this intracellular loop 3 in GPCR structures, and we can see you see this is all yellow. It means we have a cross-linking constraints for almost all positions of it, and these are very well fit in this groove here in the arrestin. So we can see, the first thing we can say, we can definitely see something that the crystal structure and cryo-EM haven't seen so far. This is the first aspect. And then we have a very cool finding about the phosphorylation cluster. So we said the receptor is activated, phosphorylated, bound by arrestin. So how is the situation by PTH receptor? PTH receptor has two phosphorylation cluster. This is a very conserved phosphorylation cluster. It's called key sites. So here are the very important phosphorylation sites that regulates binding on arrestin. There is a second cluster upstream of it, and then you see that in our experiment we can follow all three clusters, and down here there is a single phosphorylation site that we also could follow. So what is exciting about this? What here is exciting is the deposition of the first phosphorylation cluster. And the first phosphorylation cluster is located in a region of arrestin that we decided to call N-edge. So arrestin has two domains, N domain and C domain. Everybody has always spoken about C edge. This is the other edge of arrestin. We won't talk about it right now. And we decided to call this region of arrestin N-edge. What is special with N-edge? We saw that there are here a series of positively charged residues where the phosphorylated the first phosphorylation cluster can be accommodated very, very well. Why is this exciting? This is exciting because this is the second blind spot in the structures that we have. So nobody has ever seen this phosphorylation cluster where it ends up on arrestin. We have some structures of isolated peptide. This is, for instance, peptide from the C-tail from the vasopressin receptor on the arrestin. So this is not full-length receptor. This is only the phosphorylated peptide. And what they found is that phosphorylated peptide, so you see these are the N, this is the N, the key sites here, the C cluster. You see, this is here, that's the same position. And what they see when they take the isolated peptide is that the N cluster is closer to the finger loop. So it would be like it would run up here. But we show that when the receptor is full length, so in the natural environment and where this helix that's very conserved is present, then this helix pulls this part of the peptide toward the N-edge of arrestin, and N-edge is the place where this phosphorylation cluster is located and not closer to the finger loop. So I'm not saying that structure is wrong. I am saying that this might be a possible transient conformation. But definitely we have very many cross-linking hits over here that clearly show that this part of the complex is different from what expected from partial structure with isolated peptides. So if you have structure of isolated domains, always keep in mind that the reality in the full length complex can look different. And as a last note, I can say that with cross-linking, we can see also a bit of dynamic of the complex, which of course is missing in snapshots from cryo-EM and X-ray. So what we see is that we have some cross-linking position in arrestin that work like cross-linking hubs. For instance, here I have collected a couple of examples. Maybe this one is the clearest one. You see K107, this lysine down here has a handful of cross-linking, even more. Then you have D78 that has more than a dozen of cross-linkings. And it's clear to everybody that it is impossible to have 12 amino acids that are so close to a single amino acid at the same time. So because the distance of cross-linking is about 10 Armstrong, C beta, so it's impossible to fit 10 amino acids within 10 Armstrong. It's impossible. So what's the trick here? So what we are seeing here is the dynamic of the complex. If you take our model and you challenge it with molecular dynamics simulation, you will see that all of these positions that give cross-linking within the time of simulation indeed come within the cross-linking distance, but not all together. So, but they come within the time of simulation. So we are also giving a glance here at the dynamic, at the huge dynamic and the plasticity of arrestin. So this is the story that we are preparing now and will be submitting in the next week. And I hope you will be able to read it in some good journals soon. I will take 2 minutes maybe to tell you about another story. This was a story about photo and chemical cross-linkers, and I would like to tell you a short story about click chemistry, about other amino acids that are there to do some chemistry in vivo, some bioorthogonal chemistry, bioorthogonal because it's a chemistry that does not interfere with all the reactive group that are present in a normal cell. So these amino acids for bioorthogonal chemistry, at least the best one, are incorporated by a synthetase and tRNA that comes from methanogenic archaea, is the pyrolysis system. And we were very disappointed to find when we started the work that this system is not really efficient in mammalian cells. So we couldn't get GPCR. Students were angry at me, and we sat down and reasoned why is this. And so we thought that one culprit could be the tRNA of this synthetase, because this is very special. So it has not a canonical, it doesn't have a canonical shape. And so I make a long story short. What we did is that we engineered a new tRNA that allows to incorporate with the big efficiency this kind of amino acid into proteins in mammalian cells. We have shared the plasmid in Edgene. So if you want to do this kind of endeavors, I recommend you can also ask us the plasmid, of course, but Edgene is easier. Then you can get the tRNA that you need. That is very important. And the results are like this. So what we do, we incorporate the unnatural amino acid in the receptor. In this case, is a TCO. What we use is the ones that work best in our hands. So incorporate in the receptor. So you see the receptor is as also in EGFP C terminal. So you see wild type. You see the membrane localization, and then you add a tetrazine fluorophore that reacts with an unnatural amino acid. You see wild type has no labeling, and the receptors have very nice membrane labeled within 2 minutes. So 5 minutes, including one washing step, you are done. So these are all live cells. And what's important here is that if you activate these receptors, you see they internalize, they give these spots. This means they internalize, it means they are still active. They still bind ligand, and they do the physiological response that we expect from them. And the cool thing here is that we labeled a part that nobody else could label before, because what we labeled here are the loops of the receptor. These are very delicate regions that you screw up the receptor activity if you modify these regions. But if you use a very small label on one position, we show here that this is feasible. And we also show that the diffusion rate of these receptors is faster than GFP, for instance, tagged receptors. So this is more physiological situation. And we also show that you can find in the 3 receptor we tested, we could always find one position where we had quantitative labeling. So 100% labeling, and this is very important for quantitative studies, single molecule tracking, dimerization, and so on. Okay, so this was a small overview of our work, how we can apply this genetically encoded chemical tools. We have seen cross-linkers and we have seen anchors for bioorthogonal chemistry to map protein-protein interaction, protein-ligand interaction, and also to label a protein in a single size. Of course, the technique I've shown you here GPCRs, but we have also targets in the lab that are not the GPCRs. It's universally applicable as long as you can express your protein in decent amounts in the organism you're interested in. My group is very small. I've shown you work by Robert Surfling and Tore and Yasmin here, and maybe we have in the future a possibility of showing you. I have a possibility of showing you also other results. We are funding mostly by the German Ministry of Science, or Deutsche Forschungsgemeinschaft, and these are our collaborators for these projects. Thank you very much for your attention, and I'll take questions if you have any.
Okay, thank you, Irene. There are a few questions. Please raise your hands. Yeah, so one question, go ahead. Nagasuma?
Yeah, thanks, Patrick. Yeah, great talk, Dr. Irene. Really enjoyed it. So I was wondering if that N-edge region that you showed in the PTH1R arrestin complex gets ordered upon binding, or is there an order of events that you can,
this is a very good question. Thank you. We will double check on it for our manuscript. As far as we have checked, so we haven't checked details, but this is not this is not one region that is known to get a major changes between the active and inactive conformation. What's known is that the finger loop moves and opens up in a way. And so that's possible that it becomes more accessible. This is possible, but we will double check about it.
Oh, thank you. Is there any major change in stability of the complex?
Not. I mean, nobody knew that there was this binding on the N-edge, and apparently it's very difficult to investigate, because if you take isolated peptide, this doesn't bind the N-edge.
Right.
So, and to do this kind of experiments with the full-length receptor, it's of course problematic. So we don't know this, but this is very good point. Thank you about these questions.
Thank you.
So, any more questions? Okay, in the absence of more questions, let me thank you, Irene, for giving an insightful talk on our centenary occasion, and much appreciated, Irene.
Thank you. Bye.
Thank you. Bye. We'll move on to the last speaker today. So it's Professor Sarah Teichmann. I'm sure she's already logged in. Okay, Sarah is okay. All right, Sarah is here. Okay. Yeah, we can hear you, Sarah. Okay, let me introduce today's last speaker, Sarah Teichmann. Currently, she's the head of cellular genetics. Welcome, Sanger Institute. Professor Sarah did BA, MA Natural Science, University of Cambridge, 1996, PhD, University of Cambridge with Cyrus Chotia, 2000, and she did a short postdoc with Janet Thornton for 2 years, University of College, London. After she was served as a program leader in MRC for 2001 to 2012. And in 2013, she was recruited as combined professor for the Sanger Institute of Wellcome Trust and European Bioinformatics Institute, EMBL. She is now heading cellular genetics at the Wellcome Trust Sanger Institute. She won many awards and honors, naming few of them: Colworth Medal from the Biochemical Society, Francis Crick Medal and Lecture Membership of European EMBO, Lister Prize from the Lister Institute of Preventive Medicine 2012, Suffrage Science Award 2012, Michael and Kate Barnier Award for Young Investigator by the Biochemical Society, Gold Medal from EMBO 2015. She's also elected Fellow of Academy of Medical Sciences. She was elected an ISB Fellow by the International Society of Computational Biology 2016. Recently, she's elected a Royal Society 2020. Her present work focuses on understanding human health and disease by integrating computational and system biology with single-cell approaches, genomics, and immunology. Today, she will be talking on the mapping the human body one cell at a time. So, Professor Sarah, would you mind sharing your talk with us today?
Can you see it?
Yes, I could see it. Go ahead, please.
Great. Thank you so much for the kind introduction. Thank you for having me here. It's a tremendous pleasure and an honor to speak here at the 100th anniversary of the IISc Biochemistry, which is a really fantastic occasion, and I'm delighted to be here. So I'm just going to give a few slides introduction to the International Human Cell Atlas Consortium, and then I'll tell some specific stories about recent insights from cell mapping projects in my lab. So the International Human Cell Atlas Consortium is a project that we kicked off as an international community a few years ago, but it's an idea with a long history. And in his Nobel lecture in 2002, so almost 20 years ago, Sidney Brenner said, we need a program of making maps of cells and how cells talk to each other. The Cell Map project, for which we don't need a model organism anymore, will be one of the things to occupy us for the next few decades. And in this philosophical way, you know, Sidney had a lot of foresight in terms of the International Human Cell Atlas Consortium today. Of course, what he didn't know was what the specific technologies would be that would enable us to map cells at a very high resolution and at full genomic transcriptomic breadth. And those technologies have come along only over the past dozen years or so. And they include single-cell genomics, spatial genomics, high-throughput imaging methods, and advanced computational methods, machine learning, deep learning, other statistical approaches to stitch together the data. And we've reviewed this with the developmental cell atlas community this year in the paper in Nature where this image is taken from. And so it's really those technologies and that resolution revolution down to the single cell level in genomics that sort of catalyzed and buoyed the Human Cell Atlas project. And so with these technologies, what we're doing as a global community is to create a comprehensive reference map of cells from the point of view of curiosity-driven science for understanding our tissues, and also from the point of view of the utility and application to disease. And COVID-19 has already made that very clear that there is a lot of useful utility in understanding where the viral entry receptors are in our body, for instance. So you can think of the Human Cell Atlas as a Google Maps for the human body, where we're coupling single cell genomics and spatial transcriptomics to define the cells and tissues and get down to that Google Street Maps level of detail. And I want to emphasize that this is an open, scientist-driven, bottom-up project where everybody is invited to join, and you can simply sign up here. It's a very simple process. It's open for students, postdocs, PIs, staff members at all levels. It's open for people from any part of the world. We only have 60 members from India. So I really, and there are some in IISc, and I encourage more of you to sign up. It's a very short, simple process, and you'll get information about the meetings, which are all online now and streamed, the newsletters, the updates, the calls for grants, and so on and so forth. So what is nice is that we come from over 79 countries, and we are over 2,000 members now in this community. And together, we've already mapped using single-cell transcriptomics mostly, You can see many hundreds of thousands of cells for major tissues like the heart, the lung, gastrointestinal tract, and so on. And what I want to do in the next section is to talk about give two short insights into projects that have just been published in the past week and months, and where we're learning not about single cells operating in isolation, but about how cells are operating as units cooperating to maintain tissue homeostasis. And I'll start off with zonation of tissue and developmental relationships between cells in the endometrium. And then I'll go on to a development of gut-associated lymphoid tissue in the second part. So in the first part, basically, I want to talk about our effort that came out and made the cover of Nature Genetics. I think it was either this week or last week, where we're making a cell atlas of the human uterus throughout the menstrual cycle. So the endometrium is the epithelial layer of the uterus. It's surrounded by the myometrium, which is the smooth muscle outer layer of the uterus. And the dynamic nature of this tissue means that every month, basically, it's shed and rebuilt. And that's a process that's only present in humans and not the standard lab model of the mouse. And what that means is that it's been quite poorly understood because our mammalian model organism doesn't menstruate. It has a diester cycle. And also it's a dynamic process, so it's more complex. To understand the tissue, you need to have multiple time points, essentially. And so the way we set about this was to use both biopsy from human living volunteers. Oops, computer's asking me for an update. Go away, sorry. And so these biopsies are a couple of millimeters thick. They're taken for diagnostic purposes, but then we use the ones that are deemed to be diagnostically normal, not pathological. And these we can study by both nuclear and cellular RNA sequencing. And then the thicker biopsies that you can see on the left cover the whole of the endometrium and the myometrium, are from deceased transplant donor tissue that we analyze, taken from our colleagues in the clinical school in Cambridge, from the Cambridge Biorepository for Translational Medicine, the Deceased Transplant Donor Program, led by Kaur Saib Parsi, who's our neighbor here in Cambridge in the clinical school. And taking together these two tissue sources allow us to assemble a picture of both the menses, follicular, and luteal stages of the menstrual cycle. And you can see spatial transcriptomics data here of human luteal phase endometrium, where we're seeing these little voxels of transcriptomic data, or about roughly 0.5 millimeter square tissue section. The lumen of the uterus is up here. The myometrium is down here where my cursor is. I hope you can see it. And each little round feature is a 50 micron diameter transcriptomic voxel that corresponds to somewhere between 20 and 100 cells. And we're deconvoluting that transcriptomic signature according to cell types that are determined by single cell and single nuclear RNA sequencing. And so we predict which of these voxels contain glandular epithelial cells. And you can see the red is a higher fraction of the voxel predicted to be glandular epithelial cells. We predict which ones are decidualized stromal cells. So there's a particular fibroblast population that we describe in our paper on the placenta decidua in Nature in 2018, which is close to basically the top, the inside layer here is defined by these fibroblasts during the luteal phase. And then if we look at another fibroblast population, which is the basal fibroblasts, you can see that they're in a completely different location. They are sitting just above the myometrium. And so what this is telling us is that there are different stromal cells, different fibroblast populations in a sort of zonated layering across this tissue. And there's a functional fibroblast population that's in the middle that I'm not showing here. We're also learning that the ciliated epithelial cells are sat here, which is not surprising. They're known to line the inside of the uterus. And then we find a new LGR5 positive progenitor population that we also see located in this luminal location here during the luteal phase. So this is basically just giving you an impression, giving you a feeling of how we are unraveling and deciphering the architecture of this tissue during the different phases. Now, the way we do that, that calculation of where the cells are is using the Bayesian inference framework called cell to location, developed by Omar Bayraktar's group with Oliver Stegle. Vitaly Klevchevnikov drove this project, and you can see that it's taking the input of the single cell RNA sequencing data and using that input to then deconvolute the position of the cells predicted in the spatial transcriptomics data at more coarse-grained resolution. Here it's shown for human lymph node, basically taking single cell RNA sequencing data from tonsil and projecting the T and B cell states onto lymph node. And so that's how we compute and how we map basically the individual cell states onto a more coarse-grained landscape of transcriptome in the tissue. Of course, if we want to get to subcellular resolution or very fine-grained resolution, we need to use imaging as opposed to these 50 micron voxels of spatial transcriptomics. And here you can see the different phases: proliferative, mid-late proliferative secretory, mid-secretory, late secretory, and so on. These are luteal phases. For 4 markers, DAPI, EpCAM, which is marking epithelial cells, and then MMP7, matrix metalloprotease, and PAP. PAP is marking the glandular or secretory epithelial cells, which are doing the secretions to maintain the fetus as it implants during the end of the menstrual cycle. And here you can see that the architecture, what these images show, kind of in increasing resolution, going from top to bottom, and in menstrual stage, kind of going from left to right, is how the architecture of the tissue is changing. So the glands are forming here, and then at the end, they're breaking down again. You can see the PAP and MMP7 expressions of rising up, and then PAP is dominating the epithelial glandular cell expression. And then finally, MMP7 is expressed in the lumen rather than the glands. So there's basically, you can see very clearly how these different markers are marking glandular versus ciliated epithelial cells at the different phases of the menstrual cycle, and how the structure is changing. Now, I've mentioned to you fibroblasts that are in different layers, 3 different layers, from the basal layer to the luminal layer of the endometrium. And those fibroblasts are actually forming the backbone or the majority kind of the cell structure in this tissue. But then we have the epithelial cells that are kind of carrying out the business of the tissue, of making the secretions and moving them along, the ciliated epithelial cells and so on. And those epithelial cells are developing in every single menstrual cycle. And how can we understand how the fibroblasts are talking to the epithelial cells, or whether the fibroblasts are talking to the epithelial cells and helping determine this differentiation and development of epithelial cells that's happening from a progenitor cell in every cycle? Well, you have a database called CellPhoneDB that we published about 4 years ago initially, when we published the placenta decidua cell map. And we've developed that now to include a prediction of cell-cell signaling if cells coexist in the same microenvironment using spatial transcriptomics data, and if they differentially express specific complementary ligands and receptors that are known to interact with each other. And in that way, we can predict high confidence cell-cell interactions via specific signaling complexes. And so for the endometrial data, we predict Wnt activation controlling the ciliated cells and Notch the secretory cells, and so from different signaling from different fibroblast populations. And I won't go into this in detail. This is all published, as I mentioned. But just to summarize, we find that there are specific fibroblasts that are secreting Notch in the functional layer, which is the middle layer, and telling the progenitors to make secretory cells, and that's happening in a Wnt low environment or Wnt inhibitory environment. And on the other hand, in the lumen, we have a Wnt high environment with Wnt7a, Wnt5a, which is then the luminal fibroblasts are instructing progenitors to make ciliated cells via, and so in that sense, you can think of cell atlas data, basically the trajectory from progenitor to differentiated cell state, and the cell signaling, let's say in this case, it's fibroblast epithelial cells, happening in these different microenvironments. So in the glandular microenvironment, the ciliated microenvironment, you can think of it as a recipe for engineering cells in the dish. because we know the specific transcription factors from the cell atlas data in vivo, the specific tissue microenvironment signaling that's happening, and we can then adapt our cell culture conditions accordingly. And this is exactly what we did in this case. We're predicting that from the ex vivo data that Wnt and Notch signaling are important. So we take endometrial primary organoids developed by Margarita Turco in the clinical school, and then add inhibitors, Wnt and Notch, in 2 different cultures, and show that they make new organoids that are characterized by secretory and ciliated cells. So we are engineering specialized organoids that make each cell type, which we can validate by microscopy and single cell RNA sequencing. So in summary, this cell atlas tells us about these different stromal cell populations and the different layers here in gray and beige and brown. So these are the 3 different stromal cell populations that are kind of forming the structure of the tissue in a zonated manner, kind of similar to our skin in some sense. And then the different epithelial cell populations showing for the first time in human, the progenitor cells that are SOX9 positive, and then LGR5 positive and negative intermediate cells, and then the preciliated, ciliated, and luminal epithelial cells, as well as pre-secretory and glandular secretory shells, so shown in these different colors in the glands making the secretions. And this story actually also has a practical utility in understanding disease mechanisms, in the sense that it allows us to deconvolute different bulk RNA sequencing signatures from different stages of endometrial adenocarcinoma and show that the more aggressive stage is LGR5 positive, higher contribution of these cells, so we can classify the tumors in a molecular sense, and also understand that endometriosis biopsies from these aggressive disease where epithelial cells are migrating into other parts of the body is also linked to this progenitor cell population. So the human cell atlasing has implications for engineering cells in the dish. It also has implications for understanding human disease pathology. And I want to thank the amazing teamwork that was a team of teams. It was driven by Rosa Vento-Torma, who's an amazing rising star in the cell genetics program, and she's a former postdoc in my lab and started the project. And Luz Garcia-Alonso and Louis-Francois Hanfield took over the project. Luz is here. Christoph Polanski from my lab. I've mentioned Omar Baraktar, Vitaly Klevchevnikov, and I encourage you to look at the cell to location. And then I also want to thank the Turco lab, the Moffitt lab, the Protein Atlas, the CBTM, and the Newcastle University for the donor material. And I want to thank the donors and the funders, of course. And I can keep on talking, but I realize that we're over time, so I can also skip the next section, which is no problem at all. And I just want to skip on if you want us to finish on time and make up the time. And I will just finish by saying thank you and happy holidays from the Teichmann Lab, and happy birthday to IISc Biochemistry.
Okay, thank you, Sarah. So our talk is open for the discussion. Please raise your hand. Yeah, Nagasuma, please go ahead and ask. Yeah.
Thank you, Sarah. Suma here, and simply fascinating. It was spellbinding. Yeah. So a question from a data analysis point of view. I just wondered if it is possible to pull up orchestrated expression variations between adjacent cells, and whether analogous to a gene correlation protein-protein interaction network, one could think of constructing a cell-cell network to reconstruct a tissue microenvironment.
Yeah, I think that's a fantastic concept, and it's a great way of conceptualizing the intercellular conversation. And there are some pieces of work who are using graph theory to reconstruct cell-cell communication and cell-cell co-localization networks. I think Fabian Theis has had some work in this direction recently within the SquidPy framework and so on. But yeah, I think this is a very fruitful avenue of investigation going forward. Thank you.
Thank you. Thank you.
So any more questions? So I have a simple question, Sarah. I think the single cell transcriptomics, actually, does it really linearly correlate it in terms of proteomics is concerned, in terms of protein expression is concerned?
I mean, we know from bulk RNA sequencing and bulk proteomics data that the correlations are variable. So people have over the last few decades, people have always done these correlations, somewhere between 0.4 and 0.9, depending on how you calculate. For single cell RNA sequencing, the protein data sets are much smaller. They're not protein wide. And of course, we know that, there's obviously, but there have been studies that have tried to correlate protein with RNA at the single cell level on limited, a few tens or a few dozens of proteins. And the most advanced, I think, is the site-seq kind of approaches that have sort of 100 or so proteins, against the transcriptome. And I'd say that the correlations are variable for different proteins, depending on whether there's post-transcriptional regulation. If you compute, what I think is very nice is that if you compute temporal differentiation trajectories, you see the translational, like the lag between transcription and translation as a gene is kicking in, let's say in hematopoiesis, the transcript comes up and then the protein comes up. So I think, overall, I would say that, the conclusions are that RNA is extremely informative with respect to protein, but it's not a perfect correlation.
Okay, thank you. Yeah. So any more question from the audience? One question from a student. Abhinav, go ahead.
Hi, Sarah. Great talk. May I know how many target genes do you see? And by increasing the number of target genes, do you expect to see a greater number of cell types, or was your selection of target genes in such a way that it would cover all the...
So it's not a feature-based, like these are unbiased transcriptomics data, right? So it's just sequencing everything that's there. Based on poly A tail. Poly T primer.
So because I was seeing your single cell data had only, I think, 4 targets, if I'm not wrong. The MMP7.
No. It's a complete 25,000 gene. Like it's not 4. It's a complete transcriptome. Yeah.
Okay, so that brings out only the number of types that you found in your result. Like, is there a possibility that you're missing some kind of cell types in, because you are doing, like you are taking a bulk cell and then you are doing a transcriptomic on it? Is there some kind of heterogeneity in that region also that would further have?
So you're taking like every single cell alone, and you're sequencing the entire transcriptome in every single cell. And so your resolution is limited to the sensitivity of the method. But usually the minimum number of genes that we would get would be 1,000 or 1,500 or so. So you're getting a really good representation of the transcriptome. More sensitive methods can get up to 6,000, like SmartSeq. So what I was presenting was tag sequencing. The spatial transcriptomics is basically, as I said, depending on the voxel and the tissue, it's 20 to 100 cells, but our interpretation of that voxel is based on matched single-cell RNA sequencing data. And I encourage you to read the paper. Maybe it wasn't clear. I encourage you to read the paper that's on the cover. And there's also a News and Views article in Nature Genetics, in this issue of Nature Genetics, that's online at this moment. Maybe it's clear then. Sorry if it was unclear.
Yeah. Thanks.
So any more questions? Okay, and not many. So thank you, Sarah, for a wonderful talk. So to conclude this, thank you. So conclude this session, let me thank all 3 speakers and a big namaste from IISc Bangalore from all of us. Now I request Sukir to take over for the next section.
On behalf of BIX, of this session for the insightful talks. I would also like to thank the chairperson for hosting the session. I now request Anjali to take over from here and proceed with the second half of the first session.
Thank you, Sananda. We will now proceed to our next event, which is Biochemistry Alumni and Student Talks. This session will be chaired by Dr. Savanand Palani and Dr. Dibabrata Laha. Dr. Savanan joined the department in 2020 as an assistant professor. The focus of his lab is to study the organization and dynamics of the actinomycin ring machinery during cell division. The next chair, Dr. Debaprita Laha, also joined the department last year as an assistant professor. The lab is particularly interested in studying the role of inositol phosphates as cell signaling molecules in plants. I would now request the chairs of this session to kindly introduce the speakers.
Hi, thank you very much. So now, we are starting with the alumni session. The first speaker of the session is Dr. Manoj Thakur. So he completed his PhD in 2018 from Professor Muniappa lab. And then he visited Rockefeller Research Laboratories for a short time. And now he's back here again. And his research interest is to identify the holiday junction resolution function catalyzed by YGF protein from Mycobacterium. So now the stage is yours, Manoj.
Thank you very much, sir. And I would like to congratulate the whole department for this nice celebration of excellence in teaching and research. Today, I'm going to talk about a novel ATPase associated with holiday junction resolution in Mycobacterium tuberculosis. So before diving directly into the intricacies of the findings that we have made in this in the presentation that I'm going to deliver, I would like to go first into the brief description of the homologous recombination in Mycobacterium tuberculosis. Much of the information of homologous recombination proteins in mycobacteria are based on the E. coli as a model system. Although, interestingly enough, many studies from different bacteria casts doubt on the universality of E. coli paradigm. Therefore, measures have been taken in our lab to characterize each and every subunit of homologous recombination proteins from Mycobacterium tuberculosis. The pathway commences with REC-BCD protein complexes, which performs end resection precisely at the double strand breaks and lead to the generation of this kind of single-stranded tails. Immense work has been performed from our lab based on by combining molecular, structural, genetic, and biochemical approaches, which suggests that REC molecule coats these single-stranded tails, and they go and then these presynaptic nucleoprotein filaments goes for strand exchange, followed by branch migration, and the final step is resolution of these double holiday junction-like structures. With this, the genetic integrity of the DNA is restored back. Today, I'm going to gloss over the details of RAVX protein, which again is involved at the last step of this holiday junction resolution of homologous recombination, specifically in Mycobacterium tuberculosis. A detailed biochemical bioinformatics studies related to the holiday junction resolvases and other related endonucleases suggested that the holiday junction resolution function has been evolved independently from 4 different structural folds, namely RNase H, endonuclease 7, colicin E, and RASA. Within this RNase H domain, RNase H structural fold or the family, a new set of proteins were discovered, which is nearly ubiquitous in all among all bacteria, and they had the structural folds which is similar to the RAVC family of proteins. These new set of proteins are typified by E. coli YQGF, and similar to the RAVC, this set of protein or this set of proteins from different organism also has the same catalytic triad what this RAVC possesses. So this study highlighted that YQGF family proteins are nucleases with a catalytic mechanism similar to that of RAVC. After this clamorous finding, various studies reported the essentiality of this gene among pathogenic bacteria as well as in E. coli. Other Groups also reported the role of this gene in different processes like transcription, also in the pre-16 sRNA processing, and also in the translation, like the genes which has the Shine-Dalgarno like sequences at the messenger RNA, they are found to be at the low level or low abundance in this YQGF temperature sensitive mutant. None of these reports highlighted the role of this YQGF gene, from which we can say that it is behaving like RAV-C holiday junction resolvase. So when these conclusive or the strong conclusions were being made, Astha identified and isolated this YQGF gene, which we call RAV-X because of the obvious reason that I will let you know in the next slide. And she found that this RAV-X protein exhibited high specificity towards the holiday junction. That means it has the structure selective kind of holiday junction binding activity, and on which it performs dual nicks, which are symmetrically just opposed to each other on 2 arms out of 4 arms of the holiday junction. While doing so, it uses the single cysteine present in the monomeric subunit for dimerization with the formation of stable for the formation of covalent bond. And this is an unprecedented mechanism for the assembly of holiday junction resolvases. While doing her work further, she showed in one of her lab meetings that YQGF gene could complement or could rescue the sensitive phenotype of delta RAV ABC genes under unceasingly jeopardized DNA damaging conditions like UV irradiation, MMS, and mitomycin C. Comprehending over these striking observations, we thought that MT-RAV-X is behaving like RAV-ABC genes. So considering that, we denoted initially this YQGF gene as RAV-X initially. So it has already been shown by Astha that it has the function like RAV-C protein. So we thought to check the RAV-AB-like functions of RAV-X, which is like first we went ahead for checking the ATPase-like function of this RAV-X. So to gain insights further into the interaction of ATP with this MT-RAV-X protein, we incubated the increasing concentration of ATP with the constant amount of RAV-X and radio-labeled mobile holiday junction. And you can see that we were thrilled by looking at this result when we found that ATP actually accentuates the endonuclease activity of holiday junction in a concentration dependent manner. And we did not saw such accentuation when the ATP is replaced by ATP gamma S or ADP. It specifically utilizes DATP or ATP over other NTPases and magnesium as a metal cofactor. In collaboration with Professor B. Gopal MBU, IISc, we solved the crystal structure at 1.95 angstrom, which further validated the presence of RAV-C-like fold and RNase H domain, along with the catalytic triad that is specific to the or belongs to the RAV-C family of proteins. When we compare the crystal structure of MT-RAV-X with the other ATP binding, nucleotide binding domain structures within the NBDB database, we found a putative ATP binding motif, which is highly conserved among HSP70 superfamily members and the chaperone family of proteins. To validate this, the binding of or the involvement of ATP with MT-RAV-X in the resolution activities, we tested the UV catalyzed cross-linking of radio-labeled ATP with MT-RAV-X protein. And you can see here is that the intensity of the cross-linked band of MT-RAV-X increases with the increase in concentration of MT-RAV-X, and the intensity of this band drastically decreases when we challenged with the cold increasing concentration of cold ATP. Our most dramatic and unexpected finding was the intrinsic ATPase activity of MT-RAWX. You can see over here that MT-RAWX displays lower levels of ATPase activity, which is stimulated by the presence of holiday junction, which suggests that holiday junction enhances the catalytic efficiency of MT-RAWX. Based on molecular simulations and homology modeling and docking studies, we identified certain residues, and we subsequently, through site-directed mutagenesis, replaced those residues with asparagine, or in some cases with alanine. And you can see that we have found some mutants with varying degrees of severity in ATPase activity, but D28N, D115, and D142N was found to be completely dead.
No, you have 1 minute.
ATPase activity. Yeah, this is my final slide. Thank you. So and further, it was interesting to observe that this ATPase activity is strongly correlated with the holiday junction resolution activity of MT-RAWX. Those mutants which were dead for ATPase activity was found to be having an abrogated endonuclease activity in them. It was also very interesting to observe here is that in the presence of ATP, it leads to the non-specific nicks or multiple nicks all throughout the holiday junction, in contrast to the canonical holiday junction resolvases or even MT-RAWX when ATP is absent from the reaction mixture. But it is hard to conjecture at this juncture what is the molecular switch which drives the MT-RAWX from being an canonical holiday junction resolvase to non-canonical endonuclease. We have published this recently in JMB, and I acknowledge Professor K. Muniappa and B. Gopal for mentoring while this study was going on, and Disha and Ankur for their interest in crystallographic studies, Ankit for wrapping up with the revision experiments, and of course, biochemistry department and CSIR. Thank you.
Thanks, Manoj, for the nice talk. Are there any questions? So maybe let me start with something. So, can you give us an idea, how this RAWX activity is regulated? What do you know about the regulation of RAWX activity?
Recently, a report has appeared in bioRxiv, and they are suggesting that under DNA damaging conditions, all the DNA repair proteins are getting upregulated, and this RAWX is amongst them.
So it's still not known. So this is something can be interesting question in future.
Yes, for further to explore better.
Okay, great. So now I'd like to ask, thanks, Manoj. So now I'd like to ask Dr. Sravana Palani to take over for the next speaker.
Thanks, Deb. Thank you, everyone. The next speaker for the session is Dr. Mithali Singh. She's currently a postdoctoral fellow for the Institute of Pasteur of Paris, working on epigenetic inheritance using C. elegans as a model system. And she was a former PhD student from Professor Utpal Tatu's lab from biochemistry. Nice to have you, Mithali. Looking forward to your talk.
Thank you for the kind introduction. I'll just quickly share my slide. Can you confirm you can see the slides and hear me well?
Yes.
Okay, perfect. So thank you for giving me this platform to talk about my work on interplay of translation and small RNA biogenesis. And I would like to begin with a brief introduction of the RNA interference, which I'm sure most of you are familiar with, which is a conserved phenomena, and it involves recognition of a double-strand RNA by the dicer protein, which can cleave it to form small RNAs. And the single-stranded small RNAs are loaded by Argonaut to suppress gene expression. As Dr. Palani mentioned, I use C. elegans as a model system, and the RNAi pathway has few additional steps in this system, wherein the primary Argonaut doesn't silence the gene expression, rather it recruits a secondary complex, which includes the RNA-dependent RNA polymerase. This RNA-dependent RNA polymerase can use the target transcript as a template to further synthesize antisense small RNAs, also known as 22G RNAs, and they share 100% complementarity with the target sequence. And these are loaded by secondary argonauts called VAGOs, or worm-specific argonauts, which can carry out transcriptional or post-transcriptional silencing. In the C. elegans germline, the RNAi pathway is functional in special membraneless organelles called germ granules. Most of the proteins also reside in these phase-separated condensates. In the germline, there are two major endo-siRNA pathways. The first one is the pyrRNA pathway, which is involved in the silencing of transposons and genome surveillance. And it functions very similar to the RNAi pathway, wherein it recruits secondary small RNAs to silence the gene expression. There's another important argonaut system, which includes argonaut CSER1. CSER1 is the only essential argonaut in C. elegans. And interestingly, it loads small RNAs which are antisense to all the germline expressed genes. So these are the self genes. We do not know how these small RNAs are triggered, but what we know is they are produced by our RNA-dependent RNA polymerase known as EGO1. The function of the CSER1 argonaut is also quite mysterious. And over the year, it has been proposed that the binding of CSER1 can prevent the binding of PV and thus prevent the silencing of self genes. But more recently, the slicer function of the argonaut has also been demonstrated to degrade certain mRNA targets at specific stages. So we have a lot of open questions about the function of the CSER1 and how CSER1 is able to produce small RNAs against all germline expressed genes, and what are the rules of target selection. For today's talk, I will focus mainly on the biogenesis of the small RNAs. To understand the small RNA profile, in the CSER1 catalytic mutant, we compared the small RNAs with the wild type, and we see a global downregulation of small RNAs in the catalytic mutant on CSER1 targets, suggesting that CSER1 slicer activity is required for the biogenesis of its own small RNAs. What we next did was to look at how these small RNAs are distributed on the target transcript. And in the wild type, the small RNAs are homogeneously distributed throughout the coding sequence. But in the CSER1 catalytic mutant, the small RNAs were enriched at the 3' UTR, but they were depleted on the coding sequence. We further show that these 3' UTR small RNAs are produced by the RDRP EGO1, but it requires CSER1 to cleave the mRNA on the coding sequence to generate a new 3' OHN to continue the synthesis on the rest of the transcript. So we implicate CSER1 slicer activity for the biogenesis of small RNAs, specifically on the coding sequence of its targets. If you remember, I mentioned that most of the RNAi proteins, they reside in these phase-separated granules, which are perinuclear. But CSER1 is unique that it has also a cytosolic localization. So we wanted to understand where this slicer activity is functional and where the biogenesis is happening. And our experiment showed that this biogenesis happens in the cytosol. So this brings about the next question, because as I mentioned, the targets of CSER1 are germline expressed genes. So most likely these mRNAs in the cytosol are engaged in translation. And in fact, our IP mass spec data, as well as polysomal profile, shows that CSER1 can interact with ribosome. And these ribosomes are actively engaged in translation, as can be seen by the enrichment of both CSER1 and the RDRP in the polysomal fraction. So you next wanted to see if, because of this interaction with the ribosome of the RDRP and the CSER1, is it possible to synthesize small RNAs on this translating mRNA? For that, I did riboseq analysis, wherein I cloned the ribosome protected fragments and then mapped the position of the 5' and the 5' end of the antisense small RNAs with respect to each other. And we see a nice periodic pattern with a periodicity of 3 nucleotides, which is the size of the codon when the ribosome is moving on a translating mRNA, suggesting that CSER1 small RNAs are made in phase with ribosomal movement. This was specific to CSER1 pathway because we failed to see such periodicity for small RNAs coming from other small RNAs, PV pathways, for example. But then this brings an important challenge, because the RNA-dependent RNA polymerase moves in a 3' to 5' direction. And CSER1 has to cleave the RNA. And how does this cope up with the translational machinery? So to understand that, we could calculate the translation efficiency of CSER1 targets from our riboseq data. And what we see that the targets which have low level of small RNAs had higher translation efficiency, as indicated by high ribosome occupancy. And as the level of small RNA increases, the translation efficiency decreases, showing an anti-correlation between translation and the small RNA pool. Coincidentally, C7 targets also include a lot of non-optimal codons. And without going into the detail of analysis, what we found that when a ribosome is occupying a non-optimal codon and is paused, that's the place where C7 can cleave the RNA and recruit the RDRP to initiate the small RNA synthesis. But the important question was to how we can test this model. And for that, what we decided to do was to select a C7 target, which has very high level of small RNAs, bad codon usage, and very low translation efficiency. And we codon optimized this gene and got it synthesized and replaced the endogenous gene by CRISPR-Cas9, so without disrupting any UTR-mediated regulation. And then we measured the translation in the small RNA. So in the top panel, you can see the riboseq profile of wild-type KLP7 and the small RNA. On the codon optimized strain, as expected, the translation was increased, which you can see in pink, with a concomitant decrease in the level of small RNAs, thus confirming that increasing translation efficiency can indeed decrease the small RNA production on the C7 target. So to summarize what I have shown you till now is that C7 splicer activity is required on mRNAs undergoing translation to cleave them to recruit the RNA-dependent RNA polymerase to synthesize small RNAs on the coding sequence. And this happens downstream of the ribosome, which has paused on a non-optimal codon. And the level of small RNAs depend on the codon usage and the translation efficiency. Once the small RNAs are produced in the germline, in another independent project, we have shown that they can be inherited in the embryo, where C7 can use these inherited small RNAs to degrade maternal mRNAs, which are no longer engaged in translation. So with this summary, I want to make a small transition. Till now, what I have shown you is the small RNAs during germline development. And then as you all are aware, we were now in a COVID lockdown. So at that point, I started thinking about small RNAs in infection, specifically during the SARS-CoV-2 infection in human. So with a very simple idea to understand what happens to the small RNA profile in the infected cells, we managed to get some infected cells and sequence the small RNA. Here you see the SARS-CoV-2 viral genomic view with the small RNA leads map. And I want to draw your attention to this peak, which increase in intensity with the increased infection duration. And when we look more closely into the reads, what we saw that the predominant read length was of 22 nucleotide, which is the canonical size of a dicer product. We also could detect this small RNA from the patients of COVID-19 and in their swab samples. So we decided to further characterize this viral encoded microRNA. And what we found that the genomic region in the ORF7 of the viral genome from where this microRNA originates.
Mithali, you have less than a minute.
Sure, this is my last slide. Thank you. So the region from where the microRNA is derived can form a stem loop structure, which can be cleaved by the host dicer enzyme, and the mature microRNA can be loaded by the human Argonaut. We could further identify the target sites of viral microRNA in many of the interferon-stimulated genes. And we validated the impact of the viral microRNA on the expression of these interferon-stimulated genes by RNA-seq, and we could see that the viral microRNA could suppress the expression of the target ISGs. So this is quite interesting because if you are aware that impaired interferon response is a key hallmark of severe COVID pathology. So I can conclude that what we show here is that SARS-CoV-2 can encode for a microRNA, which hijacks the host RNAi machinery to evade innate immune response. And with this, I would like to conclude my talk and thank all the lab members, Germano Chetre, who's my supervisor and collaborator, along with the funding sources. And thank you all for your attention. I am now open for questions.
Thank you, Mithali, for a wonderful talk. Is there any questions? No? Okay, there's one question from here.
Hi, Mithali, Raj here. Very nice.
Hi, Raj. Thank you.
I was just wondering, you said binding of the small RNA slows down the process of translation, right?
We don't know if it is the binding of the small RNA. In fact, what we think is it's because the translation is already low. It allows the biogenesis machinery to access the mRNA to make small RNAs. Because we see most of these mRNAs, they have bad codon usage, inherently low translation efficiency.
I was trying to interpret the model that you were showing where the RNA binds to the mRNA and then that is affecting ribosome movement. Was that a point anywhere in the talk?
No, so sorry, maybe I was not very clear. So the model which we propose is that because the codon usage is the one which is actually dictating the translation efficiency. So that is even before the step of the small RNA production. So once you have an RNA which has very bad codon usage and has low ribosomal occupancy, because C7 cannot actually access the mRNA when it is covered by ribosome. So it has developed a mechanism. So whenever there is a pausing, it can access the mRNA to cleave and let RDRP continue to synthesize small RNAs.
All right. Thank you, Mitali.
Okay. You're welcome. Thank you.
Thanks, Mitali.
Thank you.
So now this is time for Dr. Devanjan Sinha, who completed his PhD in 2014 under the supervision of Professor Patrick. And he's a faculty now at the Department of Zoology, Banaras Hindu University. He is a recipient of several awards, including IBA, DST Inspire, etc. And his interest includes, the mitochondrial translocase machinery. Devanjan, the stage is yours.
We can't hear you.
Hi, we can't hear you.
You have to unmute. You are muted.
Can you unmute yourself?
Devanjan, you need to unmute.
Hello, am I audible now?
Yes, yes.
Yeah, okay, thank you. Okay. Sorry for that. Okay, so I thank Department of Biochemistry for providing me the opportunity to present at the part of the Centenary Conference of the department. So I will be talking about some of our work carried out to understand the organization of the human mitochondrial protein transport machinery and its role in cell physiology. So human mitochondria, as we know, is a single organelle with diversified functions ranging from regulation of metabolic pathways, biosynthesis of different molecules, cell death, stress response, etc. To perform all these functions, mitochondria require around 1,500 proteins. However, mitochondrial genome is gene deficient and encodes for only 13 proteins. The rest of the proteins are encoded by the nucleus and is transported into the organelle by well-defined protein transport machineries. Therefore, mitochondria possesses multiple protein transport pathways to import precursor proteins to the different compartments of the organelle. We focused on the P-sequence pathway, which imports the precursors into the matrix and is responsible for importing 70% of the overall mitochondrial proteome. The proteins that are destined to the P-sequence pathway contains an N-terminal key sequence, which is recognized by receptors present on the outer membrane. These proteins are then transported to the Tom complex and is pre-sorted to the P-sequence translocase of the inner membrane. The initial translocation across this pre-sequence translocase is membrane potential dependent, and after which the import motor takes over. Mitochondrial HSP70 forms the core component of the import motor. Mitochondrial HSP70, assisted by its J protein co-chaperones AM60 and AM18, vectorially imports the polypeptide chain into the matrix. But our understanding of the organization of the transport machinery was derived mainly from studies in yeast. But how the human mitochondrial P-sequence translocase was organized was still not known. Therefore, we started our endeavor to characterize the human transport machinery by identifying the first component of the human pre-sequence translocation, that is a J-like protein magmas, which we found to be an ortholog of PAM16. Wild-type magmas could efficiently complement the growth of yeast cells deleted for PAM16, could efficiently import the precursor proteins into the matrix, and form it a stable heterodimeric subcomplex with yeast PAM18 and thereby regulated its mitochondrial HSP70's ATPase stimulatory activity. However, in case of humans, we found the presence of two J protein co-chaperones, DNAGC15 and DNAGC19, along with magmas. Covalent precipitation as well as SPR analysis revealed that magmas form two distinct subcomplexes with both JC15 and JC19, that too with equal affinity. Both the J proteins JC15 and JC19 possessed equivalent stimulation of mitochondrial HSP70's ATPase activity and got similarly regulated by forming a subcomplex with magmas. But knockdown of these J proteins indicated that the expression of JC19 is constitutive and it is essential for mitochondrial function, whereas JC15 was variably expressed and its role was mainly supportive. Therefore, to further understand the functional difference between these two J proteins, we carried out a cross-specific assays. This illustration shows the domain organization of human J proteins isoforms along with their yeast ortholog PAM18. Typically, heterologous expression of human J proteins was associated with a cold-sensitive phenotype. So we tested for the rescue of cold sensitivity and a growth phenotype similar to the yeast wild-type ortholog with a series of mutations that allowed us to identify the amino acid residues that serve as the hotspots for diversification of the proteins inter as well as intraspecifically. Further, we created a series of chimeras by swapping domains across JC15, JC19, and PAM18 and tested for the rescue of cold sensitivity. The results indicated that though J domain is a functional domain in these J proteins, but there are other regions which are responsible for determining the functionality of a J domain in a particular species. Given the unique organization of the human import motor, we went ahead and evaluated the overall subunit organization of the human mitochondrial pre-sequence translocase. In glycerol gradients, the components of each TIM23 complex fractionated into a single peak, which indicated the presence of a single translocase. Whereas the core components of the human pre-sequence translocase, namely TIM50, TIM44, TIM23, and TIM21, fractionated into three different peaks. TIM17B along with JC19 fractionated into two peaks, whereas TIM17A overlapped with JC15 on a single peak. So overall, these results indicated that there are three human pre-sequence translocases, namely translocase A of size 240 kilodalton and translocase B1 and B2 of sizes 450 and 200 kilodaltons respectively. Further, separation of human mitochondria through blue native and second dimension SDS page showed complex organization of human TIM23 complex. Translocase A, formed by the channel subunit TIM17A, segregated into 90 kilodalton and 240 kilodalton complexes, which were overlapping with that of the yeast, whereas translocase B1 and translocase B2 formed the channel component TIM17B. Isoforms formed complexes which were unique to the mammalian system. Further downstream analysis by knockdown of different translocase components indicated that translocase B1 and B2 were indispensable with an essential housekeeping role. Their components were constitutively expressed and they were critical for maintenance of normal mitochondrial function, whereas translocase A was found to be dispensable with its components variably expressed and it was involved in the translocation of pre-sequence lacking proteins. So the mitochondria constitutes the biochemical nexus of the cell and emerged as a crucial component for the biological networking of nuclear pathways. Several cell signaling molecules visit mitochondria to various stages of cell growth, development, and senescence. Since the protein transport machineries form the gateways that connects the cytosolic environment with that of the organelles, Therefore, at present, we are trying to understand its role in hardwiring the mitochondria with the nuclear responses, particularly during cellular adaptation to its environment, in context of tumorigenicity and development. One such example that we have observed is the case of Drosophila testicular stem cell NESH, where knockout of a translocation component caused complete disruption of the NESH and germinal stem cell self-renewing capacity. Gene set enrichment analysis of exon sequenced knockout flies revealed altered expression profile of several genes associated with processes responsible for NESH sustenance. Another observation that we have seen previously was the association of the transport machinery components with functions beyond their canonical role in protein transport. For example, downregulation of DNA GC15 prevented mitochondrial permeability transition for complex mediated cell death. Knockdown of GC15 protected against the constitutive opening of the MPTP channel caused by CYPD overexpression. Similarly, knockdown of cyclin D prevented GC15-induced MPTP activation. Further, we observed that GC15 was responsible for releasing cyclophilin D from the inhibitory effect of TRAP1 and thereby coupling it to the MPTP channel, leading to mitochondrial permeabilization, cytochrome C release, and induction of apoptosis. So at present, we are trying to understand further the role played by the transport machinery components in promoting adaptation to the tumor cells through chemotherapeutic regimens in cell lines as well as in clinical samples. So this is my lab. The Pirashika, Abhijit, and Tanvi are more interested to understand the role of heat shock proteins in tumorigenicity, whereas Olivia and Aishwarya are interested to understand the role of protein transport machineries in development, particularly in maintenance of stem cell NESH. I would like to thank my PhD mentor, Professor Patrick, for being with me to the campaign. Funding from International Science Academy and DBT, and UGC is also acknowledged. Thank you.
Thanks, Debanjan, for the nice talk. Are there any questions? Student can come up, no, with the question. Any question from student? Okay. So maybe let me start with one question, so I might have missed that part. So did you look at the mammalian knockout cell and look at the whole complex, how they're assembled?
Yes, exactly. We did that basically. So we knocked down some of the components actually, and particularly J proteins, and saw how the J proteins are particularly organized as part of the translocation component. But most of the translocase components are quite essential. For example, like TIM17, B, and TIM23. So knockdown are basically lethal for the cells.
Okay, great. Thank you.
But under certain constraints and varieties survives, we did see the how the dynamics of subunit organization changes and all that.
Okay. Okay, great. Thanks, Debanjan. So now I ask Raghunandan to continue.
Okay, thank you.
Thanks, Jeff. So our next speaker is Dr. Leha Manjunath from Professor Sandeep Ishwarapa's lab, and she has completed a PhD with Sandeep, and she's continuing her work as a postdoc in Sandeep's lab. Leha, looking forward to your talk. Thanks.
Thank you so much. Can I confirm that the slides are visible?
Yes.
Yeah.
Thank you.
Yeah,
I'd
but
also
you
like
need to
thank...
put it full screen. Yeah.
Yes. I'd also like to thank the organizers for this opportunity. Today, I'll be talking about stop codon read-through in match to mRNA and its role in mitochondrial physiology. Our lab works on stop codon read-through or translational read-through, which is a process in which translation continues beyond the first stop codon to a downstream inframe stop codon. And hence, from the same mRNA, there are two protein isoforms. One, the canonical protein. Secondly, a read-through protein with a C-terminal extension. This has been seen among various classes of life, with first extensive characterization among viruses to recent studies in mammals. The read-through protein can have properties different from that of the canonical protein. It can vary with respect to localization, efficiency, stability, or function. For example, Professor Sandeep Ishwarappa in his postdoctoral work has shown that VEGF mRNA can undergo stop codon read-through. The read-through protein VEGF-AX is anti-angiogenic in nature, whereas the canonical protein VEGF-A is pro-angiogenic. In the same study, the authors screened for additional read-through candidates. One such candidate, MATCH2, is the focus of today's talk. MATCH2, the mitochondrial carrier homolog 2, is an outer mitochondrial membrane that promotes apoptosis. It also plays a role in mitochondrial metabolism. This was selected as a potential read-through candidate on the basis of the conservation of its proximal 3' UTR. Shown here is the in silico translated 3' UTR of MATCH2 from different mammoths. We observe that following the canonical stop codon, there is a downstream in-frame stop codon, and that the region between these two stop codons, termed the interstop codon 1 or the ISR1, is evolutionarily conserved. We see that in MATCH2, this conservation extends beyond the first downstream stop codon to a second in-frame downstream stop codon, and the region between these two stop codons is also conserved and is termed as the interstop codon region 2 or the ISR2. This led us to the hypothesis that MATCH2 mRNA has the potential to undergo double stop codon read-through. Hence, from a single MATCH2 mRNA, there can potentially be 3 protein isoforms, depending upon the site of translation termination: the canonical protein MATCH2, the single read-through protein MATCH2X, and the double read-through protein MATCH2XX. We carried out luminescence-based read-through assays to look at single stop codon read-through first. For this, the constructs are as follows. We had partial coding sequence of MATCH2 with the stop codon, followed by 45 nucleotides of the proximal 3' UTR, spanning both ISR1 and ISR2. All of this was upstream and in frame with firefly luciferase, such that luciferase activity would be observed only if there is read-through. A control lacking the proximal 3' UTR was used to look at basal read-through, and an additional control had a non-specific sequence instead of the proximal 3' UTR. A construct which did not have any stop codon between MATCH2 and FLuc gave us the maximum luciferase activity. These constructs were transfected in HK293 cells along with the transfection control, Danila luciferase. The relative luciferase activity is a direct indication of the extent of read-through, and we saw significant luciferase activity in the presence of the proximal 3' UTR. RT-PCR analysis showed that these constructs were transcribed at comparable levels, indicating that this difference is at the translational level. We carried out in vitro transcription and in vitro translation using rabbit reticulocyte lysate, and we saw the same results. We replaced luciferase with GFP, and again we saw significant fluorescence in the presence of proximal 3' UTR by both fluorescence microscopy and quantification by flow cytometry, indicating that MATCH2 mRNA can undergo single stop codon read-through. We used similar constructs for double stop codon read-through, in which the test construct had the coding sequence of MATCH2, the canonical stop codon, the first interstop codon region, a downstream stop codon, and the second interstop codon region. In this case, we expect to see luciferase activity only if there is read-through across both the canonical stop codon and the downstream stop codon. A negative control lacked ISR2, and the positive control lacked a stop codon between MATCH2 and FLuc. Again, by both transfection and by in vitro transcription and translation, we saw that there is significant luciferase activity when both ISR1 and ISR2 are present, indicating that MATCH2 can undergo double stop codon read-through. We transfected GFP-based reporters, and again we observed significant fluorescence activity in the presence of ISR1 and ISR2. To get further evidence for the same, we analyzed pre-existing ribosome profiling data. Shown here is an example from a mouse liver. In MATCH2 mRNA, we observe ribosomal footprints not only in the coding sequence, but also in the interstop codon regions, which is absent in the remaining part of the 3' UTR, indicating that ISR1 and ISR2 are translationally active. To summarize this part, we've shown that MATCH2 mRNA can undergo single read-through to form the single read-through protein MATCH2X and double stop codon read-through to form the double read-through protein MATCH2XX. We then wanted to see if we could detect the endogenous read-through products, for which we raised an antibody called the anti-MATCH2X antibody against ISR1. Theoretically, this antibody can detect both the single and the double read-through proteins. When we probed HK293 lysate with this antibody, we saw a band at the expected molecular weight of the single read-through protein. We transfected cells with shRNA against MATCH2, and the reduction in expression was seen with both the MATCH2 antibody and the MATCH2X antibody, indicating that this band is indeed an isoform of MATCH2. We were also able to detect the single read-through protein in different mouse organ lysates, such as heart, kidney, and liver. When we resolved HK293 lysate for long and probed it with an N-terminal match-2 antibody, which can theoretically detect all the match-2 isoforms, we saw that the single read-through protein formed approximately 20% of the total match-2 isoforms. However, we were not able to consistently detect the double read-through protein. And so we analyzed the expression levels of the match-2 isoforms. We overexpressed match-2 isoforms with an N-terminal flag HA tag. For the single and double read-through proteins, the stop codons were mutated to sense codons so that the read-through proteins would be constantly expressed. We saw that the double read-through protein had reduced expression in comparison to the canonical protein and the single read-through protein. However, all of them are expressed at comparable levels at the RNA level. We asked if this reduced expression is because of reduced stability. We treated cells with cyclohexamide, and we observed that both MATCH2 and MATCH2X had relatively longer half-lives. However, MATCH2XX, the read-through product, had a much shorter half-life in comparison to the other two. We wanted to see if this was because it was being degraded. When we treated cells with the proteasomal inhibitor MG132, we saw an increase in the levels of MATCH2XX, indicating that the double read-through product is indeed targeted towards proteasomal degradation. Since MATCH2 is a mitochondrial protein and mitochondrial proteins that fail to localize to the mitochondria are targeted towards proteasomal degradation, we wanted to look at the localization of the MATCH2 isoforms. And so we transfected endothelial cells with GFP-tagged MATCH2 isoforms, and we saw that the canonical protein MATCH2 and the single read-through protein MATCH2X both co-localize with the mitochondria. However, the double read-through protein MATCH2XX is found to be cytoplasmic in nature. To summarize this part, we see that the canonical protein MATCH2 and the single read-through protein MATCH2X both localize to the mitochondria and have a relatively long half-life. However, the double read-through protein MATCH2XX is predominantly cytoplasmic in nature and also has a much shorter half-life when compared to MATCH2 and MATCH2X. We then wanted to understand the functional significance of read-through in MATCH2. For this, we wanted to create cells that were deficient for read-through in MATCH2. We used CRISPR-Cas9 and targeted two sgRNAs in the ISR1 and ISR2, such that there would be a deletion of approximately 70 nucleotides in the read-through region. We confirmed this by means of PCR sequencing as well as by Western blot analysis, which didn't reveal any single read-through protein in the read-through deficient cells. We then looked at the levels of the canonical protein MATCH2 in these read-through deficient cells, and we found an increase of approximately 2 to 2.4 in the read-through deficient cells when compared to the wild type. Overexpression of MATCH2 has previously been reported to lead to mitochondrial depolarization in various cells and tissues. And so we used JC1, a mitochondrial membrane potential dye, and we saw that there is reduced mitochondrial membrane potential in the read-through deficient cells when compared to the wild type cells. To see how this further affected mitochondrial functionality, we looked at oxygen consumption rate using the seahorse analyzer. We saw that there is an overall reduction.
Rekha, you have less than a minute.
Sure. We saw that there is an overall reduction in oxygen consumption rate in read-through deficient cells compared to wild type cells. And linked to this was reduced levels of ATP. We saw that there was reduced expression of the mitochondrial encoded subunits of the oxidative phosphorylation complex, which regulates both ATP and respiration. Finally, we saw that this affected cellular proliferation as well, as the read-through deficient cells proliferated much slower. To summarize our work, we see that MATCH2 mRNA has the potential to undergo both single and double stop codon read-through. MATCH2 and MATCH2X are in the mitochondria with long half-life. MATCH2XX is mainly cytoplasmic. In read-through deficient cells, we see that there is an increase in the levels of MATCH2, which further affects mitochondrial functionality. Hence, we see that read-through not only regulates the levels of MATCH2, but also the mitochondrial function. I'd like to thank Professor Sandeep Ishwarappa, in whose lab I've carried out this work as a PhD student, and also the current and past members of the lab, the Department of Biochemistry, and finally CSIR for funding. Thank you.
Thanks, Lekha. That's a wonderful talk. So let me ask one question. So you have this MATCH2X and the MATCH2XX, right?
Yes.
So the MATCH2XX degrades. That's what you say. With the MG132, you could rescue it, right?
That's right.
So is there any proteasomal recognition sequence which can send out for degradation between 2X and 2XX? That sequence, is there any distraction box or a Ken box sequence there?
Right. We first used the eukaryotic linear motif to predict if there was any degron present there. And indeed, at the very end of MATCH2XX or the ISR2, there is a predicted degron. And when we deleted the degron sequence and tried to express a double read-through protein, there is a rescue in expression. However, we have not identified any specific residue which is leading towards a degradation.
Okay, sure. Thanks. Are there any questions? Okay. There's no further question. Thanks a lot, Lekha, one more time. Let's move on to the next speaker.
Thanks, Lekha, again for the nice talk. Now, this time for Anubhav Dhar. So Anubhav did his BS and MS from ISA Tripurti, and he's a current PhD student at Saravanan Lab since 2020. And his research interest deals with cell division and the proteins that make it possible. We look forward to your talk, Anubhav.
Am I audible?
Yes, yes,
Hello.
yeah.
Okay.
Yes.
I'd like to thank the department for giving me this opportunity to present today. So I'll begin.
Full screen. Can you make it full screen?
Full screen? Yeah, 1 second. Yeah. So good evening, everyone. My name is Anubhav, and I'm a second year PhD student in Dr. Salman's lab at the Department of Biochemistry. Today, I'll be giving a brief talk to give a broad overview of the kind of work we do in the lab, and then about also some other ongoing projects in the lab. So let's briefly revisit cell division first. Cell division is a process where one cell divides into two. It involves the duplication of the chromosome and its separation into two halves of the cell, and then it is followed by a physical separation of the cell, which is called cytokinesis. So our lab basically is interested in cytokinesis. In the cytokinesis, there is a formation of an ectomycin ring in the division plane, which constricts, and then because of the constriction of the ring, the cell is physically separated into two parts. More than 60% of the cytokinetic proteins are highly conserved among the model systems from yeast to mammals, and then a failure in cytokinesis can lead to aneuploidy and also predisposes the cell to a cancerous state. So as can be seen here by a budding yeast mutant, which is cell division defective. So our lab uses two model systems, budding yeast and fission yeast, to study the basic principles that guide cytokinesis. We use methods like isolation and reconstitution of reconstitution of ectomycin systems in vitro, and we also use methods like genetic code expansion from synthetic biology and combine them with traditional methods like molecular biology, genetics, and biochemistry to study some fundamental questions that are left in the field. So, like what is the basic components that are required to make a functional ectomycin, what is the minimum components, and what is the nanoscale organization of those components in the contractile machinery, and what are the temporal kinetics and dynamics of these proteins that are involved in cell division. So let's start by looking at one of the proteins that we're interested in. It's called septins. Septin are a family of GTPase proteins that are conserved from yeast to mammals. They are very important in budding yeast. They localize to the bud site, which is also the division site, the bud neck, and they are formed in basically two structures. They form these two kinds of structures. One is an hourglass structure, where the septin filaments are aligned parallelly to the mother bud axis, and one is a double ring structure where the septin filaments are aligned perpendicularly. So towards from G1 to NFase, they are in an hourglass structure, but during the time of cytokinesis, just before cytokinesis and mitotic exit, they split, the septin structure splits from an hourglass to a double ring structure, and this is the kind of this is what we're interested in studying. So we're trying to study this a transition of septin from an hourglass to a transition and hourglass to a double ring, and how is this transition controlled in space and time. So initially we took a live cell imaging based approach, and we wanted to do the kinetics of all these proteins, the septin proteins and the septin regulators and the septin binding proteins. So here we can see in the montage and the movie that CDC3, which is a septin which is labeled as MCherry, you can see here hourglass structure, and over time it converts to a double ring structure where the transition is happening. So if we plot an intensity profile of the CDC3 MCherry versus time, you can see at t equal to 0, the septin splits into a double ring, and then the intensity of the septin also drastically decreases. So from this point on, we use t is equal to 0, like the septin splitting, as a reference marker for all other septin regulators and inferring their kinetics. So there are kinases, and there are several proteins that localize to the bud neck along the septins. There are kinases, there are scaffold proteins like anilins. So we all looked at all of them using a time lapse, and here is an example of one of those kinases called GIN4. GIN4 localizes to the hourglass structure, but as you can see over time, GIN4 is disappears from the double ring structure. So when the septin transitions from an hourglass to a double ring structure, the GIN4 kinase is leaving. And so we found for most of the kinases that localize the bud neck that most of the kinases are present at the hourglass structure of the septins, but when the septin hourglass transitions to a double ring, they leave the bud neck. But for the aniline-like proteins, which are scaffold proteins, we found a contrasting pattern where they are present both on the hourglass and the double ring structure. So as a conclusion, we were able to figure out the kinetics of all these proteins that localize to the septin structures, and they come in an ordered way, and they also leave in an ordered way. So now we're currently trying to investigate how these kinetics are affected in different septin mutant backgrounds where the septin architecture is defective. But to get to a more mechanistic viewpoint, we are trying also to capture very dynamic interactions, because the interactions are very dynamic and transient and the bud neck at the septin scaffold between these proteins. We're trying two different methods. One is the yeast to habit approach, and another is a cross-linking mass spectrometry approach. Today, because of lack of time, we'll only go to the yeast to habit approach. So we have performed a yeast to habit screen, which will for generating a pairwise interaction map between all the proteins which are present at the bud neck, including the septins, actomycin ring components, and other proteins that are present. So among our yeast to habit schemes, we have many hits, and one of them is a very interesting hit called a protein called BNI5, which is interacting with the septin complex. It is also interacting with the septin associated proteins, the kinases and the scaffold protein, and it is also interacting with the myosin 2 complex, and also with the CLIP2 and the cyclin CDK, the CDK kinase. So this protein interacts with all these three components, and it also mirrors the kinases in a way in its kinetics. As you can see here, that the BNI5 comes to the bud neck or the hourglass stage is present, and when the septin ring splits into the hourglass double ring, it has left the bud neck. So as you can also see in the profile. So from other data, we also know that it interacts with CLIP2 and CDK, and it is also phosphorylated in metaphase and dephosphorylated at the nematocytokinesis. So now we're currently trying to study how this BNI5 regulation and phosphorregulation of BNI5 at the time of mitotic exit is in turn affecting the septin hourglass to ring transition, and how is it affecting the kinetics of other proteins. Now, moving on to another theme that we're interested in as a lab is understanding the localization of cytoskeletal proteins and their dynamics in vivo. So we know that cytoskeletal proteins form end-to-end polymers, which further interact to form further higher order structures. So cytoskeletal proteins form hydrostructures by using oligomerization. So once we introduce a fluorophore tag to a cytoskeletal protein like actin or tubulin, the fluorophore tag interferes with this normal localization, and it is not able to do its proper function in the cell because it disrupts those native interactions which is required for oligomerization. So this problem we try to overcome this problem with using a new approach, using a small tag and a nanobody based against a small tag for imaging. So a nanobody is basically an unconventional antibody which is found in this animal called alpaca. So they don't have any light chains; they only have the heavy chains, and the epitope binding region of the heavy chain, the VHH domain, is called the nanobody. So these are very advantageous because they are much smaller than a traditional antibody, 1/10 of the size. They are extremely stable and sturdy, and they can be expressed and purified in bacteria and can be used for multiple applications. So combining this nanobody with a versatile tag, we chose alpha tag, a tag which was published 2 years back. It was generated by a company called Nanotag. So this is a very versatile tag which can be used to tag any protein of interest on the N terminal, C terminal, or internally, and can be used for multiple applications because it has a paired nanobody present with it. So it can be used for imaging applications and biochemistry applications, and also for applications like cell sorting. So combining this nanobody alpha and alpha tag, we have come up with a new toolkit based alpha tag and nanobody based toolkit for yeast biology. So we have generated a set of plasmids which will have which are expression vectors for expressing this nanobody under different promoter strengths of both fission yeast and budding yeast. The promoters are constitutive and also inducible, and this nanobody will be conjugated to different fluorophores, amnion green, M-charity, or T-tomato, either N-terminally or C-terminally via a linker. So these will be a set of plasmids which will express the nanobody in yeast, and we'll also come up with a set of, we also generated a set of plasmids which will be like a tagging set of plasmids, which will be used to tag any protein of interest on the C terminal with an alpha tag in the yeast genome, genomically. So once these two plasmids will create the toolkit, and this is the concept that we have, that a protein of interest will be tagged with the alpha on the C terminal, and the nanobody conjugated to a fluorophore via linker will bind to the alpha tag and will be able to visualize the localization of the protein using this particular system. So we created these plasmids, and we went ahead and wanted to test out their validity, whether they work in a live cell imaging system in yeast. So here we show that this is SHS1 protein, which is a septin protein, a cytoskeletal protein, where C-terminally tagged this with alpha and expressed nanobody alpha with a conjugated amnion green. We can see that this septin protein nicely localizes to the hourglass septin structure, and also we can see the splitting of the two rings, the septin hourglass to a double ring. We also tested this out for a SPC42 protein, which is a spindle pole body complex, like a mammalian centrosome for yeast. So here also we can see the spindle pole body nicely duplicates in the mother cell, goes to the yeast daughter, goes to the daughter cell, and then the cell divides. So in addition to this, we've also tagged alpha protein. We've also tagged multiple cell division site proteins like myosin 2, aniline, and acceptin interacting protein BNF5, which I showed earlier, and also some cytosolic proteins like mitochondrial DNM1, which is a fission factor, and exocytosis factor called EXO84 with the alpha tag, and all of them are showing a good localization as expected. So this demonstrates that the system works in yeast in vivo, and we're able to check the localization of these proteins using an antibody alpha conjugated with a fluorophore, and then the protein of interest conjugated with the alpha tag. So we get a good signal to noise ratio without any phenotypic effect and no cell cycle delay or any off targeting. So we're now currently planning to extend this to various other proteins, various cytoskeletal proteins, and also to extend this toolkit to biochemistry applications like Western blotting and co-immunoprecipitation. So as a summary, we're studying septin architecture regulation in the lab, where we have shown that the hourglass to ring transition, double ring transition of the septins accompanied by sequential removal of various proteins from the division site in an ordered manner and during mitotic exit. And now we are trying to study this kinetics in a septin mutant background, and we also use a yeast to hybrid skin approach to find a nice target called BNI5, which can be a major candidate protein that may regulate the septin SDR transition. And finally, we have generated a functional alpha tag and alpha nanobody based toolkit for genomic tagging of yeast proteins, which can be used for live cell imaging and other biological applications. So we have demonstrated the live cell imaging application for both some cytoskeletal and some cytosolic proteins, and we're currently optimizing it for biochemistry applications. So I'd like to thank Wazif, who's a postdoc in the lab, for helping the septin regulation project, and our lab manager Sushmita and other members of the lab. I'll also like to thank our collaborators and our funding sources. And we'll especially like to thank the DST FIST microscope facility of the department and the operators Pooty. And so that's where my talk finishes. Thank you all for listening. And if you have any questions or comments, I can take them now.
Thanks, Anubhav, for the very nice work and the nice presentation. Are there any questions from student? Maybe I can start with Anubhav. So you did this, histo hybrid screen, and you found a lot of candidates. Do you have an idea how do you study the functional, the consequence? I mean, what is the physiological importance of this interaction? Do they localize at the same place? Are you trying to make some double knockout or things like that?
Yeah, based on the histo hybrid screen, we have identified lots of hits, and based on that, BNI5 is one of the hits. So we'll try to characterize each one interaction individually and see what it does to how it affects the septin architecture regulation, how it affects the localization of other proteins at the bud neck.
Okay, I'm not sure of this, the whole system. One more question I have. So how does this BNI5 mutant look like?
So the BNI5 mutant is actually, if you delete BNI5, it is not an essential gene. If you delete BNI5, the septin architecture is disrupted. So it's already a protein that is involved in septin architecture, implicated in septin architecture determination. But now we have identified this. It also interacts with these three particular components of the bud neck, and then it also gets phosphoregulated, which we could not show the data here because of the lack of time. But it also gets phosphoregulated, like it gets phosphorylated metaphase and dephosphorylated cytokinesis. So we're also currently trying to study what is the implication of that phosphorylation and a function in septin regulation.
Great, nice. Is there any question?
Sir, can I ask?
Yeah.
So, Anubhav, can you hear me?
Yes, sir.
So what is the level of expression of these nanobodies? Is it in nanogram, microgram amounts?
So currently we have not exactly checked using a Western blot. We just tried for the live cell, but then we have these expression vectors with different set of different promoter strengths. So we have promoters like ADH, which are less strong, and promoters like GPD and TEF, which are even stronger. And we also have constitutive promoters and inducible promoters.
Yeah,
so we're trying.
Can you make a statement that this is the ideal level of expression?
Sorry, I couldn't hear you, sir.
Using all these different promoters, can you say this should be the ideal level of expression for the nanobody expression? What should be the ideal level of expression?
Yeah, ideal level of expression, I think, will depend on the protein of interest that we're looking at. So if your protein of interest is in excess, then probably we'll have to increase the amount of nanobody as well. So ideally, I feel that the nanobody level should be in excess of the protein of interest level in order to get but I feel that has to be optimized for each protein of interest that we look at. So that's why we created a different set of promoters for each, so that we can tune around and play around with the promoter strength and then express the nanobody at different levels.
Also, if you can give the actual expression levels from different promoters, then people can have some idea.
Yes. We're actually planning to do that.
That would make sense.
Yeah. So in this whichever.
They have to try all.
Yeah, so we're in the future we're trying to do that because we have this particular antibody which recognizes nanobody in general. So we get that antibody, and then we can try to also from the yeast lysates we try to see how much of the nanobody is expressing with each different promoters.
Okay. If there are no questions, then thanks, Anubhav. Someone will continue now.
Thank you, sir.
Okay, so we come to the end of the session. So our last speaker is from Devabrata's lab, Nagjyoti Pulagurola. She works on jasmonic acid in plants, and she's from Devabrata's lab, and she started a PhD August 2020, and she did her master's from Osmania University, biochemistry. Are you here? Yeah.
Hello, are you able to hear me?
Yes, yes.
Okay. Thank you for the introduction, sir. Firstly, I would like to thank BioEC Centenary Organizing Committee for giving me opportunity to talk about research of top interest of our lab. So today, my topic of talk is decoding the molecular basis of inositol phosphates homeostasis in plants. Starting with the presentation, as you all know that the plants are sessile organisms. They are restricted to the site of germination. The environment they are surrounded with, either they can be they are prone to many threats, maybe either biotic stress we talk about or abiotic stress. Then have you ever wondered that how plant, like without running away from the predator, how would it survive and how would it protect itself? So over the course of evolution, plant have evolved many unique mechanisms. One of the mechanism of that is secondary metabolites. So secondary metabolites have been integrated into the signaling network, and they have been established into signaling networks. And one of the signaling network is myoinositol derived signaling pathway. So this myoinositol is a sugar molecule, a cyclic polyol having 6 hydroxyl group at 6 carbon position. If it is phosphorylated at 6 different positions, and then it is called phytic acid or INSP6. And this is a major storage form of the phosphate in seeds, containing up to 75%. And it is located in the aileron layer of the seed. Further, due to its highly negative charge, it chelates with the divalent ions as iron and zinc and other calcium and magnesium. Due to this property, it leads to iron and zinc deficiency in humans and termed as anti-nutrient. Further, it is hydrolyzed by the phytase enzyme, but monogastric animals doesn't have a phytase activity in the digestive tract. So the absorption of the phytic acid doesn't happen, and they get excreted and leads to the pee pollution and eutrophication of the groundwater. Further, this implies these two properties give the reasons to generate low phytic acid crops in plant breeding programs. And now the main objective of the biotech companies is generation of the low phytic acid crops. But unfortunately, most of the low phytic acid crops generated over the last 4 decades are associated with the poor agronomic performances, which is linked to many negative pleiotropic effects regarding mainly seed viability or plant development. Here, for instance, if you can see, this is a plant wild type, and this is containing the low phytic acid, that is low INSP6, and it is more susceptible to the pathogens. And here another one, this is a rice plant developed with a mutation of ITPK61, and this mutation led the stress tolerance levels very much low compared to the wild type. So this implies that the low phytic acid plants have a less stress tolerance. And further, in tobacco plant, there is with the heterologous expression of the exo-pH, which is a phytase enzyme, and converts INSP6 to INSP7, which results in low INSP6. And another mutation of IPK1 in Arabidopsis thaliana, which is an enzyme which phosphorylate INSP5 and INSP6. So these two plants, if you see, these are the low phytic acid crops. And if you see compared with the wild type, these two, the low IP6 plants are severely compromised in growth and development. And this all negative pleiotropic effects emphasizes the pivotal role of inositol phosphate metabolites in plant and their maybe the involvement in signaling networks. Further, to establish This phytic acid, most important, regulating the cellular epigenetic regulations and cellular signaling network hormonal regulation in plants. So we cannot avoid now the phytic acid and its important role. And we have to take into consideration the inositol pyrophosphates, which are called derivatives of inositol IP6. So one is IP7 and one is IP8. These are the phosphorylated forms of IP6. And further, taking the inositol pyrophosphates, now in eastern mammals, the inositol pyrophosphates are well established. That KCS1 is a kinase enzyme which phosphorylates INSP6 to 5-INSP7, and VIP1 which further phosphorylates 5-IP7 to 1,5-IP8. In plants, in spite of discovery of IP6 100 years ago, the detection of the IP6 kinases in plants would take a long time because of the lack of technique which would identify the inositol pyrophosphates in plants. Detection of these kinases got delayed, and one of the now recent studies have identified ITPK1 and VIH as in IP6 kinases which phosphorylate IP6 to IP7 ITPK1 and which further get phosphorylated by VIH to IP8. And recently we should thank to the SAX HPLC technique establishment, because due to which we could identify the inositol phosphates in plant extract. Here is HPLC result analysis of the plant extract. Here you can see the lower inositol phosphates are eluted first and then higher inositol phosphates. And here, taking the advantage of the system, VIH2 mutant that doesn't have an IP7, that doesn't phosphorylate IP7 to IP8, have a reduced levels of IP8 in HPLC analysis result. So by this, it has been led to the identification of VIH as an IP7 kinase. Further, what is the physiological consequence of this mutant? So the Mammister brassica caterpillar, which was taken Mamica brassica caterpillar larvae, which were conditionally fed on VIH2 plant and colonel, and they've seen that the caterpillar which were fed on VIH24, where the weight was way much more than compared to the wild type, which says that the VIH dependent INSP8 was involved in activating the defense response. Further, with the previous knowledge, with the previous studies, we got to know that there is in the receptor complex, inositol phosphates have been found. So the coincidence detection, this study suggests that the INSP8 and the coincidence detection of jasminic isoleucine and IP8 in the receptor complex suggests that they improve the complex formation, promotes the complex formation, and promotes the degradation of the JAS repressor and activate the jasminate responsive genes. Further, the recent identification of the ITPK1 as an IP6 kinase, which phosphorylates one INSP6 to 5-INSP7, which was a gene responsible is ITPK1. So the mutant analysis, the HPLC analysis of this mutant line, we have seen the reduced reduction of IP7 levels. And analyzing the phenotype of the ITPK1-2, we have seen that it was primary root elongation was impaired, and gravitropic root curvature and leaf venation was impaired in ITPK1-2. And this all develops into an auxin deficient phenotype. So ITPK1 is critical for plant growth and development. And having this knowledge that ITPK1-2 mutant is showing an auxin deficient phenotype, and having the knowledge of the receptor complex, that auxin receptor complex have found the INSP6 was found in that. So maybe we were wondering that maybe the INSP7 was responsible in the formation of the complex. So we took advantage of is2 hybrid assay and took the strains EGY48 wild type and the mutant line VIP1 mutant, where we can see that the VIP1 mutant line will have an elevated level of IP7, as which will not be further phosphorylated to IP8. So in this Y2H experiment, you can see here that in the mutant line, there is a robust interaction with the AFP2 and IIS7, which potentiates the, here this suggests that INCP7 potentiates the auxin receptor complex formation. And having this and the very recent identification that ITPK1 have a role in phosphate homeostasis, and we were intrigued that single inositol phosphate molecule linked with the different signaling pathways. So I was just wondering that whether this and still ITPK1 dependent INSP7 was involved in jasmonic acid signaling pathway. So to testify our hypothesis, I took advantage of mutant lines established ITPK1-2 and a complementary line having the ITPK1 in expressed genomic ITPK1 expressed in mutant line. So we have grown these three wild type and the mutant and the complementary line in the in addition with a 50 micromolar of methyl jasmonate, and we have seen that the growth reduction there was ITPK1-2 mutant line was relatively more sensitive compared to the wild type, and which the phenotype was nicely rescued by the complementary line. These are the images of the root length we've taken at different time points, and this suggests that ITPK1 regulates the JA-dependent root growth. And further, these are all findings made us to immediate for future directions to investigate the functional role of inositol phosphates in auxin perception by addressing the question that does this ITPK1 dependent INSP7 promote ubiquitination of auxin repressor, and can INSP7 contribute to the dynamics of hormone receptor complex? And due to the because of the results of methyl jasmonate experiment we got regarding the ITPK1 mutant, how does this ITPK1 coordinate different hormonal pathway is an interesting question to look forward. And here finally, I would like to end my presentation by thanking my lab members for their immense support and encouragement. And here I would like to thank my funding agency also. And I would like to add a special note and thanks to the DN Rao Sir lab and Utpal Nath lab for their support to establishment of lab.
Thank you, Jyoti, for a great talk. So is there any questions? Okay, so let me ask one simple question just to clarify. So the VIP1, the VIP1, so yes, what is the phenotypic thing in yeast? So you can delete them, right? The
yes,
VIP1?
Yes, we have a strain which was deleted.
So what is the phenotypic looks like in yeast?
In yeast, we didn't find much, like I don't have, we didn't analyze the yeast phenotype, but we have used as a tool for the studied VIP1 gene in our studies.
Okay, so that brings me another question. So when you show this interactions, right, that
yes,
blue color changes the XGAL thing, right?
Yeah.
So did you quantify the beta galactosidase assay here? Like
actually, we did an ONPG assay also, but okay, it was not like we could not promote. We didn't do much experiments regarding that. We just ended up doing this interaction with the XGAL plate also only.
Okay, so you future plan to do some IPs and so on. Is that
yes,
correct?
Yes.
Okay, great. Thanks. So is there no further questions? So I'll end the session. Thank all the speakers for the wonderful talk. And thanks, Deb, as my co-host. And thank you, everyone.
I thank all the speakers of this session for their wonderful talks. I would now request Deepraj to take over from here and continue with the next session.
Thank you, Anjali. On the completion of session 1 and the BC Alumni Student Talks 1, we thank all the speakers, the chairs, and the BICS representatives, Sananda and Anjali. We now welcome you to session 2, which is focused on cellular biochemistry. This session will have 2 talks by Professor Hemlata Balaram and Professor A.P. Nare. And the session will be chaired by Professor D.N. Rao. I request all attendees to use the raise hand button to indicate if they have any questions for any speakers. Questions will be addressed only after the respective talks end. Please do not unmute yourself for questions before the chair of the session instructs you to do so. For participants in YouTube, please post your questions in the chat box. Professor D.N. Rao joined the department in 1989 and is an honorary professor in the department now. He is also the convener of the previous and the present Centenary Conference. His group studies DNA-protein interactions using Helicobacter pylori restriction modification enzymes and Neisseria gonorrhoeae DNA mismatch repair proteins as model systems. May I now request Professor D.N. Rao to kindly introduce the speaker.
All right, thank you very much, Debraj. In this session on cellular biochemistry, we have two eminent speakers. One is Hema Balram from the Jawaharlal Nehru Center in Bangalore. She'll be the first speaker. And the second speaker is Professor Naren from the Cedars and Sinai Institute in Los Angeles in California. Hema Balram completed a PhD at the Indian Institute of Science when I was also a student here. So I've known Hema since then. And then she spent some time in, I think, if I'm correct, she went to do a postdoctoral stint in Dan Santi's lab in the University of California in San Francisco, and then came to the AstraZeneca Center, or the other way. And then in 95, she joined as a faculty at the Jawaharlal Nehru Center for Advanced Scientific Research, where she joined as a faculty fellow and now currently a professor there. Her research is broadly categorized in three themes: the molecular enzymology, protein structure function analysis, and the crosstalk between nucleotide and energy metabolism in the malarial parasite, the Plasmodium, and studied extensively on the purine nucleotide metabolism in Plasmodium falciparum. Hema is an enzymologist par excellence in this country. Very few people like her do this kind of work, and she's a rare species in the area of enzymology and biochemistry. She's a fellow of the two academies, the Academy of the Bangalore Academy and the National Academy of Sciences in Delhi. She was the Dean of the Academic and Faculty Affairs in Jawaharlal Nehru Center. And it's a pleasure to have Hema give a talk this evening. She also spent some time, I don't know, 1 year or less than 1 year in Professor Padmanabhan's lab. So she's an alumnus of our department. Hema, it's all yours.
Thank you, Dian. And to begin with, I'd also like to thank the organizers for inviting me to give a talk in this celebration. The Department of Biochemistry has had 100 years of very glorious time. I wish the department continued success in the years to come. As Dian said, I'm trained as a chemist, and I had a strong desire to transition into biology. And I spent a period of about 1.5 years as a postdoc in Professor Padmanabhan's laboratory in the biochemistry department. Both the department and GP's lab were a great place for me to transition into biochemistry. I'll just start my lecture by sharing my screen. Can you see the screen?
Yes, yes.
Okay. So over the past next 30 minutes, I will present some of our recent work on aspects of metabolism in the malarial parasite Plasmodium. As you can see from this map of the world, all portions that are shaded in yellow and red are areas which are either endemic for malaria or areas in which transmission of malaria still continues to exist. And the title of my talk is on an aspect of metabolism in the parasite, which involves the TCA cycle intermediate fumaric acid. I'll begin by a brief introduction to the life cycle of the parasite, because I'm going to consider two stages of the parasite life cycle itself. Infection of a vertebrate host, and in this case, I've shown a human over here, begins with a bite of an infected mosquito. So the infected mosquito injects stages known as sporozoites into the bloodstream of the host. This reaches the liver, undergoes hepatic schizogony, and from there it enters the bloodstream and infects erythrocytes. It is during this erythrocytic schizogony that the disease itself is manifested. This reproduction through the erythrocytes is an asexual form of reproduction, and some of these parasites in the asexual stages differentiate into gametocytes, and if these are taken up by a mosquito, they undergo sexual reproduction in the mosquito midgut. And here, what I'm going to talk about are two stages of the parasite life cycle. One is the erythrocyte life cycle, and the other are a couple of stages within the mosquito life cycle. 2002, the entire genome sequence of Plasmodium was published. And in the paper, there was also a metabolic map that was mapped out from the genome sequence that was available. I'm going to talk about two species of Plasmodium that we work with. One is Plasmodium falciparum, which we can examine only the erythrocytic life stages, which can be cultured in the laboratory, and you can examine all the erythrocytic stages. And Plasmodium berghii, which cannot be cultured in vitro, it needs the mouse, but you can manipulate it genetically fairly easily compared to P. falciparum. And one can also examine some parts of the mosquito stages even without having a mosquito colony in the laboratory. Both the parasites divide very rapidly. Plasmodium falciparum divides every 48 hours, where a single mother cell gives rise to about 20 to 34 daughter cells. And these are the erythrocytic stages. Plasmodium berghii erythrocytic stages divide every 24 hours, giving rise to single mother cell, giving rise to daughter cells. And all these are asexual forms of reproduction within the erythrocytes. Yeah, so you can see that when they're dividing very rapidly, metabolic requirements of the parasite are very high, and interference with metabolic reactions would form a basis for therapy. And in fact, there are many drugs that we take now that do interfere with various metabolic processes of the parasite. There are certain unique features with regard to metabolism in malarial parasite. And what I've alluded to over here are the erythrocytic stages, the metabolism in the erythrocytic stages. The organism itself, during its life cycle in the erythrocyte, it's a microaerophile. It requires very low concentrations of organism for its growth. So during its intraerythrocytic stages, bulk of the ATP that the cell needs is primarily produced by glycolysis, and the contribution of the mitochondrial pathways is very little. The other key features which allude to my talk over here with regard to metabolism are absence of these pathways over here. One is amino acid biosynthesis. It takes salvages from the host. It doesn't synthesize any amino acids. There is no heme degradation pathway, despite the fact it polymerizes the heme from hemoglobin into hemozoin pigments. It doesn't synthesize purine nucleotides de novo. It cannot salvage pyrimidine nucleotides. So these are just few metabolic aspects which are relevant to the talk that I'm going to be giving. So as was told in the introduction, I have been looking largely at the nucleotide metabolism in Plasmodium, and shown here is the pathway that is operating. There is no de novo synthesis. It salvages all preformed bases from the host, whether it's mosquito or human, and converts it into an intermediate, which is inosine monophosphate. This is the precursor for both the key two purine nucleotides that a cell needs, this AMP and GMP. In the parasite, what I'm going to focus on is what is known as purine nucleotide cycle, and this is expanded over here on the right side. In this cycle, a molecule of IMP is converted first into succinyl AMP, and in the process of conversion, it takes a molecule of aspartic acid and forms succinyl AMP from IMP. This is converted into AMP, and during the process of this conversion, this dicarboxylic acid, alpha beta unsaturated dicarboxylic acid, fumaric acid is released. So for every molecule of AMP that is formed, aspartic acid is consumed, fumaric acid is released. The parasite also has an enzyme known as AMP deaminase, which completes this cycle, which is known as purine nucleotide cycle. So you can increase the flux of fumaric acid production if such a cycle is operating. This cycle is flux through the cycle is increases in an exercising muscle, because you would have more fumaric acid produced, enters the TCA cycle, and thereby more ATP would be produced through oxidative phosphorylation. And the cycle also modulates AMP levels and prevents inhibition of adenylate kinase. The cycle also actually will, as I will show you, will modulate NAD, NADH levels. The cycle where aspartate and fumarate are recycled will modulate NAD and NAD plus levels. So the question is, what is such a cycle doing in the parasite? Is it really operational? And why are we asking this question? So the process of TCA cycle intermediate being replenished from outside is known as anaprolysis. So what I'm talking about is purine nucleotide cycle producing fumaric acid. For every molecule of AMP synthesized, fumaric acid is being produced, and this can anaprolotically feed into TCA cycle. And this is what happens in exercise muscles so that you increase the flux through the cycle. In Plasmodium, glycolysis is the primary source of ATP, and flux through the TCA cycle during the intraerythrocytic stages is very low. So the question that we asked was, in this context of metabolism, where you have high levels of nucleotide synthesis, you have a low flux through TCA cycle, ATP generated largely by glycolysis, what is the fate of fumarate? Is there anaprolysis of fumarate, and is there any significance with regard to this anaprolysis? Yes, so to answer this question, the first few experiments that we did before we set about creating a lot of knockouts of genes was to see if fumaric acid is indeed metabolized by the cell. It is indeed metabolized by the cell. If I add labeled aspartic acid, labeled fumaric acid is not thrown out into the medium, and labeled fumaric acid is converted into malic acid, and that's expected from the TCA cycle. The labeled fumaric acid also radiolabels DNA. It also radiolabels proteins. So we mapped this metabolic aspect using NMR, where we used, instead of radiolabel, we used C13 enriched fumaric acid. And we found that if we incubate these cells with C13 fumaric acid, I get C13 malic acid, I get aspartate, I get pyruvate and lactate. So if I now go back and look at metabolism in the context of the Plasmodium, I see that parasite has an enzyme, fumarate hydratase, which can convert fumarate to malate. It has not malate dehydrogenase, but it has both. It has malate quinone oxidoreductase and malate dehydrogenase. But the enzyme which we feel which is probably plays a role is malate quinone oxidoreductase, because it's known to be in the mitochondria. Then to see aspartic acid and pyruvic acid and lactic acid, we have genes for all these enzymes. This helped us to reconstruct what could probably be happening with regard to anaprolysis of fumaric acid. So this model, with a large number of studies we could put up, we have a purine nucleotide cycle which generates fumaric acid. This, we feel, goes into the mitochondrion, and two steps happen in the mitochondrion, which liberates oxaloacetate that has to go out if it has to be converted back into aspartic acid and lactic acid. And since we see these intermediates all by NMR, this is the pathway that we have put forth. Thereafter, we ask the question, is this anaprolysis of fumaric acid important to the parasite or not? This is the purine nucleotide cycle that operates within the parasite. If we knock out the genes for either adenylosuccinate synthase or adenylosuccinate lyase, fumaric acid will not be produced. But at the same time, I will deplete the cell of all AMP, and to knock out these two would be lethal to the cell. So we rewired metabolism in the parasite by putting yeast adenosine kinase into the parasite. So we expressed yeast adenosine kinase, and then our hope was, if this expresses and is functional, we can now inhibit using a chemical inhibitor. It's an aspartic acid analog, adenylosuccinate synthase. And then ask the question, is anaprolysis important of fumaric acid, or is it, just a mechanism that is just an intermediate that is there and doesn't have to make any metabolic significance? This study shows that if I don't have yeast adenosine kinase and I put hadacidin into these cultures, the parasite dies immediately. That's the red line over here. On the other hand, in lines which are expressing AK, the parasites do grow, but over a period of time, the parasite's growth rate dramatically reduces, indicating that even though AMP is being produced, this fumarate going in through the TCA cycle, one little quarter of the TCA cycle, is important for Plasmodium falciparum intraerythrocytic stages. Around the time we also went on to establish, as I said, that this is a pathway which involves both the mitochondrion and cytosol. We went on to ask the question, are these enzymes within the mitochondrion or within the cytosol? Because we have two compartments which are now playing a role in this metabolism. And we established the fact that the first enzyme, fumarate hydratase, localizes to the mitochondrion. We've also shown that malate quinone oxidoreductase actually localizes to the mitochondrion. So both of them are within the mitochondrion. Fumarate generated in the cytosol goes into the mitochondrion, gets converted to oxaloacetate in the mitochondrion. And so this localization was important to establish the fact that we have multiple compartments playing a role in this pathway. We also, while screening inhibitors, we also hit upon an analog of malate, which is thiomalate, which was a potent inhibitor of the enzyme in vitro expressed. We also found that it inhibits the P. falciparum growth itself in vitro cultures. And I must point out here that fumarate hydratase in P. falciparum is an enzyme which has an iron sulfur cluster and belongs to what is known as class 1. Human beings don't have this enzyme. And the thio analog of malate does not inhibit the enzyme, even at extremely high concentrations of 10 millimolar. So fumarate hydratase is anaplerosis is important. And fumarate hydratase, which is the first enzyme which metabolizes fumarate in the TCA cycle, is actually essential for the parasite, and chemically inhibiting it will kill the parasite. At the same time, a little before our studies were completed, there was this paper published by Akhil Vaidya's group, which showed that of all the 8 enzymes of the TCA cycle, 6 of them can be genetically ablated. The only 2 enzymes which cannot be knocked out are fumarate hydratase and malate quinone oxidoreductase. The 2 enzymes which metabolize this metabolizes fumarate to malate, and MQO metabolizes malate to oxaloacetate. They also showed that apart from, they also showed another thing which we are talking about, glutamine anaplerosis. They talk fumarate anaplerosis. They showed that glutamine anaplerosis is there in the parasite. Yeah. So since these 2 genes could not be knocked out, we decided to use P. burgii as a model system because for various reasons. This is a rodent malarial model system. It, like P. falciparum, has a haploid genome. All the pathways in it are almost completely conserved. The enzyme sequences that we are dealing with are also highly conserved. However, unlike falciparum, though genetic manipulation of P. burgii is also difficult, but there is greater ease compared to P. falciparum. And having P. burgii as a model system, it enables us to study the mosquito stages. So we can knock out genes by using, double homologous recombination, replace the gene of interest with a marker, drug selectable marker. And after having transfected P. burgii with the DNA, which is going to knock out the gene, we infect these transfected parasites with, inject them into mice, allow infection to establish, then we put drug pressure. If the gene has been knocked out and the drug metabolizing gene has been replaced, the gene of interest, then I will have parasite recovery. But this is an in vivo model system, unlike P. falciparum, which all can be done in vitro. These both have the advantages and disadvantages. So when we try to knock out the FH gene in P. burgii, we found that we could not knock out the FH gene in burgii. And all of our studies we did by using the whole mouse BALB-C strain of mice. So in all of our studies, we used BALB-C strain of mice, and we found that we got various kinds of drug-resistant parasites, but all of them were either wrong integrants or integrants obtained by single crossover recombination, or there were cases of gene duplication, and FH fumarate hydratase continued to be expressed. So in all these clones that we got, we found that the fumarate hydratase gene was not knocked out. Around this time, there was a paper published which said that in C57 strain of mice, if you used to establish P. burgii infection, you could get a knockout of the FH gene, the fumarate hydratase gene, but you got a slow growth phenotype. So we probed this question further. Is there a difference between 2 mice strains, which we had no intention of doing, looking at differences in mice strain. But since we had a paper that was published and we have done all our work in BALB-C, and this paper that got published was done in C57. We redid these experiments and we found that to generate knockouts in BALB-C, irrespective of the way we did it, we could not get knockouts generated of FH gene in P. Berghii if we used BALB-C as the host system for establishing infection. And in our hands too, the C57 mice, we could delete the gene and establish knockout parasites which had FH gene knocked out. Yeah, so we went on to ask the question as to why in BALB-C we are not able to get knockouts, and what is the difference between these strains of mice which can both get infected by P. Berghii, but if certain knockouts are viable, can be generated in one strain of mice and not in the other strain of mice. Are there any metabolic differences between these host strains which now enable a particular knockout to be generated and another kind that cannot be generated? In BALB-C, what we see is that to begin with, immediately after transfection and before drug pressure has been applied, we have the FH knockout with the FH gene has been replaced by a marker. In the same mice, we also have a population of cells in which there has been a wrong integrant. We have the wild type FH gene, but the marker integrated in a long loci, both parasites growing simultaneously. The moment we put the drug pressure, these are unable to grow as well as the wild type, which has got a wrong integration. And eventually, by the time we can see resistant parasites appearing, the true knockouts have been completely depleted and overtaken by wild type parasites, which have a wrong integration and which are able to grow. This only tells you that when you do lots of knockout experiments, how you can have complications. Now, to look at metabolic differences, when we looked at the literature, we found that there is one key metabolic difference. And that metabolic difference between these two strains of mice, both which can get infected with P. Berghii, is in the absence of this NNT gene in C57 and not in BALB-C. This now impairs glucose metabolism, and it goes to show that how you can have parasite host metabolism now playing a role in whether you can abrogate a particular gene and have parasites growing or not. So with this information on the FH gene, we put up a picture of what could be happening with regard to fumarate metabolism in the parasite, fumarate that is coming from this cycle, and general fumarate metabolism in the parasite. If I knock out the FH gene, which we couldn't knock out, actually, we can do it very conditionally, and in P. falciparum, you can't knock out, you have accumulation of fumarate. Fumarate continues to get produced, though the TCA cycle flux is low, it comes from here, and we have accumulation of fumaric acid. This can now drive this reaction backwards. This reaction which converts SAMP to AMP can go backwards equally well. And if accumulation of fumarate happens, this reaction goes backwards. Another feature of fumaric acid is a dicarboxylic acid where alpha beta carbons are unsaturated. Highly reactive molecule which can undergo a reaction known as Michael addition, and it can modify cystenyl residues by succination, and it can also modify glutathione. And these have deleterious consequences not only in Plasmodium but in other cell types too. This will also lower, if I block FH, if I delete FH, this will also lower oxaloacetate pools, and it will prevent recycling of aspartic acid. There is no amino acid biosynthesis in the parasite, and hence this will impact multiple things: purine, pyrimidine, nucleotide synthesis, asparagine synthesis, and eventually protein synthesis. This will also perturb NAD levels, and I'm going to show you that in a few minutes. Yeah, so with this information of fumarate anaplerosis and the essentiality of FH, we ask the question, what will happen if I now knock out MQO? If I knock out a transporter which will transport oxaloacetate produced by MQO in the mitochondrion, prevent this transport, and what will happen if I prevent the cycle itself by knocking out first MDH and doing a double knockout of MQO. And since we are doing this all in P. Berghii, I can look at the asexual stages. I can also look at the sexual or the gametocyte stages and the mosquito stages because I'm working with P. Berghii. Yeah, first to establish that MQO is indeed functional. And we did this again by NMR. We have shown that fumarate hydratase is functional. It's essential for P. falciparum. It's essential in certain contexts in P. Berghii. Next is the enzyme MQO, which converts the generated malate into oxaloacetate. And this oxaloacetate is then converted by aspartate aminotransferases, and it recycles aspartic acid, lactic acid, pyruvic acid, so on and so forth. How do we show this as functionality within the cell? The way we did this was by NMR. And as I told you, in a normal parasite, I can see fumaric acid getting converted to malic acid, aspartic acid, pyruvic acid, and lactic acid. But if I inhibit this process by using a ubiquinone analog, the conversion of malate to oxaloacetate in the mitochondria in this parasite is dependent on the availability of ubiquinone. Simultaneously reduces ubiquinone to ubiquinol. Ubiquinol is recycled back to ubiquinone by complex 3, but it can be inhibited by this drug known as atovaquone. It's antimalarial, it's anti-toxoplasma by inhibiting complex 3. If I do that, and indeed this pathway is functional in the intraerythrocytic stages of Plasmodium, I should abrogate the production of aspartic acid, pyruvic acid, and lactic acid. And that did indeed happen. Moment I put atovaquone, I block it at the level of malic acid. So going to show that this pathway within the cell, NMR helps me show, is indeed operational. Yeah, but the knockout, surprisingly, we could generate a knockout of this viable knockouts in both C57 and the BALB/c mice. So we went on to see the transporter. Once oxaloacetic acid is formed, it has to be transported out into the cytosol if aspartic acid has to be generated starting from fumaric acid, and then I have pyruvic acid and lactic acid. So the molecule that we homed in was a dicarboxylate tricarboxylate carrier, which because in the literature it was shown that it has specificity for oxoglutarate, it has specificity for malate and oxaloacetate, the metabolite that we are interested in. Surprisingly here, just like in fumarate hydratase, the knockout generation was possible only in C57. So hence there was a mouse strain dependence in the ability for us to generate knockouts. The next is MDH, because as I said, this will also perturb the MQO and MDH together will perturb NAD and ADH levels in the cell. MDH was already shown to be not essential, and we also did it, and it was shown to be not essential for intraerythrocytic asexual stages of P. Berghii. To generate a double knockout of both MDH and MQO, in P. Berghii, it's not an easy task to do, unlike in many other systems, because we have a large paucity of markers for selection. And we primarily use only one marker for selection. And so we have to keep recycling markers. So once we have deleted one gene and replaced it with a marker, we now remove the marker and then use this strain with marker deleted for deleting its second gene. This is it's simply said, but it's a long time consuming process, and we made a double knockout. And the double knockout was completely viable. The asexual stages, intraerythrocytic asexual stages, were completely viable. So far, what have I said? There are differences between P. falciparum and P. Berghii. In P. falciparum, you can't knock out any of these genes. The parasites are not viable. In P. Berghii, you have, in some cases, you have a mouse strain dependence when you're trying to generate a knockout. And in other cases, you can, irrespective of the mouse strain, you can generate a knockout, and they are viable intraerythrocytic asexual stages. As I said before, the pathways are conserved, the enzymes are conserved, but then you see a difference between the two. There could be a possible difference in metabolic flux that we are not aware of. One is an in vitro model, P. falciparum, other is an in vivo model, and there will definitely be differences. And we have two different species of the parasite, falciparum and P. burgii, infecting different hosts. Definitely, there are differences in host metabolism, which could be influencing our results. And I should mention here that P. burgii, like P. vivax, which infects human beings, has a preference for reticulocytes. Though in all these studies of these knockouts, we didn't see the knockouts preferentially infecting reticulocytes. But these are the features that could be there. As I said, I can look at the sexual stages too, at least the mosquito stages, some of them. And the stages that we can look at, gametocytes, that's in the blood itself, of mice infected with parasites. I can look at zygotes and I can look at ookinetes. Those are the stages over here. And ask the question, what happens to metabolism? Is it rewired or is it different when I go from one stage to another? And these have implications not just in parasite biology, but diseases like diabetes and cancer, so on and so forth. So how do we, this is just a slide which tells me that I can harvest wild type parasites from the mice, treat them in vitro with the reagent known as sulfadiazine, kill all the asexual stages and keep only the gametocytes in the blood. Then I treat it with a chemical known as xanthinuric acid in vitro and allow gametocyte development into gametes. Fertilization occurs in vitro in a test tube. And then I have both zygote and ookinete formation. And these are wild type parasites staged and imaged in the laboratory, going through various stages after fertilization.
Heba, you have a minute more.
Okay, I'm almost done. So this is just a summary of the data of what happens when we knock out. Gametocytes are all formed, whether I have knocked out any of these genes. However, none of the sexual stages are formed. We don't see zygotes, we don't see ookinetes. Either they are highly reduced or we don't see them at all. To summarize, we have metabolic consequences of the blood stages. They don't seem very critical as far as P. burgii is concerned, but in P. falciparum, they're critical. And in the mosquito stages, now everything changes. It becomes very critical in the mosquito stages. These pools of fumarate are essential. The amount of OAA that is produced in the mitochondria and the ubiquinol pool now becomes very critical. You have the transporter, which now also plays a very critical role, which transports OAA out into the cytosol of the parasite. You have MDH now, which again is critical because you don't have sufficient NAD levels being produced. And when I make a double knockout, it's not surprising because the individual knockouts themselves affect the mosquito stages. To conclude, you've now got two kinds of parasites that you've studied. One is P. burgii, the mouse model, where we can do in vivo studies, P. falciparum, where it's largely in vitro studies. And you can see here that in one case, it affects the blood stages itself, so we don't even go to the mosquito stages. Whereas here, the blood stages are not affected in the in the mouse malaria model, whereas the mosquito stages are completely affected. So going to show you have stage-specific roles of these pathways. Different stages of parasite development exploit metabolism in completely different manner. And these information can be eventually used for exploiting and developing therapeutic targets. This is just thank you for your attention. And bulk of the work here in the laborious generation of all these knockout parasites was largely carried out by Arpita, and earlier studies were carried out by Vijay and Aparna. Thank you for your attention.
Thank you very much, Heba. Any questions from the audience?
Deepankar.
Yeah, Deepankar, go ahead.
Yeah, very nice talk, Professor Balram. I had the difference between the Black 6 and BALB-C was very striking for me. And one of the big differences between Black 6 and BALB-C is the difference in inflammatory responses. Black 6 is more inflammatory, BALB-C is relatively less inflammatory. Do you think that might also be contributing to some of the phenotypes that you see?
I must say that C57BL6 is a good model for cerebral malaria. With regard to metabolism, I've been looking at metabolic differences between these two strains of mice, and the key features that I see are in that dehydrogenase, which I pointed out. But as in terms of inflammatory response, I would say that it's a good model for cerebral malaria. With regard to these metabolic variations, I'm not very. I think that needs more investigation actually.
All right. Okay. Thank you.
Anybody? Any other questions?
I can ask one.
Yes. Pulshat.
Thank you, Professor Balaram, for a very nice talk. I think your study was a very good example of how for the same gene, you see different biologies depending on the system in which you're looking at. I'm just curious, based on the FH example, what do you think? Would still FH be a good target for targeting malaria, or I mean, how does one choose a system in such kind of cases going ahead? Because it's hard to work with both Burgi and falciparum. So,
I would say FH is still a very good target because in P. falciparum you can't delete the gene, and the human enzyme belongs to a completely different class. Both catalyze the same reaction, but there are class 1 and class 2. One is an iron sulfur cluster dependent enzyme. Other is an enzyme which doesn't require an iron sulfur cluster. There is no similarity in sequence, and there is no similarity in structure. What is sadly lacking is we can't do all these studies in vivo. We don't have a good, humanized model for looking at P. falciparum in vivo in a good model organism, which we can do. But if it is indeed essential, even in an in vivo system, then I would say FH is still a very good target. Both FH and
that's where the problem is, right? I mean, your studies indicate that in one case it is, and in one case it's not. So if one has to go further, which one would one believe and move on? Which set of results?
Yeah, I would say that if we can take, we are for right now doing whether, if I inhibit FH using the same analog in P. Burgii model, whether it will die or not, one has to have, we still don't have an answer to it, but that should partly answer. But, right now I don't have a direct answer, but, FH cannot be abrogated, and it's a very different from the human enzyme. If it is indeed essential for P. falciparum, then I guess it is a good target.
Thank you.
Okay, wonderful. Thank you very much, Shema.
Thank you, Dean.
We'll move on to the second speaker in this session, the cellular biochemistry, and that's Dr. Professor Naren. Naren is a former student of our department. He did his PhD, he got his PhD in 1993 from Dr. S. Krishnamurthy, and later on, because Krishnamurthy passed away, he was Paul Vidyathal's student. Naren, after completing his graduate studies here, went to the University of Alabama and then went to the University of Tennessee as a professor, as a faculty member, and then was called to the Cincinnati, the Children's Hospital, to start off completely a new program, personalized program of medicine in cystic fibrosis. At Cincinnati, where he has been for a long time, only recently in July, he moved to Los Angeles. Naren was a Thomas Board Chair in the Cystic Fibrosis Research, the Division of Pulmonary Medicine. He was the director of the Cystic Fibrosis Research Center in the Cincinnati Children's Hospital. He just moved, as I said, to the Cedars-Sinai Medical Center in Los Angeles, and his research work focuses on two disorders: the cystic fibrosis, in which the CFTRI chloride channel is hyperfunctional, and the second one is the secretory diarrhea, in which the hyperfunction of the CFTR chloride channel can occur due to infectious toxins. A lot of nice work, pioneering work. I visited Naren sometime back, a couple of years, went to his lab, and some of the very nice work that he has been doing for the N number of years, last 10, 15 years on cystic fibrosis. So it's a pleasure to have Naren give this talk, and we would like actually him to come back to the department to physically visit us and give a talk here in Bangalore in the department. But Naren, this is all yours.
Greetings from LA. Thank you, DN Rao. And it's an honor to be presenting, and also the committee for having invited me. Can you all hear me?
Yeah.
Yes.
It's an honor to be presenting to a distinguished group of researchers. What I'll do today is talk about some of the work that we have been continuing here. I still have an appointment with Centenary Children's as well. So I have nothing to disclose. So this is the God molecule in the lab. We call it CFTR, or cystic fibrosis transmembrane regulator. So this protein has two nucleotide binding domains. The cryo-EM structure is shown here. This nucleotide binding domain has an ATP binding cassette, and therefore the name ABC transporter. What is unique about CFTR is this regulatory domain shown in brown. The phosphorylation of this domain determines whether the channel opens, and that is how it regulates salt and water transport. So CFTR is present on the apical surface, and in response to PK or PKG, it phosphorylates the channel. Chloride comes out. As chloride comes out, sodium follows it, and it creates an osmotic driving force for water, and it allows water to transport across the surface. And that transport is important for mucociliary clearance in the airway, and the mucus allows to be flowed. If not, what happens in diseases such as cystic fibrosis is you have thick mucus leading to Pseudomonas infection, biofilm formation, and that is how the morbidity and mortality happens. On the other hand, due to cholera or traveler's diarrhea, your enterotoxins or cholera toxin, what it does is excess activation in the gut leads to excess chloride, excess sodium, and excess water, and that's how diarrhea happens. So the point is, too much or too little activity of this channel causes a disease status. So the scientific premise in the lab is that CFTR does not function in isolation or singly. It functions as a macromolecular complex. Which complex predominates at any given time or space depends on its physiological and functional needs. So one of the earliest study we did as an independent faculty back in Memphis was that a macromolecular complex of adrenergic receptor, CFTR, and NERV1 can be found in airway epithelial cell. And more recently, my graduate student, Kavisha, a brilliant kid, what she did was she identified that adenylate cyclase 6 was critical in this complex. And then what she finds is that complex is not only important for the cilia-generated flow on the airway, but also it plays an important role in the height of the cilia and the ciliary biogenesis by the autophagia pathway. But of relevance to this particular talk is the study done by this brilliant kid, Chin-Ning Lee. What Lee did was in the gut showed that there was spatiotemporal coupling of a cyclic AP transporter, that is MRP4, to the CFTR chloride channel function in the gut epithelium. What it showed was that the agonist, the adenosine receptor, through the signaling pathway can generate cyclic AMP in a compartmentalized fashion that can activate CFTR. And this compartmentalized cyclic AMP has to be eliminated very quickly. And that happens either through the diestrase or can be pumped out through MRP4. And if you have a specific inhibitor, MK571, or if you knock it out, what he found was that led to a diarrhea-like phenotype. But one of the questions we had was, is this context dependent? Can we demonstrate this in real time? So we knew drugs such as irinotecan and AZT were substrates of MRP4. So around the same time, Hans Cleaver had demonstrated that he could use this intestinal stem cell. He had published the work in Nature back in 2007. So using the same model system, a brilliant young kid, Moon, had, in collaboration with Hugo over at Netherlands, developed this methodology in which she isolated these crypts from the intestine. And the reason we like these crypts, the 24-hour crypts, day 1 or day 2, is because they have this circular structure, have typically 500 to 600 cells within them. And the beauty is CFTR is present on the luminal surface. And when you activate CFTR with forskolin, which increases cyclic AMP, and in response to cyclic AMP, chloride comes in, sodium follows, and water comes in, and you have this beautiful swelling. And this swelling is CFTR dependent, especially in the upper GI, and it is primarily driven by CFTR. And in CFTR knockout mice, you do not see any secretion at all. So using this model, we were able to demonstrate that irinotecan, which is a substrate of MRP4, not only increases secretion, but it is CFTR dependent using a specific inhibitor of CFTR. MK571 was used as a positive control in these studies. And as we were doing this secretion, we did this protein-protein interaction using proximity ligation assay. We were looking at CFTR and MRP4 interaction. What we showed was using MRP4 knockout mice, the enterospheres do not see any interaction. And then in wild-type mice, which have MRP4, you see an increase in interaction. But as we see the secretion, the interaction increases. The point we were trying to make was these are context-dependent, and then when the protein is present, the interaction, as the secretion increases, the interaction increases as well. So this was the point of the whole study. This came in the cover of JBC, and I want to use this lyrics from Dr. Hook, the thrill that gets you when you get your picture on the cover of Rolling Stone. Ladies and gentlemen, this is the closest I'm going to come to the cover of Rolling Stone. So how do we study this interaction? We use various methodologies. One of the brilliant kids, Chandrima, who joined my lab, she, in collaboration with Naiki Fuji, a medicinal chemist, designed a methodology of using click chemistry. This is a CF corrector. This corrects the delta F508, which is the most common folding defect in CF. This drug corrects this molecule. So what she did was she introduced this alkane group, and using ASI group, you can click this reaction in place. And what she's able to show is that this drug binds directly to this mutant form of CFTR. And she was able to show that VX809 induces F908 conformational change during the synthesis and processing. But she can also use this method to kind of look at other interacting partners as well. And Kavisha, on the other hand, introduced a flag tag on the outside of CFTR and uses a quantum dot. And what she does is she does live imaging. This is wild-type CFTR. You're looking at a single molecule on the surface. This is live imaging. Now, F508 is a trafficking defect. The protein gets stuck in the endoplasmic reticulum. But what she has done is she rescues it, introduces it at 28 degrees for overnight, then the protein comes to the surface. But the point she's trying to make is when it comes to the surface, you see increased trajectory. The reason is the protein-protein interaction of this rescued protein is impaired. And what she's trying to do is she's trying to stabilize this protein by using various cross-linking approaches so that she can increase the half-life of this molecule. On the other hand, Yunji came up with a very novel approach. What he tried to do was he identified an interaction in the endoplasmic reticulum, this DNA JB9, which interacts with the ER lumen. And this interaction identifies the misfolded protein and adds ubiquitination residues and then takes it to the degradation pathway. So what it does is if it knocks out this protein using a knockout model, generating a knockout mice, or if he can perturb this interaction, what he finds is that he can keep this molecule enough in the endoplasmic reticulum, then to introduce a corrector, then he can push it out to the endoplasmic, from the endoplasmic reticulum to the plasma membrane and correct this molecule. Now he's an assistant professor in Indianapolis over at University of Indiana and running his own program as well. So why are we using all these approaches? There are more than 2,000 different forms of mutations described for CFTR. Although there are 2,000 different forms, they can be classified into 5 or 6 different classes. Class 1 is amber mutation. Class 2 is what the holy grail of CF research is. It is a trafficking defect. It gets stuck. 70 to 90% of the patients have this kind of mutation. The protein gets stuck in the endoplasmic reticulum, and it is a temperature-sensitive trafficking defect. Class 3 is the one it comes to the surface, and you have a drug called Kalydeco or VX770, which can bind to it and open the channel. Okay, these are all various aspects of the protein is shown at the bottom here. So why did we get into precision or personalized medicine? As I said, there are more than 2,000 different mutations described. So can we develop patient-specific screening tool? There are serious disease variability among CF patients. Even if they have delta F, let's say homozygous, they still have some variability. Some mutations are so complex, especially the splice mutations, that it will be a long time before we can develop cell line. So the question we asked was, can we come up with a biopsy of tissue to isolate organoids or aerospheres from CF patients? And would they accurately mimic in vivo mutant CFTR function? If so, can we potentiate it, correct it? And so we were in Memphis, so we decided to move to Cincinnati. The story we told everybody is we wanted access to human samples. But truth be told, when the end of the world comes, I want to be in Cincinnati because it's always 20 years behind the time. Ladies and gentlemen, let me tell you something. Omicron COVID variant is coming. Climate change is already here. The world is ending. So all of you might as well come to Cincinnati, okay? So seriously, this was the reason we moved to Cincinnati. We have a bunch of really brilliant kids I was able to recruit, and we had access to multiple organ tissue and samples, and we had access to patients as well. So the beauty of this group was we were able to generate model system in the upper airway, the tracheal epithelia, the CF lung on a chip, the pancreas, it's a multi-organ disorder, the ductal organoids. We built a pancreas on a chip. And of course, today's talk of interest is the intestine, small intestine is what I like the most, the enteroid or organoids, the monolayers and the human intestinal organoids. We also built a gut on a chip, which I'll not talk about today, okay? So what we did was I partnered with JP Clancy and Jeff. They are both physician scientists, and we built a personalized cystic fibrosis therapy and research center. The idea was a patient walks in, can the patient come out with an improved outcome? The idea was we would build a personalized clinic core. JP Clancy would run that, with the idea of the core being able to do nasal PD intestinal current measurement, multiple breath wash, spirometry, and lung transplant samples, nasal and tracheal tissue, upper and small intestine. And this core would supply all the samples needed for the personalized model core that I would run with Michael Helmrath. And this core would have organoids, iPSCs, pancreas, CFRD, and asset development and banking and things like that. And Jeff would run this core, Atlas of the Respiratory Epithelial Core. Much of it was to do with short circuit current fact sorting and, importantly, single cell RNA-seq analysis. And of course, the idea is, can we come up with improved patient outcome? And the second aspect was to have a next generation of investigator. And this was the overall theme of this particular program. And And I'm going to now talk about the story of St. Nick, wherein we kind of incorporate all these things into this program. So here is the study that was done mostly by Kavisha and Yang Fen. They did all the basic science studies, and John Brewington, who did N of 1 clinical studies, and Nambi here did the induced pluripotent stem study. So as I told you, we use this organoid system. I talked about this organoid that we used to look at CFTR-dependent fluid secretion. We had done all this in mice. So now can we do it in human was the question. So normal CFTR, you would see secretion like what I showed here. But in mutant, you would see no secretion. And mutant with intervention, you would see intermediary secretion. So the idea was to have the core one was to do about 200 patients, and the idea was to recruit and consent, biopsy on all the 200 patients, and then blood from some very selective patients. And then you would have the enteroids from every one of them, and then, of course, data analysis, and iPSC from a select few patients. I'll talk about an example here. And then the core 3 would help with single-cell RNA-seq analysis. So here is the flow chart. You'd get the biopsy, and then you would have the organoid. And then typically within 72 hours, you can get the assay done, and you'll have the data. So does it work? Here is the non-CF. You get a robust response to forskolin, whereas delta F, you do not see anything. Forskolin, you don't see any response. Now, delta F is a trafficking defect. You put it at 28 degrees Celsius. Here is proof of concept. You see a nice response to forskolin. You use a corrector. This is FDA approved corrector. You see a better response. But now at 37 degrees, this FDA-approved response, you do see a correction. So we had a drug that was working, and we could see a readout that we could take it to the clinic, okay? So we have done over 180 samples so far, and we have done N-of-1 clinical studies on about a dozen patients so far. So here is one such patient. So the first mutation on this patient is a G542X. That means it's an amber mutation. There is no function at all. And the second allele is a complex set of mutation, R74W, V201M, D1270N. So we did not know what to expect from this patient. The sweat chloride was high. So typically we all have a sweat chloride of 20 or 30. If we have any mutations in our CFTR, we will have high sweat chloride because in the sweat, in our sweat gland, we absorb CFTR is absorptive in nature. So what happens is if it doesn't absorb your chloride, the chloride comes into the sweat, and therefore you'll have high amounts of chloride in the sweat. So the fecal elastase is normal. So this patient was considered partial CFTR function. So we got his biopsy, and we are looking at the function here. Here is what the patient's function looks like, little or no function. This is non-CF. You see a robust function. And with the VX770, we saw a robust function. This is acute treatment. And then this was very exciting for us. So then what we did was we designed an N-of-1 trial. Essentially, we did, the idea was to have a nasal PD MRI sweat chloride FEV1. We did a pre-treatment wash out for 7 days and a sweat chloride at FEV1. And then we were going to do a kaleidocope treatment because we knew the organoid responded. And then depending on how the patient responded, we were going to design the rest of the aspect of it. And this is Nick. We call him Saint Nick. He's looking at, we have permission from his father to use this picture. And Mike is looking over his son's shoulder. He's looking at his organoids that were grown in the lab. He's on kaleidocope 770. He has improved lung function. His FEV1 has improved dramatically. His BMI has improved dramatically. His GI symptoms has resolved. And his quality of life has improved significantly. So he's doing very well. And what we were able to translate for what has happened in the lab to the clinic. Now, the day he came, we also consented through the FEST core and got some blood from him. And through our stem cell core, we were able to generate fibroblasts. And from the fibroblasts, induce pluripotent stem cell. And now we, along with Michael Helmrad's group, generated human gut spheroids. This is basically a combination of both epithelial and mesenchymal stem cells. And what we do is we take these stem cells, spheroids, and surgically engraft it into the mouse kidney cortex and let it grow for about back in the kidney for about 4 to 8 weeks. And then you get the human enteroids. You can either get the human enteroids or HIOs, human intestinal organoids. And when we look at stem cell markers, all the intestinal markers, these are all human markers. We have essentially grown Nick's intestine in this mouse kidney cortex. So of interest to this study, does this recapitulate what happens in the patient is what is shown here. We don't see a response, fluid secretion to forskolin. Only when we have VX770, we see a response here, suggesting that we have indeed recapitulated what happens with the patient. But of excitement to us was this study in which we showed after the experiment, when we did immunohistochemistry, that CFTR was indeed expressed in this patient. This is control. This is the patient sample. And the junctions look okay. But it had mucositis as expected. CF patients have increased amount of goblet cells. But of interest was, surprising was that we found an increased amount of lysozyme A, suggesting that the stemness was indeed altered. And this may explain why CF patients are at higher risk of cancer as well. So these are some of the studies that that is ongoing. So the core 1 looks at, in summary, looks at some of consenting some of the patients, and core 2 looks at various assays, and the core 3 looking at various single cell RNA-seq analysis. I have not talked about that aspect of the study here. And back to the question, can we target MRP4 interactome in CF? So this is a slightly busy slide, but I will try to summarize the study in this small sentence here. So what Christina Bayer's group has argued is addition of MK571, which is a very specific inhibitor of MRP4, augmented the potentiation mediated by VX770 observed in nasal culture derived from both these patients, and confirmed that MRP4 is expressed using RT-PCR. The point is, at the very right extreme here, you see that the patients who had these inhibitors of MRP4 showed an increased response as well. And Lukáš over at Montreal also showed that what was happening when you inhibited with MK571 was a specific phosphorylation residue S686 of CFTR was phosphorylated, suggesting that the phosphorylation was happening in a specific residue. So we think it is a legitimate target. So what we are trying to do is we are trying to do rational drug design. We are trying to look at some of these using a line-out system in collaboration with Alexandria with the University of New Mexico and John with St. Jude, looking at a small medium throughput screen, identify the drug, short circuit current, make sure that it works. Then using the submucosal gland, human patient-derived submucosal gland, put it on a chip. This is a modified chip with a smart membrane. This PDMS has been treated with UVA to make sure. Yes, I'm done, sir. So these are the kind of studies we have done. And in conclusion, to give you an idea, my lab looks at defining CFTR interacting proteins. We are interested in developing small molecules. We look at translational research, personalized model system, and look at lung on a chip. And of course, my lab has a very strict dress code, and but in reality, these are the young kids who helped me build my lab when we moved from Memphis to Cincinnati. I'm very grateful to them. And Kavisha here is going to be a young faculty starting in Cedars as well. And with that, I want to thank everybody and for your attention. Thank you.
Any questions from the audience? Yeah, 1 minute. Sandeep is here, who has a question.
Great talk, Professor Naren. Have you tried any, in the AMBER mutation patients, have you tried any read-through inducing agents? Like a
yeah, that has been, that is the next big thing in the CF world. So we now have drugs for more than 90% of the CF patients. The last 10% of the patients are the AMBER mutation. So the gene therapy is where they think they're going to go with it. There was some push. There was some thought that there may be some drugs there. There was a drug PTC102 or something that was developed, but unfortunately it didn't have any success. Okay. So that is the big push right now.
Okay, thanks.
Professor Dian, can I ask you a question?
Yes, please. Please.
Hey, Naren, nice to see you after a long time. Quick question, if you partially rescue CFTR function, will it lead to some collateral sensitivity into something else? Because more so with lung, because are you going to change the microenvironment for that kid?
Yeah. now that Trikafta has come, okay, so you have the delta F patients, right? So the delta F patients have no function. So in the pancreas, they're exocrine insufficient. So they do not have pancreatitis. So as they grow older, they get CFRD. So they're not at risk of pancreatitis. But carriers of CF have pancreatitis. They're at the highest risk of pancreatitis. But now, when the delta F patients are on the drug, as their function gets improved, they are now at high risk of pancreatitis. You understand? It's really a cool concept. You know, we're trying to figure out why that is.
All right.
Narayan, this is Srivatsan from UCLA. Hopefully we'll meet one of these days. I'm on cancer.
Yes.
Couple of things. One of them is the gene therapy, am I right? There have been a gene therapy study to improve the expression of this gene, am I right?
Yes, yes, that's a big push, yes.
And anything happened? I thought there was an article about it. One patient was successful and at the NIH.
Yeah, I think the study was more from, there was a study from, I think in England, there was a group that worked on a lentiviral-based strategy. I think they had some success with it. It was not a very big, this one. There have been some phase 1 studies in some of the, the CF Foundation is putting a lot of money into it. That is the big, this one-time cure is the big emphasis right now. And I think nearly a few $100 million has been poured into it in the last year. So I think that is the big push right now.
The other one was a CRISPR, am I right? CRISPR was also tried or being tried.
It is being tried. It's in, I think it is in various phases, early phases, I think. Even mRNA is being tried as far as I know.
The mRNA might be successful now that the COVID has been in control.
No, the problem with CF is delivery into the lung. That is a bigger issue than, because the lung itself is very thick and mucus and sticky. So the delivery is the bigger issue right now. And now with the current thinking of ionocytes and things like that, they don't know which cell type to target. Do you target the basal cells? Do you target the, that's the bigger issue right now.
All right. All right.
Yeah, I don't want to take your time. Organoids, maybe put it back.
We would love to.
Yeah. Okay, we'll have to close the session now. We have to start the other session because the speaker is waiting. So thank you, Naren. Thank you, Hema, for both these excellent presentations. And we will move on to the next session. Thank you very much.
Thank you.
Thank you, Professor Balram and Professor Naren for the exciting talks, and Professor D.N. Rao for chairing the session. We now welcome you to today's plenary talk 1. For this plenary talk, I welcome Neetu to introduce the chair, Professor Muniappa.
Thank you, Devraj. We will now move on to the first plenary talk, which will be chaired by Professor Muniappa. Professor Muniappa joined the department as a faculty in 1987 and is currently an honorary professor here. With the expertise in DNA damage recognition and repair mechanisms, his lab focuses on understanding the molecular mechanisms underlying chromosome synapses, homologous recombination, and double-stranded break repair. May I now request Professor Muniappa to kindly introduce the first plenary speaker.
Hello, everyone. Can you hear me?
Yes.
I would like to welcome you all to this first plenary lecture by Makuni Jairam. Professor Jairam is currently at the University of Texas at Austin. He will talk to us today on promoting CRISPR locus assembly and disrupting site-specific recombination. This is a subject in which we all should be interested in because of its immense implications in genome editing technologies. I'm honored to have this opportunity to say a few words of introduction about Jairam. Jairam is one of the most distinguished alumni of our department. Jairam, after he obtained his PhD in the department, he went to do first postdoctoral with Max Delbruck, the legendary phage geneticist, working on photoreceptors in phycomyces. Then he switched his lab as well as the interest to work on the golden standard of recombination, the yeast. And over the years, in the last 30 years or so, Jairam has made a kill in studying the topological interconversions of DNA using the East 2 micron plasmid as a model. Jairam has been an household name, including my own, in the field of site-specific recombination. He is also well known for his unique style of traditional chuck and talk teaching methods and for its very crisp presentations in academic conferences using again chalk and board. I'm not sure whether he's going to do that today, but let's wait and see. Another person I know who follows the similar kind of style of presentation is Frank Stahl, who is the DNA replication field. Well, I hope we'll have enough time at the end to quiz Jairam in the Q&A session. And please join me in welcoming a warm welcome to Professor Jairam, especially a Bangalore welcome to Professor Jairam to make his presentation on, again, the CRISPR locus assembly and disrupting site-specific combination. I look forward to it, Jairam. All yours.
Thank you. Can you all hear me? I
Yes,
assume yes.
Yes, yes.
Good.
Yes, yes.
Professor Muniappa has set a very high bar for me, and I'll do my best to live up to at least part of the expectations. It's always a special pleasure to return to Bangalore and to the Institute, even vicariously, as is the case this time. My sincere thanks to Professor D.N. Rao and his fellow organizers. My life as a practicing biochemist, which started here, is now half as old as the biochemistry department. August centers of learning like this department are ageless, immortal. They become renewed, grow more vibrant with the passage of time. If I can go to the slide, can I share screen here? Let me try that. This is the old biochemistry building of my student days, austere with Spartan amenities.
Jairam, we see in the presenters mode.
Can you switch to full screen, Jairam?
No, it's yeah.
Is it not full
screen? Now it is, yeah.
Okay, are you good?
Good, good.
Is that good? Can you all hear me? Am I
crystal clear?
Yeah, is this good now? Is this full
Very
screen
good, yeah.
or not?
No, it's good. It's good. I think you can proceed.
Okay. And look at this sprawling new building that houses biochemistry now. There's no doubt that the department will continue to enhance its reputation and prominence in the years and decades and centuries to come. If only time were as kind to mortals like me that walk the halls of the old building. To prove my point, I'm going to show you a picture of me then and one of me now. But then I thought the better of it and decided to spare me the embarrassment and spare you the discomfort. So on to my scientific talk now, which is focused on the contrasting roles of water in two biological contexts. One, CRISPR locus assembly, and two, site-specific DNA recombination. So let me first give credits to colleagues and collaborators here at home in Austin and collaboraters from Łódź, Poland. The brunt of the work was carried out by my long-suffering assistant, Chen Ma, and Ashik Kachru was a student here in Bangalore with Dr. Mahadevan. He has gone on to do some impressive genomics on the functional complementation of yeast genes by their human counterparts. St. Georgi described water as the hub of life, the matter and matrix. Life is water dancing to the tune of solids. Poetic, but with a bit of poetic license. Water is not an unmixed blessing in biological reactions. It has a bright side and a dark side. So first to the dark side, and the context here is site-specific DNA recombination carried out by the tyrosine family recombinase FLP or FLIP. A quick glance at the catalytic center of the FLIP active site, which contains a conserved tyrosine family pentad comprised of two arginines, 191 and 308, and please pay attention to them. We'll return to them soon. A lysine, a histidine, and a tryptophan, and then an invariant tyrosine nucleophile, which is directly involved in the strand cutting step of recombination. So in the transition state, the sessile phosphate non-bridging oxygens are stabilized by the arginine side chains, and the tyrosine becomes covalently linked to the 3' phosphate. So the first step of recombination involves the cleavage of two strands, one pair of strands, resulting in the formation of this phosphotyrosyl bond and an adjacent 5' hydroxyl. These hydroxyl then attack the phosphotyrosine bridges crosswise to form a pair of recombinant DNA strand in this Holiday junction intermediate. The chemistry carried out on the red strands here will exchange these strands and complete recombination. The point I want to make is that the strand cleavage step and the strand joining step are both vulnerable to interruption by water. At 56 molar concentration, water can outcompete the tyrosine hydroxyl or the 5' hydroxyl, and the result would be hydrolysis of either the DNA phosphodiester or this 3' phosphoryl tyrosine diester. The result then is the breakage of these strands, hydrolysis, loss of the conserved energy of the phosphodiester bonds, and termination of recombination, leaving in its wake damaged DNA strands. So question is, where does the protection come from? In the case of Vaccinia topoisomerase, which follows the same chemistry as tyrosine recombinases, the vulnerable entity is this phosphotyrosyl bond, and the electrostatics, the negative charge on the phosphate, is what is responsible for shielding it from water. It turns out that in the case of FLIP, the phosphate charge does offer protection, but it's much less impressive than in the case of the topoisomerase. So where is the source of protection against water during FLIP recombination? So we turn to the active sites. For example, could the electrostatics of the arginine side chains be shielding the phosphodiester in DNA or the 3' phosphotyrosine bond from attack by water. If you mutate this arginine or this one to an alanine to query their protective role, then the active site becomes completely dead on a normal phosphate-containing substrate. But there is a solution. So here is the phosphodiester. The O1 and O2 are the non-bridging oxygens, the 3' chain and the 5' chain of the DNA backbone. If you remove the negative charge on the phosphate by substituting either O1 or O2 with a methyl group, then the arginine 191 alanine mutant or the arginine 191 arginine 308 alanine mutant will become active on the methyl phosphonate substrate. You will also notice that by substituting one of the oxygens with methyl group, we have turned this symmetric phosphodiester into an asymmetric or a chiral center. So you have two stereoisomers, the RP form that retains O2 and the SP form that retains O1. All the initial reactions that we will do are done with a racemic mixture of the two stereoisomers. So let me show you what reactions we do. We assemble a very simple substrate that contains one flip binding element, which binds a monomer of flip represented by these parallel arrows, place a 5' p32 label to follow the reaction. And this dot here represents the sissyl phosphate, now methyl phosphonate in our case. And the bottom strand, the 5' hydroxyl, is phosphorylated. So the first step of the reaction is cleavage by the tyrosine, formation of this phosphotyrosyl bond. And if there is hydrolysis of this methyl phosphonate or this tyrosyl methyl phosphonate bridge, then you get this labeled product, which we call the hydrolysis product or HP, and that's shown here. So with the wild type flip, which contains both arginine 191 and arginine 308, hydrolysis is a slow reaction, but detectable. And you can see the relative amount of the product compared to the substrate. Now, if you mutate the tyrosine nucleophile to a glycine or an alanine, then you block this reaction. So all the hydrolysis is from the DNA phosphodiester, in this case, methyl phosphonate. And that's the reaction shown here. So if you compare these two, you might think that it's the tyrosyl phosphate that's more vulnerable to hydrolysis. But that may be a bit misleading because in this case, there is no 5' hydroxyl to compete with water. So essentially, water has a free rein. But the point is, hydrolysis in the case of the wild type flip or the flip Y343G mutant is a detectable reaction. So water indeed can pose a threat to recombination. And now if you look at the mutant, first look at the arginine 191A mutant and the double mutant in which we have mutated the tyrosine nucleophile to a glycine, the picture looks almost the same as the wild type reaction that you saw in the previous slide. In other words, losing the arginine 191 residue does not make the DNA phosphodiester or this phosphodiester any more vulnerable. than the wild type situation. So R191, arginine 191, probably is not involved in protecting against water. And so if you go to the next slide now and look at arginine 308 to alanine mutant, now you see a very different picture. Now the kinetics of hydrolysis have become much faster, and the overall amount of hydrolysis is much higher. Not only that, if you look at the flip R308A and flip R308A Y343G double mutant, there is really no difference between the two. In other words, when you use the arginine 308 residue, it's the DNA phosphodiester that becomes vulnerable to attack by water, and protection is offered by the arginine, presumably by side chain electrostatics. So if you want to look at the cleavage reaction and the joining reaction, the two together make recombination, we can make the substrate a little different. All you do is remove this phosphate and replace it with a hydroxyl. And let's look at that reaction now. So in this case, the cleavage reaction produces the phosphotyrosyl intermediate. And then this hydroxyl, the 5' hydroxyl, can now attack that phosphotyrosine bridge to form this pseudo hairpin recombinant product, which we call the joined product. So you can see both hydrolysis and joining in this reaction. And so first, let's look at a reaction where this is the normal phosphate DNA. And with wild type flip, you see that the recombination is a highly efficient reaction. And it's a well-protected reaction because there is hardly any hydrolysis product. Now, if you do the reaction with either the 191A mutant, arginine 191A mutant, or the arginine 308A mutant, you get no reaction at all. Now, the methyl phosphonate substrate is a very sluggish one. And with wild type flip, now you see that the recombination is a weak reaction, and the more prominent reaction is hydrolysis. So the phosphate electrostatics also must play a role in protection, but as I indicated to you, it's much less impressive than in the case of topoisomerase. Now, here is the interesting result. With this methyl phosphonate substrate, the arginine 191A mutant is much more active than flip, gives you fairly robust recombination, and the hydrolysis is not increased any further, consistent with no protective role for arginine 191 as we saw in the previous reaction. Now with arginine 308A mutant, the overall active site power is the same, but now the entire product is hydrolysis with very little recombination. In other words, if you remove the negative charge on the phosphate, either of the two conserved active site arginine residues becomes totally dispensable for active site function. Here, it's recombination. Here, it's all hydrolysis because there is no protection because you have lost arginine 308. What we have done basically is to compensate an active site mutation, the loss of a positively charged side chain, by making a corresponding substitution in the chemical group, namely removing the negative charge on the phosphate with which the active site interacts. Here then is a chemical equivalent of true genetic second site suppression, except that the second site is no longer in the protein, but in the DNA substrate. But then come to think of it, once the transition state is formed, there is really no difference between substrate and enzyme, as they have shed their individual identities and formed a sort of chemical amalgam. Now let's return to the phosphodiester. Once again, the non-bridging oxygen atoms, the nominal negative charge in the ground state. And so in the RP stereoisomer, you have O2 intact, O1 no longer there. And in the SP stereoisomer, you have O1 intact and O2 is gone. Now we can use the purified stereoisomers in our reactions to query the role of the non-bridging oxygen atoms, O1 and O2, and their interaction with the arginine residues during recombination. Or we can use them to figure out the stereochemical course of flip reaction. And so let me show you the reactions now. So here is the SP reaction, O1 intact, RP reaction, O2 intact. And you can see that there is a strong SP preference for wild type flip. That is, O1 and its contact with the arginine is what determines the normal stereochemistry of the reaction. Now, if you look at the arginine 308A mutant, the stereochemical preference is the same, or in other words, R308, arginine 308 is not critical for the normal stereochemistry of recombination. So if you look at the other mutant, arginine 191A, now the reaction has lost its stereochemical preference. Now SP and RP are equally reactive. If anything, RP is slightly more preferred. So in other words, arginine, in the absence of arginine 191, you lose the stereochemical preference of the normal reaction, or arginine 191 is what is more important in determining the normal SP preference of the reaction. So let me summarize this point. So water poses a serious threat to flip recombination by hydrolysis of either the 5', 3' DNA phosphodiester or the 3' tyrosyl phosphodiester. And arginine 308 electrostatically protects the phosphodiester from DNA hydrolysis. The catalytic function of the recombinase active site, lacking a conserved arginine residue, can be restored by electrostatic compensation in the substrate DNA. The recombination stereochemistry is specified by O1 contact through the conserved arginine dual, and arginine 191 is dominant over arginine 308 in determining the normal SP stereochemical preference of recombination. So I'm going to stop sharing the screen here, and maybe if you have questions or comments, we can pause for a couple of minutes if that's okay with everyone.
Probably we could pick it up at the end here.
I hear a question.
No, I think I was just suggesting that whether we could touch upon the questions at the end, or if somebody has a very pressing question, they can come up with.
Okay, in the absence of questions, let's continue to the next part.
Jairam. So,
hi, Apurva, how are you?
Yeah, how are you, Jairam?
I'm
Yes.
doing all right, yeah.
Yeah. So I'm just trying to, remember the work actually that when I did in your lab actually. This is essentially the recombination. I was just trying to understand that what would be the role of tryptophan 330 actually.
Oh, I think it orients the tyrosine through a hydrophobic stacking interaction.
Okay, so
there is also another aromatic residue elsewhere, non-immediately related to the active site, that sort of stacks the tyrosine between two flat aromatics and orients it for cleavage of the phosphate.
So is that the tryptophan and tyrosines are pretty close to each other that to form the aromatic contact stacking interaction?
Yes, tryptophan is 330, I believe, and tyrosine is 340, and in the structure you can see them layered one over the other.
Okay. Thank you.
Sure. Okay, maybe I should go on to the next part. So we finished the dark side of water, and now I'm going to tell you the bright side of water in a different biological context. So let me share the screen again here. Okay, I hope everybody is with me. And so the context now is the assembly of the CRISPR locus. In particular, the reaction that we are talking about is the excision of the protospacer from an invading DNA by the Cas1, Cas2 complex, and the insertion of that protospacer into the CRISPR locus as the spacer or an immunologic memory, the bacterial immunologic memory of this infection. And the reaction is chemically related to recombination. The theme is slightly different. In this case, it's DNA transposition. And the protospacer is the transposon, and Cas1, Cas2 is the transposase. So normal transposition reactions include a transposon, which is usually several thousand nucleotides long. And in the case of transposon phages, the length would be something like 50,000 base pairs. And the target site on the recipient DNA would be normally spaced anywhere from 5 to 9 base pairs, depending upon the transposon. So given the large length of the transposon, there is really no difficulty orienting the 3' hydroxyls for attacking these target phosphodiesters. So the intermediate of transposition contains this transposon and the target DNA pried apart on either side as single strands, and then gap-filling DNA synthesis and ligation will complete transposition. So that's the same mechanism that's used by the Cas1, Cas2 transposase to insert the protospacer transposon into the CRISPR locus. But here there is a serious problem. So the normal transposon is thousands of nucleotides long. The protospacer transposon is only 30 base pairs long. So it's a miniaturized transposon. And given the persistence length of DNA, there is no way you can orient these hydroxyl to attack phosphodiesters spaced 5 or 9 base pairs apart. So as the saying goes, if the prophet cannot get to the mountain must come to the prophet. So what has happened is the target of transposition in the CRISPR locus, and what I have shown here is a minimal CRISPR locus containing the leader sequence, followed by the repeat in orange, leader in red. And the repeat is the target and the spacer. And this would be followed by a second copy of the repeat, a third spacer, a second spacer, and so on and so forth. So the target, instead of being 5 to 9 base pairs, is now the same length as the protospacer, 30 base pairs long. But that's only postponing the topological problem to the next step, because in this integration intermediate, where the protospacer has attacked the 2 repeat phosphodiester targets, one at the leader repeat junction on the top strand, and on the bottom strand, the repeat spacer junction. That intermediate is under torsional stress, and it's believed that either DNA replication or transcription relieves that stress to form this intermediate where the repeat strands are played out as single strands, and these gaps are now filled again by repair synthesis and ligation. So the best way to analyze this reaction is actually do the reaction with Cas1 and Cas2. So that's what we do here. We place the minimal CRISPR locus on a supercoiled DNA, and the protospacer is tagged by fluorescence. So if you get an integration, double-ended integration, both strands integrated, you would get this relaxed circle, and potentially one-ended integration could give you either this product or this product. I should tell you that integration is a two-step event. The first integration always takes place at the leader repeat junction, or in our previous slide, this phosphodiester bond on the top strand. Okay, when you do the reaction, and these should migrate roughly with the same mobility as a relaxed or a nicked DNA. But if you look at the amount of product, that is minuscule compared to the amount of the substrate or the fluorescence tagged protospacer. But this is not because Cas1 and Cas2 is inactive in reaction, because if you stain the same gel with ethidium bromide, you see that the supercoiled DNA has now been relaxed into this ladder of less supercoiled topoisomers. So what's happening here is the integration starts with the protospacer attacking the leader repeat junction phosphodiester to form this semi-integrant. But then the 3' hydroxyl of the leader attacks that newly created phosphodiester junction to disintegrate the protospacer, disintegration reaction. So what's happening in the supercoiled substrate and this linear protospacer is this Sisyphean cycle of semi-integration and disintegration, leading to this relaxed isomers of the original supercoiled circle. And that is better seen in the slide. You can see the relaxed topoisomers, and the optimal reaction occurs at about 2-to-1 ratio of Cas1, Cas2, consistent with the structural and other biochemical data. What if we provide the target as a linear DNA molecule? Again, leader, repeat spacer, red, orange, or yellow, and blue, and the color scheme is followed throughout, and protospacer is shown in green. And this sort of reaction is done with the strand labeled at the 5' end with P32, either on the top or the bottom. And so if you get integration of the strand at the top phosphodiester leader repeat junction, you get an 89 nucleotide product. If it happens here, you get an 81 nucleotide product, and these are the markers. And the real reaction shows you both products, even though the reaction is not very robust. So it's detectable, and this is the unreacted protospacer. So is the weakness of the reaction due to disintegration again happening in the linear target, as we saw in the supercoiled target? So to follow integration and disintegration, we assemble a substrate by hybridizing oligonucleotides that resemble the first integration event. So this strand is integrated here, and the free protospacer strand we can label at the 5' end with P32. And so if this strand is integrated, that is, the second integration event happens here at the RS junction, then we will get a product that is 81 nucleotides long. And sure enough, that reaction happens with fairly rapid kinetics. But you also see an 89 nucleotide product that arrives with slightly slower kinetics. And how does that happen? That can happen if this hydroxyl attacks this phosphodiester here and restores the leader repeat junction, disintegrating the protospacer. And once that phosphodiester is restored, the disintegrated protospacer can integrate here, and that would give you the 89 nucleotide product. So this must arise as a result of disintegration and reintegration. So how do we follow disintegration? We can modify the substrate slightly, replace this label by removing it from here and placing it here on the leader strand. And so that is the substrate here. And if disintegration happens and the LR junction is restored, you get a 92 nucleotide disintegration product. In fact, that reaction is as rapid as the second integration event and much more robust than the second integration event. So is the second integration event coupled to disintegration because of the torsional strain that we talked about, the shortness of the protospace? So to ask that, we have to design a substrate like this. So the question is, if we block disintegration, can we block the second integration event? That is, if there is no disintegration, will we block the integration of this strand here, giving rise to the 81 nucleotide product? So substrate 1 is the one that you saw earlier, the 3' hydroxyl here, fully competent in integration. So in the modified substrate, we change this hydroxyl to an oxy group, dideoxy, and therefore this is incapable of disintegrating the integrated strand. And in the third substrate, we remove this leader strand altogether, so there is no 3' hydroxyl for disintegration. But in all three substrates, you see the 81 nucleotide product. Of course, it's a weak reaction, but really there is no difference between the disintegration competent substrate and the disintegration incompetent ones. But then you think of the flip reaction, you think of water and its 56 molarity in biological reactions. And you wonder whether water is acting as the nucleophile for disintegration. So to address that, we make a similar substrate, but we place the label in a different position. So now you follow the disintegration by placing the label on the integrated strand. So if there is disintegration, you release the strand as this 226 nucleotide disintegration product. So this is a case where the 3' hydroxyl and water can both act as the agents of disintegration. In this case, there is no 3' hydroxyl, so this gives only water-mediated disintegration. And so this is the sum of 3' hydroxyl and water-mediated disintegration, and this is just water-mediated disintegration. And you see that if you calculate, quantitate these reactions, water-mediated disintegration is at least half as efficient, or 60% as efficient as 3' hydroxyl-mediated disintegration. So to address the question of disintegration during second integration, the only reasonable way to answer that is to know the fate of both strands. This strand which integrates and this strand which potentially disintegrates during integration of the second strand. So how do you ask the question of disintegration coupled integration? As I said, the only way to do it is to follow the fate of both strands at the same time. And that is quite an arduous challenge, almost impossible to meet. But then almost impossible means it is still possible. And so we design a substrate that looks like this in the next slide. What we do is tether this integrated strand to the unintegrated strand via the repeat and the spacer using a long single strand nucleotide tether. This is 30 T residues or something like that. So now you can follow both strands at the same time. And so in this set of reactions, we place the label on the 5' end of the integrated strand. And so if this integrates here and there is no disintegration, you can add up the number of nucleotides in each of these segments. What you would get is 183 nucleotide labeled product. And sure enough, you see that, but that's an extremely weak reaction. But what is stronger is disintegration of the strand and release of this 26 nucleotides fragment that is shown here at the bottom. So disintegration, as we saw in the previous reactions, is a much stronger reaction than second integration. This substrate will not tell you whether integration occurred with disintegration of the strand, because that would automatically remove the label from the product, and therefore it would be invisible on this gel. But to see the product, we can do a trick. We can replace the label here, remove it from here, and place the label at the 3' end on the bottom strand here. So that's done by adding a single labeled nucleotide to the 3' end using the Klenau polymerase reaction. So if you look at the next slide, so now you can follow the product of integration without disintegration, which should be 184 nucleotides long, and that is this product here, corresponding to the 183 nucleotide product that you saw in the previous slide, this one here. And if the second integration event is accompanied by disintegration, then the product will be shorter by 26 nucleotides and would be 158 nucleotides long. And indeed, that is the product here. And in fact, it's much more abundant than integration without disintegration. In other words, almost every second strand integration event is accompanied by disintegration of the originally integrated strand. So in the Cas1, Cas2 mediated transposition, disintegration is a very prominent reaction, and you almost never get two-ended integration events. And so how do you fit disintegration, couple integration into a model for protospacer acquisition by the CRISPR locus? So let's go to the next slide, and I'll try to explain this briefly. So Cas1, Cas2 forms a complex with a protospacer transposon, integrates the first strand at the LR junction. And now if the second integration over here is coupled to disintegration by this 3' hydroxyl, you will restore the LR phosphodiester here, and you will form this semi-integrant on the bottom strand. Now this is competent for reintegration here. So these two semi-integrated states can sort of oscillate one between the two forms, between themselves. Now, if after the first integration, the second integration is followed by water-mediated disintegration here, that would relieve the torsional strain and generate this intermediate where you have a gap in the bottom strand corresponding to the repeat sequence and a gap in the top strand corresponding to the repeat sequence plus a nick. So this intermediate has a gap in one strand, a gap plus nick in the other strand. And this will generate an identical intermediate, except that now the gap plus nick is in the bottom strand and the gap is in the top strand. And this is a structure that the DNA repair machinery of the bacterium would relish to act on. And so either displacement DNA synthesis or gap repair and ligation would complete spacer acquisition by the CRISPR locus. So to summarize quickly, the shortness of the protospacer transposon engenders torsional stress during its insertion at the CRISPR locus. The 3' hydroxyl or water-mediated disintegration relieves this torsional strain. 3' hydroxyl-mediated disintegration restores the phosphodiester bond that's targeted for new integrations, and potentially oscillations of the semi-integrated protospacer can occur between the LR phosphodiester junction or the RS phosphodiester junction on the top and bottom strands respectively. And water-mediated disintegration generates an intermediate which by the action of bacterial DNA repair machinery matures into a normal spacer integrant. The point I want to make is that both the 3' hydroxyl and water-mediated disintegration reactions are functionally relevant to spacer acquisition. 3' hydroxyl, as we noted, by generating a target phosphodiester competent for integration, and water by relieving the torsional strain and generating a repair-ready intermediate. One could draw a rough analogy to the torsional or topological challenge faced by the DNA replication machinery. Here, the positive torsional stress built up ahead of the replication fall is relieved by strand cutting and joining by topoisomerases, which might be looked upon as both DNA damaging and repairing enzymes at once. The transesterification chemistry that they utilize ensure that strand breakage and strand rejoining occur in quick succession, leaving no room for net DNA gain or DNA loss. So the contrasting mechanisms for topology simplification used by topoisomerases on the one hand and Cas1, Cas2 on the other are consistent with their biological purposes. one to maintain genome integrity during replication, and the other to bring about an orderly DNA rearrangement, in this particular case, at the CRISPR locus on the bacterial chromosome. So let me leave you then with this image of disintegration coupled integration of the protospacer. Here is disintegration of the protospacer strand at the left, repair, and then persistence of the spacer at the CRISPR locus at the right. And below it is an artist's rendering of disintegration and persistence, seemingly of a very different sort. But then on second thought, protospacer to the bacterium and memory to the artist are related in a sort of beguiling manner, for both signify the remembrance of events from the past. So with that, I leave you to think about the role of water, both as a promoter of biological reaction and as a disruptor of biological reactions. And I welcome questions and comments from you. Thank you.
That was a great, very crisp and fascinating presentation, Jairam, as always.
Thank you. And I'm sure you want to ask me something or criticize me as usual. Always.
I have a very naive question. I think I'll come back at the end. Anybody else has a question or a clarification or a comment for Jairam?
Jairam, I'm wondering, I mean, you used a very interesting small system to look at the integration for the photospacer. How this caste-based system affects the non-random deletions or insertions away from it?
No, the transposition mediated by the Cas1, Cas2 transposase is a site-specific transposition because the protospacer 30 base pairs is cut out by Cas1, Cas2, and probably Cas9, and a couple of other Cas proteins are involved. By recognizing the PAM sequence, which is later involved in the interference complex as well. So you define the end by PAM, cut out 30 base pairs, and then you integrate it into the target site. So this is site-specific. Most transposons are only half site-specific. They are specific to the ends of the transposon, but then they tend to integrate the transposons randomly in target DNA because the biological purpose of transposition is to spread the transposon. So specificity would minimize the spread, and therefore they have evolved to do the first excision event specifically and the second integration step sort of randomly.
I have a naive question for you, Jairam.
Sure.
In your second part of presentation with the Cas9 and Cas2, Cas1 and Cas2, my impression is for your reactions with branch DNA substrate, Cas1 alone would do the job in the kind of products that you have seen. So if that were to be true, what is the role for Cas2?
Yeah, Cas2, first I have to clarify that Cas1 alone will not do the second integration event because the organization of the Cas1, Cas2 complex is sort of like a butterfly, 2 Cas1 units here, 2 Cas2, Cas1 units here, and a dimer of Cas2 sort of holding the wings of the butterfly as a body. So you have to recognize both ends, the LR repeat phosphodiester on the top strand. So that's where the first integration happens. And it's sort of, the same complex carries out the second integration by sort of measuring the distance, 30 base pairs. That's where the second active site is. So the Cas1 provides the active site. So the reaction, the catalysis is carried out by Cas1 entirely, but the structure of the complex requires Cas2.
Oh, that's very nice. But Cas2, just remind me, is an exonuclease. So
yes,
my impression is
Cas1 and Cas2 have non-specific nuclease activity. And so it's not, so if you just incubate any DNA, particularly if you have single strands or AT-rich sequences that breathe, either Cas1 or Cas2 alone will cut DNA randomly. So it's not clear what that single strand nuclease activity represents. It's possible that during the original processing of the protospacer, it's some of the bacterial nucleases, for example, REC-BCD, might be involved in cutting the original DNA, the invading DNA to some manageable DNA fragments, and then Cas1 and Cas2 comes along, the complex comes along, and then cuts out the protospacer. But preceding that precise excision of the protospacer, there might be some endonucleatic cleavage in which the Cas1, Cas2 single strand nuclease activity might be involved. It's not at all clear why these activities are there. Certainly they are a nuisance as far as the specific integration, disintegration reaction goes.
Great. Anybody else? Well, if there are no questions from others, I would take this opportunity to thank Jairam again and for very lucid and engaging presentation. Thanks, Jairam. I hope you will visit us in the near future and tell us more about this system.
Much success. Thank you all.
Thank you again.
Thank you very much, Jairam. Thank you.
Over to you guys. I'm done.
Thank you, Professor Jairam, for the wonderful talk, and Professor Munyappa for chairing the session. We will now move ahead for the next plenary talk. For this talk, I request Devraj to initiate the session.
Thank you, Neetu. This is the last event of today and the second plenary talk. This talk will be given by Professor Chetan Khosla, and the session will be chaired by Professor D.N. Rao. Since Professor D.N. Rao has already come in session 2, we do not need any further introduction. So without any further ado, may I request Professor D.N. Rao to kindly introduce the speaker.
All right. Good evening and welcome to the last talk of this today's program. This is going to be given by Dr. Chetan Khosla. Dr. Chetan Khosla is currently the Wells Rauser and the Harold Petitprint Professor of Chemical Engineering and Chemistry at Stanford University. He got his B.Tech in Chemical Engineering from the Indian Institute of Technology in Bombay, and then went on to do his PhD at the California Institute of Technology in Pasadena in 1990. He did his postdoctoral work at the John Innes Center with David Hopwood, and then became a professor at the Stanford University, and he has been there since 1992, also the chair of the program. He had been the Baker Family Director of Stanford Chemistry, Engineering, and Medicine for Human Health till 2020, and his area of research largely focuses on how to understand and build molecular understanding of polyketide synthesis. And the second area is focused on the biochemistry of celiac disease, particularly involving the tissue transglutaminase. His early research on natural product ketides led to the founding of one company called the Cosan Biosciences in 1995. And his work on the celiac sprue has resulted in founding of the Alvine Pharmaceuticals at the start of a non-profit celiac sprue research foundation. Professor Chetan Khosla was elected to the National Academy of Engineering in 2009. And in 2020, he was elected as a member of the National Academy of Sciences. And for his work on engineering molecular assembly lines, developing metabolic engineering technologies, and advancing biopharmaceutical discoveries. He's a fellow of the American Academy of Arts and Sciences and a recipient of the Distinguished Alumnus Award from Caltech. So it's a pleasure and privilege to invite Professor Khosla to give this talk. Chetan, it's all your show now.
Thank you. Thank you very much for the introduction. And let me start by thanking the organizers of this very, very special event to give me a chance to participate in it. I've been looking forward to it. Initially, I was looking forward to it, to being there in person, to celebrate and congratulate all of you who are the guardians of this most special institution in India. The centenary of this institution is something for all of us to celebrate. Every scientist on this planet, but certainly scientists of Indian origin, have a special reason to celebrate it, because not just because of the great science that the department has produced over the decades, but also because of the great scholars that have passed through its hallways and had a huge impact on the broader world of science and society. And most of us are beneficiaries of that. And most recently, but equally importantly, I think more than most other examples, the impact of a really special center, an academic institution, is often that it becomes the seed from which knowledge-driven enterprise blossoms and grows into something really special. And when the world looks at where Bangalore is today as a thriving center of knowledge-based industry, one sees your department there at ground zero. And so I humbly salute to you and your academic ancestors who have not just built this very special place, but also nurtured it with the highest possible standards and brought it to its current level of prominence. So thank you for letting me participate in this event. I want to spend the next 45 minutes or so sharing with you my laboratory's contributions to the understanding of one problem in biochemistry that has been close to my heart since we launched our laboratory. It'll be 30 years this Christmas since I started my laboratory at Stanford. And so this problem essentially defines our journey over the past 30 years. And the problem has to do with understanding at a mechanistic level a class of very remarkable enzymes called polyketide synthases that operate on an assembly line type of a paradigm. I show you here examples of a few of numerous antibiotics that are made by these assembly line polyketide synthases. And at the time when my laboratory started studying these assembly line polyketide synthases, which kind of predates the origin of this graph, there was really only one polyketide synthase known to humans. And even for several of the first few years, there were only a handful of polyketide synthases because the effort it took to genetically characterize these enzyme systems, these extraordinarily large enzyme systems, was quite Herculean, the equivalent of 1 or 2 or sometimes even 3 to 5 PhD theses to be able to do that. And so what you're seeing in this red line over here is the growth rate of the diversity of polyketide synthases which made known antibiotics. And this has steadily grown over the past 30 years, albeit at a relatively slow pace. In contrast, what has happened is that there has been an explosive growth thanks to genomic sequencing of what we call orphan assembly line polyketide synthases. These are enzyme systems that essentially get decoded when people do large-scale genome sequencing projects and deposited in GenBank or equivalent public databases. But nobody knows what these antibiotics are, what these assembly lines are doing, what they're making chemically, or why they're making what they're making. And so back when we started studying these systems in 1992, our desire was primarily to understand their mechanism, and that desire was motivated by the engineering possibilities afforded by understanding these mechanisms in the context of a handful of very well understood antibiotic biosynthetic pathways. Today, however, there is an entirely legitimate second motivation for mechanistic understanding of these enzymes, and that is to de-orphanize these orphan assembly lines that are growing at an exponential pace. The prototypical assembly line polyketide synthase that my lab studies is this enzyme system that is responsible for making 6-deoxyerythronolide B. It's this beautiful 14-membered macrocyclic ring system that is the core of the antibiotic erythromycin. This enzyme system is very typical of these polyketide synthases in the sense that it has about 25 active sites, different types of active sites that are organized in multiple modules, where each module is responsible for one round of polyketide chain elongation and elaboration, together with stereochemical determination of the various relevant stereocenters of the final natural product. Now, for those of you who are not intimately familiar with these enzyme systems, the first module of this erythromycin synthase, this 6-deoxyerythronolide B synthase, the first module over here is fairly representative of the way these modular systems work. It harbors 3 evolutionarily unrelated enzymes, an acyltransferase, a ketosynthase, and a ketoreductase, each with their own discrete active sites. So you can think of each of these little blobs over here as a typical enzyme-sized domain within this larger enzyme system. And then there is a tethering protein called an acyl carrier protein, or ACP for short. Now, the overall catalytic cycle of the first module of the 6-deoxyerythronolide B synthase involves 5 distinct reactions, but I want to draw your attention to 3 of these reactions because they'll be important for the rest of my talk. This elongation event, which is formally a decarboxylative Claisen condensation-like reaction, is effectively the hallmark of polyketide biosynthesis. It is the reaction by which this nucleophilic malonate equivalent, in this case it's a methyl malonate equivalent, attacks an electrophilic thioester to form a carbon-carbon bond, and the net growth of the polyketide chain by 2 carbon atoms along the backbone. Now, in addition to this elongation event, there are two translocation events, which are formally thiol to thioester exchange reactions. This one results in the entry of the polyketide chain from the upstream module to module 1 of the erythromycin synthase. And this one results in the exit of the product of module 1 to module 2, the next module. So these thiol to thioester exchange reactions we're going to call translocation events for the rest of this talk. Now, a key invariant property of all assembly line polyketide synthases is that they operate as homodimers. What this means is that DEBS, or 6-deoxyerythronolide B synthase, is comprised of 2 copies each of these 6 modules, or 22 active sites. So this begs the question, is there a preferred pathway along which the growing polyketide chain is channeled as it is elaborated into the final product of this polyketide synthase? So more than 2 decades ago, Cami Cao and Rembert Piper in my lab answered this question by showing that chain elongation, that carbon-carbon bond forming reaction that I referred to, involves a collaboration between the ketosynthase of one of the monomers of each homodimeric module and an acyl carrier protein across the dimer interface. And so in this particular cartoon, I'm showing one monomer equivalent in the dark colors and another monomer equivalent in the light colors. And so when module 1, which is this yellow module, catalyzes one chain elongation event, the dark colored ketosynthase collaborates with the light colored acyl carrier protein to generate this acyl carrier protein bound diketide product. So elongation involves ketosynthase acyl carrier protein interactions across the dimer interface, whereas what Cami and Rembert showed was that chain translocation, the movement of polyketide chains between modules by thiol to thioester exchange reaction, the translocation event involves ketosynthase acyl carrier protein interactions within the same polypeptide. So once this chain is formed and bound to this light colored acyl carrier protein, it'll move to the light colored polyketide, the light colored ketosynthase of module 2. And that light colored ketosynthase will collaborate with the dark colored acyl carrier protein of the same module for that next elongation. So as you can see, each polyketide chain is kind of zigzagging its way across this homodimeric assembly line along a spatially unique pathway. And that's really important. So if you're like me, who you probably prefer to see these enzymes as molecules, not as cartoons. And so about 15 years ago, we formulated this 3-dimensional model of the 6-deoxyerythronolide B synthase. While several aspects of this model have evolved over time, its core features have more or less remained unchanged, and they've served us extremely well in our mechanistic investigations thus far. So in the model I'm showing you in this slide, the entire DEBS 6-deoxyerythronolide B synthase fits in a box about 350 to 400 angstroms on a side. So if you drew a cube with of about 400 angstrom side, this entire enzyme machinery would fit in that box. And if the protein were completely static through the entire catalytic cycle that produces one of those 6-deoxyerythronolide B molecules, then the growing polyketide chain that led to the formation of that product would have to be channeled along a trajectory involving about 20 plus active sites. And that trajectory, if you simply measure it as the crow flies, would exceed 500 angstroms in length. Clearly a spectacular feat by any measure. Now, in case you're wondering how we generated a model like this, it was through a divide and conquer approach. The full deoxyerythronolide B synthase assembly line has a mass that exceeds 2 million Daltons. So to give you a sense of scale, that is about the size of a bacterial ribosome. By now, we have about atomistic or near atomistic resolution snapshots of more than half of the pieces of the entire Debs assembly line. And these pieces have ranged between as little as 10 kilodalton fragments to as large as 300 kilodalton fragments. Now, given that the missing pieces shown in gray, for which we don't have direct atomistic insights, have homologs within the rest of the assembly line that are more than 50% end-to-end sequence identical, in this day and age, it's relatively easy to build reliable 3D models of the missing pieces. And so when you put all those pieces together with a few basic structural and mechanistic experimentally derived constraints, you end up with the kind of model that I showed you in the previous slide. Now, the main point at this stage that I want you to take away from my talk is that while cartoons like the kind that, say, I showed you here in this figure are still an effective way to communicate our hypotheses. So you can open your favorite issue of JACS or biochemistry or your favorite journal, and almost in every issue, you'll see one more article of an assembly line synthase that is described, and it's usually described in the language of cartoons. And while cartoons are an effective medium to communicate hypotheses, we're really past the era of Lego-based hypotheses involving these assembly lines. And so whether one is testing a mechanism or engineering an unnatural natural product, one's hypothesis needs to be formulated on a legitimate structural foundation of these enzyme systems if you're going to be taken seriously. Now, our research into these enzyme systems started with a fairly rudimentary goal, and that was the goal of assessing how modular these assembly line synthases are. So two early experiments that we did, summarized in this slide, did in fact suggest that assembly line polyketide synthases were quite modular. And they were modular both with respect to structure as well as function. So in this first experiment over here, we sought to shrink the 14-membered macrocyclic natural product by 2 carbon atoms into a 12-membered ring system, while at the same time constructing a chimeric module in which the catalytic domains of module 5 were recombined with the carrier domain of module 6, and the thioesterase domain that talks to this for cyclizing the final product. We recombine this to construct a chimeric module which releases exclusively a 12-membered natural product. And so this basic experiment suggested that it was possible to recombine modules and domains within modules to be able to generate what one called back then unnatural natural products. In this second experiment, what we wanted to do was to push this idea of generating constrained ring systems further by designing an even more truncated enzyme system that might make an 8-membered ring. And then we quickly realized that if this carbon atom here represents the terminal thioester forming carbon atom, you would have a delta hydroxyl in the growing polyketide chain that would preferentially attack the thioester to form a less strained 6-membered ring. And so if one wanted to generate an 8-membered ring unnatural natural product, one would have to formally figure out a way to enzymatically eliminate this hydroxyl group. And the way to do that allows us to test the modularity of these systems again. And that involves adding to module 2 two additional enzymes, a dehydratase and an enol reductase, that would further reduce this hydroxyl into a methylene equivalent, thereby facilitating the formation of an 8-membered ring. So experiments like this showed that these systems, these enzyme systems, had some level of inherent modularity. And that started motivating us to ask the question, well, what are the limits to the modularity of these assembly line polyketide synthases? It turns out that the limits are quite significant if you dig deeper. Probably the best example comes from the systematic work of Maya Klaus in my lab several years ago. What Maya did was she recombined the first module of the 6-deoxyerythronolide B synthase systematically with a panel of a dozen or so non-cognate modules from enzyme systems that made erythromycin, rifamycin, rapamycin, etc. And what she observed was that while every chimera produced the expected polyketide product in detectable quantities, when she reconstituted these chimeric systems in a test tube, she showed that their turnover rates of the chimeric systems were 1 to 2 orders of magnitude below those of the cognate system that nature had evolved. And so to identify the source of the kinetic bottleneck in these chimeric systems, Maya developed an assay in which she quantified the occupancy of the acceptor module at steady state. As you can see, if you compare to the natural system, which is fully occupied at steady state, most heterologous acceptor modules, their occupancy is extremely low. Moreover, there is a strong correlation between the acceptor module occupancy and its turnover rate. So for example, this particular module, which has a somewhat modest occupancy compared to many others, also happens to be the one that is significantly better at its turnover rate than many of the other modules. Going one level deeper, a bottleneck at the module-module interface would imply one of two things. It would either imply that the acceptor module here discriminates against a non-cognate substrate, since the rapamycin module 4 didn't evolve to accept the product of the erythromycin module 1. It discriminates against that non-cognate substrate, and that is the basis for the kinetic sluggishness. Alternatively, one could also come up with a hypothesis where the acceptor module is discriminating not so much against the substrate that is delivered by the upstream non-cognate module, but the protein itself that is delivering that substrate. And of course, you could have a combination of both. Now, it turns out that enzyme substrate recognition is not a major issue in these systems. How do we know that? We know that through experiments like the one that is described in this slide that have been done over the years in my lab. In this particular case, we're quantifying the specificity of the downstream modules of the erythromycin synthase for the diketide product of module 1. So this particular product that module 1 of the erythromycin synthase generates, we're asking the question, what is the kcat over km of these downstream modules compared to the native module? Now, Nick Wu's experiments over here taught us that module 6 over here actually has a higher substrate specificity for this diketide than the native module 2. And so clearly substrate specificity does not explain why Maya observed a 1 order of magnitude lower turnover rate of the 1,6 chimera as opposed to the 1,2 system. And going further, Maya observed that module 3 over here has yet another order of magnitude worse than module 6 in accepting the product of module 1. But clearly that is not reflected by the kcat over km of module 3 for the substrate of module 1. So if enzyme substrate recognition is not important, then what counts? For more than 2 decades, our data has consistently emphasized the dominance of protein-protein interactions over enzyme substrate recognition to explain the inherent limits to modularity of assembly line polyketide synthesis. This has been one of the key contributions of my former coworker and alumnus of your department, Rajesh Gokhale, to our field. So in one of the very first such studies, what Rajesh and his then student collaborator, PhD student collaborator, Stuart Suji, did was they essentially reconstituted the interface between module 2 and module 3 of the 6-deoxyerythronolide B synthase, and identified that short peptide linkers flanking successive modules played a very pivotal role in channeling biosynthetic intermediates across these non-covalent connected module junctions. So in this native system over here, you can feed a substrate of module 2, have it elongated and processed by this module. That product hops on to module 3, gets elongated and processed by module 3 and released as a product with kinetic parameters shown over here. If you take the short peptide, and just obviously these cartoons are not drawn to scale, to give you a sense of scale, this module over here has a molecular mass of about 300 kilodaltons as a homodimer. And this peptide over here is like a 30 residue type of a peptide. So if you take this 30 residue C-terminal peptide over here and replace it with its counterpart from a different module interface over here, you end up with a pair of modules that individually are perfectly fine catalysts unto themselves, but simply can't talk to each other. You can do the same experiment at the N-terminal interface of module 3 and bring in a heterologous peptide from a different module, and you end up with the same pair, with the same situation, a pair of modules that individually are good catalysts but are unable to talk to each other. And if you now take this chimeric module and combine it in the same test tube with this chimeric module, you end up with an enzyme system that is perfectly capable of talking to each other like the native system. So by now, the use of these docking peptides has become a real cottage industry in our field, in large part because they've proven to be highly modular protein-protein interfaces that can be deployed to facilitate the specific docking of any two polyketide synthase modules. Now, not all protein-protein interactions in these assembly line polyketide synthases are as modular as those docking sites that I just showed you in the previous slide. So for example, these acyl carrier protein domains onto which substrates and intermediates are alternately tethered, they're also involved in catalytically relevant protein-protein interactions. The catalytic cycle I introduced to you in the previous slide corresponds to the first module of 6-deoxyethranolide B synthase. Other modules of DEBs also operate on a very similar catalytic cycle. If one looks at the data for module 3 of DEBs, one can kinetically isolate the acyl ACP mediated chain translocation event that delivers the substrate of module 3 to it. And if one compares the specificity of module 3 for its cognate donor acyl carrier protein versus a non-cognate donor acyl carrier protein, keeping everything else the same, the docking domains, the substrate chemistry, everything else is the same, what one sees is that there is a 2 orders of magnitude kinetic hit in the specificity of this chain translocation event. One can also kinetically isolate the chain elongation event of module 3, and one sees more or less the same thing. The cognate acyl carrier protein is about 2 orders of magnitude better than non-cognate acyl carrier proteins. Now, back when this data started to stare at us in the face, it wasn't at all obvious how, or even if, nature would modularize the ability of these small 10 kilodalton acyl carrier proteins to be recognized by multiple enzymes. It turns out that there is, in fact, some degree of modularity associated with these molecular recognition events. So during chain translocation, the acyl carrier protein is recognized by its downstream module through this blue face of the protein, whereas during chain elongation, the same acyl carrier protein is recognized by its partner module through the orange face. Now, while this kind of an insight certainly provides some basis for engineering polyketide assembly lines, if this is all you have to work with, then you're in somewhat of an analogous situation to where researchers like Jim Wells were when they started off attempting to engineer growth factors before they decoded the structures of their receptors. To enable legitimate structure-based protein engineering, one needs high-quality insights into how each module alternately engages 2 acyl carrier proteins during chain translocation and chain elongation. Now, our quest to visualize these interactions went through a number of dead ends over the past many years. And so in the interest of time, I'll only tell you what worked. You all are probably familiar with FAB antibody fragments. Because FABs can be expressed in phage display formats these days, several groups have generated large, extremely diverse FAB libraries and use them to identify the rare FABs that are able to bind and stabilize proteins that are otherwise too dynamic for high-resolution structural analysis. And you've probably seen a number of brilliant examples of that in the literature over the past 20 years or so. So about 8 years ago, with the help of Charlie Craig's lab at UCSF, a student in my lab, Ted Lee, started to pan for FABs that selectively bound to individual modules of that Debs assembly line. And Ted was able to generate dozens of fabs that bound with nanomolar affinity to folded domains, modules, and submodules. I'm showing you here examples of a subset of antibodies he generated against a single module, shown over here. These FABs have proven to be unbelievably powerful tools for probing polyketide synthase function. For example, here I show you a panel of 5 FABs that bind to the same module, module 2 of Debs. This FAB1B2 blocks the chain translocation step. AC2 over here and BB1, they block the chain elongation step, but don't alter the chain translocation step. AA5 over here blocks the translation step, but leaves the other steps intact. And BH4 over here binds to a neutral site. Hello? Everything okay? I guess so.
Yeah, go on. Please
now
continue.
So these antibodies, these FABs, have proven to be useful not just for functional analysis of polyketide synthase modules, but they've also had a huge impact on single particle cryo-EM analysis of these modules and bimodules. Before we had these antibodies, we had been stuck with single particle cryo-EM resolution in the 10 or so angstrom range. I liken my encounters with cryo-EM before 2015 to those of a frustrated drug addict. They were doing enough to keep me hooked, but not enough to satisfy me. And then around 2015, something happened in addition for us to be able to get access to these antibodies from Charlie Craig's lab. Hua Chu moved from Baylor to Stanford. And Hua and his co-workers quickly realized that cryo-EM analysis, single particle cryo-EM analysis of FAB-bound polyketide synthases could allow us to get near atomic resolution. So they basically developed this amazingly sophisticated workflow. Some of you who've had some exposure to single particle cryo-EM by now have a feel for how this workflow goes to get to near atomic 3 to 4 angstrom level structures. And obviously we were able to do that in these kinds of systems. But what really blew my mind as we started getting down to that 3 to 4 angstrom resolution was that the discrete images Hua and his team started to generate on our systems at that resolution had all the hallmarks of discrete states in the catalytic cycle that we were looking at from a functional perspective. So here's a direct image of a module interacting with its own acyl carrier protein during chain translocation. There is absolutely no debate about what we're seeing in this image because one can literally clearly see the phosphopentethine arm of this acyl carrier protein snake its way into the active site of the ketosynthase. One can also see that the epitope that is involved in docking this acyl carrier protein to this module matches beautifully to that orange epitope that I showed you in that earlier slide. So clearly what we're seeing over here is catalytically legitimate. But perhaps the most important insight that emerged from cryo-EM analysis of these polyketide synthase systems has been the existence of asymmetric conformations of symmetric homodimers. Now, to date, until we started getting these images, every published structure from our lab or from any other labs of these dimeric polyketide synthases or even fatty acid synthases showed C2 symmetry of these homodimers. And so for the first year or two of our joint efforts with Wa's group, we kept throwing away these asymmetric states, thinking that they were garbage. And ultimately, Dylan Kogan mustered up the courage to start testing the hypothesis that these asymmetric states were in fact on-path conformations of this amazing catalytic machine. So for example, here you're seeing a state where you have one acyl carrier protein of the homodimeric module interact with the keto synthase of the other subunit. And that's exactly what 25 years ago, Rembert, Piper, and Kamikau's experiments had predicted. While the acyl carrier protein of the other monomer is simply too flexible to be visualized at this resolution. Conversely, the keto reductase domain from the subunit with the invisible acyl carrier protein is juxtaposed onto the keto synthase from the opposite subunit. And the plausibility of these interaction models is further underscored by the fact that the salt bridges that hold these domain-domain interactions together are derived from highly conserved residues in otherwise relatively unconserved portions of these assembly lines. And so this keto reductase domain, 1 out of 2 that is visualized in this, is now going to undergo a 90-degree rotation to dock on the keto synthase of its own subunit. And presumably that's what's going on with the other invisible keto reductase domain in this cryo-EM snapshot, which is why you can't see it. So this really begs a fundamental question that is perhaps at the core of understanding all these assembly line polyketide synthases. The sine qua non of these assembly line polyketide synthases is their ability to channel these reactive intermediates in a vectorial fashion. And so now that we've seen asymmetry as being inherent in these systems, if asymmetry is inherent in a directional, a vectorial catalytic cycle of this assembly line, one could ask the question, is asymmetry actually driving catalysis? Now, you're probably looking at me right now thinking I've gone nuts, but this is really not as ridiculous as it might sound. Because in recent editions of biochemistry textbooks, we have a growing number of examples where you have symmetric catalysts that are transiently desymmetrized to be able to drive them forward. And no other example is as elegant as the FOF1 ATP synthase, which makes ATP in all aerobic life forms. In this system, both the catalysts, the proton transporter, as well as the ATP synthase, are symmetric structures. The proton transporter is a C8 symmetric system, transmembrane system, whereas the ATP synthase is an alpha-3 beta-3 hexamer. And so people have asked for the longest time how directionality arises out of symmetry. And it turns out that the asymmetry of this coupling central stalk, which is a gamma delta epsilon type of a heterotrimer, allows the directional motion of rotation of this proton transporter to energetically couple to the asymmetry and generate asymmetry in this otherwise symmetric. So what is symmetric on a holiday, on a working day, goes through multiple transient asymmetric states. And that is a critical principle in a lot of molecular machines today, where energetic coupling is key. Now, in a polyketide synthase assembly line, energetic coupling is absolutely essential for vectorial biosynthesis. What do I mean by that? In this catalytic cycle of this particular module, we have an analogous type of energetic challenge, energetic coupling challenge. The reactions that impart directionality to the assembly line, that's these two translocation events, they are thermoneutral, thiol to thioester exchange reactions. Delta G is 0 to first order. And so the only exergonic event in this entire catalytic cycle is here in this decarboxylative chain elongation event. And so a question that comes up is, how does nature harness the delta G at this step to be able to make these two thermoneutral events directional? And how does asymmetry of these systems influence that outcome? Several years ago, based on multiple lines of data, my lab proposed a model for how this energetic coupling occurs. We proposed that exergonic chain elongation induces a conformational change. So this chain elongation step induces a conformational change in the module that precludes back transfer of the chain via thiol exchange react, thiol thioester exchange, as well as the entry, premature entry of the next equivalent of the substrate into this module for a reasonably long time in order to give the module adequate time to finish its unfinished business, to catalyze all the post-elongation chemistry that is needed, and then pass the chain on to the next module. Now, because we didn't have a way to visualize these conformational changes, we use the term turnstile to describe this mechanism, because in effect, our data was arguing that elongation results in closing something like a turnstile for long enough in this module to allow the module to complete unfinished business in an orderly manner and then start the next cycle. Now, back when we proposed this model, we got pushback, even though we had multiple experimental lines of evidence to support the model in this paper. I suppose what you can't visualize, you don't believe. Well, thanks to cryo-EM, that's changed now. By trapping the polyketide synthase module in its pre-elongation and post-elongation states, Dylan and his partners in crime have been able to see both the turnstile open and the turnstile closed states at nearly atomic resolution. And here's the difference. Here you've got the turnstile pre-elongation, turnstile open state where the acyl carrier protein comes in. And in this state, this acyl transferase domain has flexed enough to essentially close the domain for access. So this domain will flex as a result of chain elongation, close this cavity, and now the carrier protein can come in over here. And then that flex state stays in that state for long enough for the whatever stuck on the carrier protein to go through the other follow-on chemistry, move to the next module, and on that time scale, the protein relaxes back to its state. And that flexing is essentially just a single flexible tether that is bending the system enough. So there's enough energy that is being trapped in this particular state that as a result of that decarboxylative condensation type of a reaction. Now, of course, from an engineering perspective, it's these simple structural elements like flexible loops that give us the most grief, right? So I've probably gone over my time, so I should stop over here. Let's see, the take homes that I'd like to leave you with from this talk are, we haven't really talked about what we've learned about the active site chemistry within these assembly line systems, but I do want you to have reasonable comfort that by now, after 30 years of studying these systems, the chemistry that happens at individual active sites within these assembly line polyketide synthases is pretty well understood. The main things I've focused on in this talk is to point out the importance of good structural insights across multiple length scales. If you're interested in a question that involves a specific active site, you need very high level resolution at that active site. If you're interested in a protein-protein interface that is critical, that is giving you 2 orders of magnitude specificity changes, you need high level resolution at that. On the other hand, if you need to be able to see how these systems are working as machines, you need length scales that are much bigger resolution, and you can pay the price at the level of resolution so long as you've got near atomic type of resolution. And so having these structural insights at multiple length scales is absolutely critical. In terms of pathway specificity, this was a phenomenon where protein-protein interactions, not enzyme substrate recognition, drive specificity, was something we knew even before we had these high-level structural insights. And so the structural insights essentially allowed us to confirm what we had seen before in a manner that was very satisfying. In contrast, being able to see energetic coupling and understand the role of asymmetry, there's no way we could have been able to do that absent structure. So those were examples of situations where structure begets functional insights. And that's, I guess, the beauty of the field we work in. You go both ways, and each way, regardless of which way you go, things tend to be very appealing and pleasing at the end. So let me thank a couple of people. I've acknowledged a lot of people along the way because this is a long-standing story, but much of the most recent work has been the work of Dylan Kogan, a postdoctoral fellow in my lab. And we benefited exceptionally hugely from these 2 collaborations with Charlie Craig's lab at UCSF and Hua Chu's lab at Stanford and at SLAC. And this work has been supported by the NIH in my lab for the past nearly 30 years, for which I'm very grateful. Thanks for your attention, and I'm happy to take any questions.
Dr. Khosla, any questions? We have one question from a student, a very general question, and he wants to know, is it possible for an AT domain to be absent in just one molecule of a multimodular PKS and the polyketide synthase to still be functional?
Yes, yes. In fact, nature has done your experiment for you, Sinduri. Nature has created systems like that, and it's solved the problem you've presented to nature in one of 2 ways. Most of the time, it solves the problem by generating a separate acyltransferase, standalone acyltransferase, your garden variety 40 to 50 kilodalton protein, that comes in, transiently docks in with the acyl carrier protein that doesn't have its own dedicated acyltransferase and deliver a malonate or a methylmalonate to it. Occasionally, there are also systems when nature will say, well, I can't be bothered to generate an in-trans acyl transferase. And in those cases, it'll just recruit one from within the rest of the assembly line. But yes, that is possible. And the reason that works is because from a kinetic perspective, that transacylation reaction in the catalytic cycle I showed you is not rate limiting. It's about 1 to 2 orders of magnitude faster than whatever is typically the rate limiting step in a module.
There's another question from a student, a bit more general, that he would like to know the role of polyketides in bacterial system.
Oh.
And as it is a secondary metabolite, is it only for difference purposes?
It's a good question. Well, so I think if you had asked this question to me 20 years ago, perhaps even 10 years ago, I would have given a different answer than today. Because 20 years ago, or certainly 30 years ago, when I started my lab, the only known systems like this were making antibiotics. And so the general thinking was, this is a bacterium's arsenal for high-end warfare. When a bacterium needs some serious ammunition to be able to protect itself or kill off competitors, it pulls in these assembly line polyketide synthases. I think we now know that that is an oversimplification. There are many systems where bacteria are using these assembly lines to make molecules that are essentially just smart communication devices. So they're your iPhones of the bacteria. I'm sure there'll be other insights one gets. The word you use, secondary metabolites, is particularly interesting because it's one of those phrases that puts you in the realm of philosophy where you can't test your hypothesis. Essentially, what a secondary metabolite means is, I don't know what this molecule does for biology.
Right. Wonderful. Yes, sir. Yeah, we have a question from here.
Thank you, Professor Khosla, for a very nice talk. I'm just curious, you seem to be making a compelling case for asymmetry being important for the function of this particular enzyme. And I saw you gave the example of F1 ATPase perhaps doing a similar thing. But if asymmetry were so important for function of enzymes, why do you think there are not too many examples of this in nature? Have you considered the possibility that asymmetry in this case could have could be doing something other than what you're proposing here?
Yeah, I take your point. It's a very legitimate point, and I'm taking the position today that asymmetry is more than just a bystander, but it's actually a driver of catalysis in these systems, largely to be provocative. Although I didn't show you, we have some interesting functional data too that suggests that that might be the case. Your point about could it be doing something else and not involved in driving the process is well taken. In systems, why nature doesn't use it? So are you familiar with the existence of a class of closely related assembly lines called non-ribosomal peptide synthetases?
A little bit, yeah.
So non-ribosomal peptide synthetases operate along a polyketide synthase carrier protein type of a model. They make peptides instead of polyketides. So they're making amide bonds instead of carbon-carbon bonds. Those systems don't have this problem and therefore probably don't need this kind of asymmetry, because in those systems, translocation is coupled to the exergonic event. So peptide bond, essentially what happens in those systems is you have the nucleophilic amino acid that's stuck on one carrier protein. You have the growing peptide chain stuck as a thioester on acyl thioester on the previous module. And within the module, you have a catalyst that will take the amine of the amino acid, attack that thioester, and make another peptide bond and move the chain forward. So in that case, vectorial directionality is coming from the reaction that has the favorable delta G. And it turns out those systems are not dimeric, they're just monomeric systems. They don't have the challenge where they have to couple the battery step. They have to generate a battery to be able to couple the where the delta G lies, the step in which delta G lies to the step in which directionality has to be imparted. Does that make sense? Now,
yes,
obviously,
yes,
that's just one example, and your point is completely well taken. And I hope before I die or retire, whichever comes first, I can answer the question that you're asking with and hold my head up higher than I am right now, because it's a hypothesis right now. But yes, I take your point.
Thank you. Anybody else?
Yeah, there is a question.
Any more questions from the audience or people who are listening to us? If not, I think we will conclude this session and thank Professor Khosla for this wonderful talk that we heard from you.
Thank you very much, and congratulations again.
Whenever the pandemic is over, we would like you to come to Bangalore, to the department.
It would be my pleasure. I promise you it would be my pleasure. Thank you.
All right. Thank you very much. I now hand over to the students. Yeah.
We thank Professor Chetan Khosla for this insightful talk and Professor D.N. Rao for chairing the session. At the end of day one, we thank all the speakers, including the students and alumni, and the chairs of today's sessions for their wonderful talks and discussions. We also thank the audience for attending today's events. We hope to see you all again tomorrow at 4 p.m. IST, either on Zoom or YouTube. Until then, thank you everyone. See you soon. Stay tuned and good night.
Thanks.